Good morning, everyone. Good morning and welcome to the third day of the Open Session 2022. As yesterday, today we will start with a presentation on fast diseases. And I would like to thank you for the great participation that we had yesterday here in the room, and also the colleagues attending online. We had yesterday 930 colleagues attending through YouTube. Our first keynote speaker today is Nick de Regge. He's a bioengineer with particular interest in vector-borne diseases. He has the responsibility for the URL on Capripox viruses and act as a deputy coordinator of the URL for foot and mouth disease. He and his team are involved in several research projects, particularly in Capripox virus host interactions, transmission routes, vaccine safety, and efficacy testing. I'm very grateful that Nick came here today with us, and he will provide a presentation on a headline of the Capripox events occurred globally in the last 2 years. Thank you. Nick, the floor is yours.
Thank you, Fabrizio. Good morning, everyone. Thanks for being here so early for the first session. Also, good morning to everybody who's following us online. So, first of all, I would like to thank EuFMD for the opportunity to present you something on Capripox viruses, one of the fast diseases for which we are the European reference laboratory at Cienzano in Belgium. So in my presentation, I'm going to try to tell you something about the main epidemiological aspects of those viruses over the past years. But before going to that, I will provide a short introduction. So for my presentation, we're going to move away from all of the RNA viruses we have been talking about the last 2 days, and we're going to move to the pox viruses. And so the pox viruses are quite a large virus family with viruses in many different genera. And I think the pox viruses that you know probably best are belonging to the orthopox viruses, because those contain smallpox virus, cowpox virus, but also the monkeypox virus, which caused the epidemic earlier this year. But for my talk, we're going to focus on the Capripox viruses. And within the Capripox viruses, there are 3 different viruses. You have lumpy skin disease virus, sheeppox virus, and also goatpox virus. And those are large DNA viruses of about 150,000 base pairs, and they share quite some high nucleotide identity. But if you look at the phylogeny, you can see that the viruses really cluster separately, so it are truly 3 different viruses. And what's important to mention is that these viruses are quite host-specific. So lumpy skin disease is mostly found in cattle, goatpox virus in goats, and sheeppox virus in sheep. So what can you expect when these hosts get infected by their respective pox virus? So after an incubation period of a few days, most often you're first going to see swelling of the lymph nodes, and then the viruses can develop quite some fever, which can be rather high. And this is often associated with a decrease in milk production, and you also often have a general depression of the animals. And then a couple of days later, you can see the typical pox lesions caused by these viruses. So in cattle, these are the clear lumps, the huge nodules, which can be seen on the head, also on the body, on the udder, and on the genitalia. I need to say these clear clinical signs are not present in all of the animals. Some develop smaller lumps, just a few lumps, or there are also existing subclinically infected cattle. These nodules can also be found in sheep infected by sheeppox virus, but there the nodules are generally smaller. You can find them on the head, as you can see here, and the bottom. No. And the place where you find the nodules in sheep for the first time mostly is under the tail and around the perineum. And what's also important to mention is that the lesions are not only found on the surface of the animal, but also internally. So you can find them in the respiratory tract or in the gastrointestinal tract. So they really contribute to the disease that's caused by those viruses, to the morbidity, and sometimes they also lead to mortality. And then the main difference between lumpy skin disease virus and goatpox and sheeppox virus is the way they are transmitted. So for lumpy skin disease virus, this is considered as a vector-borne disease, and several vectors have been implicated in the transmission, and it's mainly by mechanical transmission. That's contrast for sheep and goatpox, which are not considered as vector-borne disease, but there the main transmission is by direct transmission via aerosols, by close contact between the animals and contact with saliva and nasal secretions and scabs which contain high amounts of infectious virus. And then the sheep and goatpox can also be transmitted by indirect transmission, so humans which bring the virus from one animal to the animal. There are also fomites, like trucks that can move between farms. And those pox viruses are also quite stable, and also in the environment, so that can also play a role in the transmission. Okay, that was a very short introduction. Then I'm going to move to the epidemiology of the sheep and the goat pox. So in this map, you can see the endemic region where these viruses normally are found. So you see that huge parts of Africa are endemic for the viruses, also the Middle East and large parts of Asia. And the incursions of these viruses in Europe, the last decades have been very limited, with a few outbreaks in Greece and one outbreak in Bulgaria 5 to 10 years ago. So this is the same slide, but in blue it shows the countries that have notified outbreaks of sheep and goat pox in the WAHIS system of the WOA. And there you see that several of the countries in the endemic region have reported outbreaks, but what is of course most surprising was the outbreak, and I'm sure that most of you have heard of it, was the outbreak of sheeppox virus in Spain a little bit over a month ago. So this outbreak really came as a big surprise because the virus had been eradicated from Spain around 50 to 60 years ago, and now all of a sudden it was detected in the south of the country. So in the, in the south of the country, in the province of Granada, where vets detected the disease in sheep on a farm, and the animals showed the clear pox lesions, and there had also already been some mortality due to the virus. So that indicated that the virus was probably already there for a couple of days or maybe even a few weeks before it was detected. So the disease that was then very rapidly confirmed by the National Reference Lab, and then all measures were put in place to start to contain the outbreak. Going from stamping out of the infected farms, installation of protection zones, and a lot of epidemiological investigations. And these allowed to identify 8 more outbreaks in that region, which all had more or less epidemiological links to the index case. Then there were also some outbreaks about 200 kilometers to the north. And there, the disease was detected in an assembly center for sheep, and there was also a clear link with the first cluster in the south because animals had been moved from the south to the north. So that was the most probable route of introduction over there. There, the same measures were implemented, and they found 7 more outbreaks in that region. So in total, till now, 17 outbreaks have been reported by Spain, and the good news is that the last outbreak now dates from more or less 10 days ago, so we are hopeful that the disease could be contained for the moment, but it will definitely be important to follow that up the coming weeks and even months. So I think this incursion of sheeppox in Spain learns us a very important lesson, because it shows that any of our countries can be confronted with the introduction of one of the fast diseases, and that the work that we are doing on awareness and preparedness for disease is really, really crucial, and that also all the efforts that are done by EuFMD, by FAO, by WOA are really important and should be go on. Okay, then for the last part of my talk, I'm going to move to lumpy skin disease virus. And before I'm going to tell you about the most important events, I want to present you the epidemiological situation as it was till around 2017. So lumpy skin disease has first been detected in Zambia around 1930, and from there it spread to South Africa and then to many other African countries. So most African countries are now considered to be endemically infected with the virus. Then beginning of the 2000s, the virus spread via Israel to the Middle East, and over a period of 10 years, it reached several other countries in the region, and including Turkey. And then we end up in 2015, where most of you will remember that the virus reached Europe, and in the Balkan region, and where Europe did huge efforts to eradicate the virus, which was finally successful by a mass vaccination campaign. Around the same time, the virus also spread to the Caucasus, and it went into Kazakhstan and into Russia. And when we look at the virus strains that were circulating till then, the picture seems fairly easy, because the strains that were first detected in Africa, in Kenya, had a high similarity to the strains that were also present in the Middle East and into Europe. You can also see this in this phylogeny, where all these strains cluster nicely together as classical field strains, and they form a different clade from the vaccines that had mostly been used. And maybe it's important to say, the vaccines are developed from the so-called needling strain of the virus, and which has been passaged multiple times through cell lines and to embryonated chicken eggs to get attenuated. But this picture drastically changed in 2017 when colleagues from Russia reported the finding of a recombinant strain in an outbreak that they had in the Saratov region. So that's over here. And so they described that they had an outbreak in the field, and the strain they detected, it seemed to have the backbone of the vaccine strain, but within its sequence there are multiple regions which had sequences that were rather related to classical field strains. So it's important for the rest of the talk to remember that these recombinant field strains, that they cluster in phylogeny more closely to the vaccine strains, but they are in fact behaving as wild-type strains, and they can cause clinical disease in the field. Okay, then this slide summarizes what has happened afterwards. So when there's the huge epidemic that has occurred in Asia. And based on the colors, you can follow a little bit how the virus spread. So in 2019, China and India reported cases of lumpy skin disease virus. The year after, also Nepal, Vietnam, Myanmar reported cases. Last year, we had Laos, I think also Malaysia, and this year it spread even further to the south and it infected Indonesia. And also the last 2 years there were additional countries in the Middle East that got infected, like Afghanistan and Pakistan. So disease has gone through most of Southeast Asia in a very huge pace, and it caused a lot of mortality in the animals, but also a lot of economic consequences and a negative impact on the livelihood of many of the farmers, which really depend on their animals for their income. So one of the questions that came, of course, up around that spread through Asia is, where was the virus coming from? Was it virus that spread from Russia further to the south, or was it rather virus that was coming from the Middle East and spread further to the east? And we can have a quite good indication on that when we look at the strains that were circulating, Because after the first outbreak in Russia from Saratov, they have reported several other outbreaks where the recombinant virus was present. And then in the meanwhile, also some viruses have been sequenced from Asia, like from China, Vietnam, Thailand, and so on. And all viruses that are found over there all belong to the recombinant strains, and they nicely cluster together. So most probable route of spread through Asia is virus that was coming from Russia and spread to the south. The situation in India, Afghanistan, Pakistan, that's a little bit more difficult to assess. There are a limited amount of sequencing data, which are a little bit contradictory, so that will be definitely something that we need to follow up closely. And it could also become a very important region because it can be a place where the classical field strains come together with the recombinant strains, and then we will have to see what will come up from that. Okay. I have been talking a lot about recombinant strains, so I wanted to give you a little bit of background on those. So recombination of lumpy skin disease virus, as for other viruses, can occur when different strains infect the same cell. And when during replication, a new strain detaches from its template together with a DNA polymerase and then reassociates with another highly homologous strain and continues the replication. And so you can end up with a virus here. The first one is a single recombination where you have a part of the first strain and a part of the second strain. But in reality, the recombinants for LSDV are much more complex, and it seems that multiple recombinations have taken place. And this picture shows what's happened in reality. Because what's interesting is that all the viruses that are found in Russia and also in Asia, it seems to be possible that there are recombinations by two vaccine strains, namely the needling vaccine strain and another vaccine strain which is called the Kenyan sheep and goat pox strain. So by doing multiple recombination of those two strains, you can make up all these recombinants. Okay, you can imagine that when those recombinants popped up, that they raised a lot of questions. So the main question is this possible that this has occurred during natural recombination in the field? And so also quickly, it posed questions to the safety of the live attenuated vaccines that have been used for lumpy skin disease virus. But so luckily, Chris de Klerk, who was at that moment head of the EURL, and I think you all know Chris, he was able to lay his hands on the vaccines that had been used in Kazakhstan just before the Russian colleagues reported the recombinant strain. And he decided to do the classical vaccine control that we do in the lab. And it quickly became obvious that there were some problems with that vaccine. So first, they were tested in a series of PCR tests, which shows that indeed the needling vaccine strain was present, but that probably also other field strains were present in the vaccine, and classical PCRs even showed that there could be sheeppox or goatpox in the vaccine. They had then a more closer look by some partial sequencing of 6 different regions of the genome, and this confirmed those results. Needling vaccine was present, also a field-type goatpox strain was present, and there were even suggestions that recombinant strains were already present in the vaccine. And this has then recently been confirmed due to next-generation sequencing of that vaccine, which showed that it was not only one vaccine strain that was present, but 2, the needling strain and the Kenyan sheep goatpox strain. There was a field strain of goatpox present, and also the recombinants that are now found in Asia, some of them were already completely present in the vaccine. So it provides a clear, rather strong evidence that the strain that is now circulating in Asia that's causing the huge epidemic was released in the field due to a badly manufactured vaccine. So that's very important, and it also stresses the importance of doing an independent quality control of those vaccine batches. Okay, then I just have a couple of slides more. So the recombinant strains, they are also having important impact on the diagnosis of the disease and on the control of the disease, because labs that would receive samples that are suspected from lumpy skin disease virus will normally first test them in what we call a pan-Capripox PCR. So it just detects if the genome of Capripox is present, and those work very well to detect the field strains, the vaccine strains, and also the recombinant strains. So if that PCR is positive, normally the labs will do a couple of so-called DIVA tests. And these tests allow to differentiate whether the strain that's present in the animal, is it a vaccine strain or is it a field strain? And these have shown to work very well for the field strains and also for the vaccine strains. But there is a problem with the recombinant strains, because those DIVA PCRs will say that your recombinant strain is either a vaccine strain or it will not even detect the strain. So it could be that the lab will say, okay, no problem, it's just a vaccine that's circulating, while it's in fact a wild-type strain that can cause the epidemic. So that can hinder the progression of the measures that should be implemented to contain the disease. So that's why we recently developed a new DIVA PCR, which also detects the recombinant strains as wild-type strains. This has in the meanwhile been validated, has also been communicated to WOA, and it should be published normally rather soon. And this is my last slide, where I just want to mention that I think the time has now come that we really look to these recombinant strains in detail, and that we check the virulence of these strains, that we look how they are transmitted, and that we check if the current vaccines, if they protect us good against recombinant strains as they did against the classical field strains. And this, I think this is important because there are some preliminary data in literature which show that the recombinant strains could be more virulent than other strains, and also very important, that there are some indications that the recombinant strain could be spread by direct transmission, so that it should no longer be really a vector-borne disease. So these are preliminary data based on a limited number of animals, so I think it's something we should look into detail in the near future. And so I've come to the end. Thank you very much.
Thank you very much, Nick. Thanks a lot. A very relevant presentation, considering, as you said, the impact on particularly lumpy skin disease on livelihood all around the globe. Now there is time for a couple of questions, if you have. Perhaps the first question would be, are there any hypotheses on the introduction on sheeppox into Europe, in Spain?
Yeah, I think that's the question I get most of all. And I think, Spanish colleagues can correct me if I'm wrong, I think there is no clear idea where it's come from, but there are some hypotheses. So one main hypothesis is that it could have been introduced from Northern Africa, because there is quite some interaction between Spain and Northern Africa. There are many trucks that are coming in. There are workers from Morocco, Tunisia that work in Spain, so it's a suspicion that the virus came in via that route. And we also get some indirect indication that that could be the case due to sequencing that we have been doing. So we received samples, we determined the whole genome sequence, and there, based on the whole genome sequence, the strain from Spain clusters with strains from Egypt, but there are not a lot of strains in the database, so that's difficult. But when you look at specific regions where there is more information, there we found that there is really an insertion, which can also be found in strains that are found in Morocco, in Egypt, and also in Turkey. So it gives some extra indications that it could be from that region, but of course we remain careful in giving conclusions to that.
Thank you. Question on the back from Keith.
Thank you. Very, very nice talk. Thank you for that. I'm just wondering if you can tell us more about what you think is the portal of entry, how sheep and goat pox enters the body. What's the sort of entry route? Because I'm sort of intrigued by finding Siatko is here and others of the amount of virus in saliva. I think for lumpy skin disease that's true, and I think that's also true for. So is it possible that we basically have a direct transmission and possibly through the oral epithelium?
Yeah, I think for sheeppox and goatpox, it's clear that there's a direct transmission via probably the oral or the nasal route, because of contacts with aerosols that contain much virus, or with saliva that contains much virus. So that seems to be the most probable route of entry.
Last question here in the front. Michelle.
It's not a question. I just wanted to add that it's possible to pass pox virus by clipping machines, through the clipping machines.
The clipping. Okay. That's a good question. I know we have detected pox viruses in ear notches and stuff like that, so probably it could be possible. Yeah. Yeah, that could be, probably it's quite resistant, so you could probably also detect over there, but never test that.
Thank you very much, Nick. Thanks a lot. An applause for our colleague. And we will start now the fifth session of the Open Session 22, which is focused on risk assessment and modeling. The session will be chaired by Germán Casares Garrido. who is the member of the Standing Technical Committee. He's also the head of the Epidemiological Department, Animal Health and Traceability Ministry of Agriculture, Fisheries and Food in Spain. And Herman has been assisted in the preparation of this session by Roberto Condoleo and Melissa McLaws. Melissa will also act as moderator of the panel. She's an epidemiologist working with FAO and EuFMD since 2010, I think. And yeah, Silvia Baluca will also assist in moderating the session. So please come on the stage, and we can start this session focused on risk assessment and modeling.
Okay, good morning. Welcome to everybody, those in the room and those following us by internet, by technology. Well, as you know, diseases evolve in a very complex world that we have. The evolving situation is a product of the interaction between the host, the agent, and increasingly faster changing environment where the human factor is quite key. So in this context, to be able to characterize the risk and to know or to try to know where and when the risk can change, can increase, can reduce, is very important. And for that, because not just from a point of view of epidemiology and management of the disease, but also from an economical point of view, because the resources put on hands of the official veterinary services are constantly decreasing, and we have to make a more efficient use of this, and the risk-based surveillance is very important. So in this context, technology and innovation is incredibly important. So it made possible the modern epidemiology, computers, softwares, and it's a field that is constantly evolving, and today we are knowing about some examples of this evolution. So the session today is on risk assessment, that is an important field within the epidemiology, and modeling, that is another technology that is put for the risk assessment purpose. The title of the session is to showcase advances and innovation in risk analysis and modeling as suitable tools for using data and transforming them into meaningful information to assist decision makers to manage the risk of foot-and-mouth disease and similar transboundary animal diseases. And for that, we have the pleasure to have here and the honor to have three keynote speakers today and 4 short presentations. The first one is Dr. I hope to pronounce well, Thibaut Porphyre. He's head of the industrial chair in veterinary public health at the Veterinary and Agricultural College of Lyon, France. Please, doctor, go on. The floor is yours. Thank you.
Good morning, everybody. Thank you very much, Chair, for this invitation. So this morning, I was asked to talk about data and the importance of data. So as modeler, and we heard pretty much the whole day yesterday and the day before about the importance of modeling and for risk evaluation and control purpose and control design. But we didn't talk much about the data sets that we use for modeling, the quality, how they talk to each other, and so on and so forth. So the data is maybe one of the most important things when we are developing models. We need to make sure that they're of qualities and they're working well together. There is quite a lot of interplay also between knowledge, data, and models. There's a cycle between those three. And as a modeler, and when I was a little bit younger, I always have that utopia, or at least this belief that we're going to get some data, and we're going to spend all our efforts in models, in developing the models, the best model possible to get good uncertainties and those kind of things. And I had this kind of structure that was actually published recently, but I had that in mind was, all the data was at different level working together, and we can just focus on developing the models that are relevant for research, science, but also for policymakers and industry partners. However, with more experience, I realized that actually is very much the landscape of data is very big mess. There's a diversity of data type and data source. There's diversity of geographic and temporal scales. There's diversity of quality of records and diversity of data owners and data managers. And I point out that data source doesn't mean it's the same number of data owners and data managers, and that data types can be also not equal to the number of data sources. So there's a lot of diversities, and sometimes we can get lost in all the data that we can have access, or we know that's circulating, and we might have access, and data that we know exists, we don't have access, and there's also a lot of space where we think they should have some data, but we don't know. So some of those data can talk to each other. We know sometimes it's possible, sometimes it's possible, but we don't know how well. And so when we start modeling, we usually ask those kind of questions. What database contains what I need to answer the question that have been asked, and also what information are available in these data sets. So we don't know the details, we need good metadata. What are the collection of reporting methodologies? Because, having data doesn't mean they are good data. How do we link these databases? And so all of that, we try to answer those questions in order to minimize the impact of the garbage in to avoid the garbage out. And when we're younger modelers, we are overwhelmed by that diversity and all those questions. So just to let you know, there's a lot of systems now that exist of very good quality and that try to answer some of the questions. So you do have some that are specific to questions, like the one in mission rabies, where they're purposely designed integrated data sets and integrated systems to answer their disease control questions and missions. There's others for surveillance, like the one developed by Osvet for Indonesia. They combine a lot of different data sets together and make it useful and help also to collect better in having some interrelations between the different data sets. Obviously, we're in France. I had to put some very good data sets on surveillance and integrated system for surveillance, in both in public health and in animal, plant health, and food safety. So and that is a 3 platform funded by the Ministry of Agriculture for surveillance, and they all have very good system, integrated management systems. But the systems are very important because they get a lot of benefit out of them. They allow automatic visualization of the data, and also because they're interrelated, of the interrelations between the variables that can explain and the observation of disease and health issues. And therefore, they improve the operationality of the information. It improves policymakers to take informed decision. I didn't say good, I say informed. And also, it helps them to improve communication with the public or with policymakers, other policymakers, with the industrial partners and different customers. So it is very important to have them, and the impact of visualization is perfect. But that is because these they've been designed on the FAIR principles. The FAIR principle is to make sure that they are findable, so we know they exist, the data they exist, they are structured, they're accessible, so we know how to get them, they are located, they all have a DOI number or equivalent, so we can find where they can be downloaded or found the metastructure. Interoperability is to know what is the key variables, how we can talk to each other. There's more technical aspect out of it. They can be used in different platforms, and they can be reusable, meaning that we can use them even if they're updated and there's good version of it. So there's a very good YouTube movie from the University of Miami that highlight those kind of things and how we built models and data curation. But to be honest, the FAIR principles are facing very difficult things with reality. It's time-consuming, it's not quick to develop a FAIR database, it's not cheap, therefore. Because data curation needs time and is hardly available during outbreak response. It needs benchmarking, it needs reliable testing to make sure the data is of quality, and also we need sometime confidentiality agreements and data sharing agreements for each of the single data sets that we put in our system. We means that we are very much dependent of lawyers and the lawyer time, which can be variable and very long. So there is another factor that is very important. It can also take time when developing an integrated management system, is to make sure that it's embedded within the socio-ecological system that is deployed. Bear in mind that it's not only data sets that we, from surveillance or we get regulated data sets, it also can be from research, can be from citizen communities and collected apps. So we need to make sure that it's been embedded into the socio-ecological system, so there's no harms on the local communities, and also we make sure that we've got the information we need. To that, and also there's time consuming, but also it's future proofing. So data change in time. And so why that changing time? Because they change because the livestock industry changed, they change in the monitoring and reporting of methodologies. We saw that many times with the COVID response. And this changed due to the learning of the data. So once you're modeling, when you're integrated, you have models, and they inform the data, they inform the policymakers, they ask questions, and so on. So there's a cycle of FAIR data, and it's what we call a data model policy lifestyle between the policies, the models, and the data, and they inform each other, and therefore the data evolved and changed. So you need a very good versioning controls. To do that, we need, therefore, a data ecosystem linking the data, the models, and all the infrastructures that are related and use the data systems. And the data ecosystem needs to be effective, efficient, and equitable. Make sure that everything is, we are not having issues with communities and so forth. It's very important because we can very easily move a data system into a negative benefit, less benefit and harm communities if we are not collecting and modeling, analyzing the data correctly. And so we need to make sure that it's equitable. And because the data change a lot, we need a good traceability between the inputs and the outputs and the models. So make sure we get all of that. So recently we've got I participated in the development, but it's not me who've done all these clever things, definitely not me. But those very clever people developed the FAIR data pipeline. That is a pipeline that is based on GitHub, which is therefore open access, that allow a good traceability between inputs and outputs and the models that create those outputs. And you get a good control of the version of inputs and version of outputs and the version of the models. So the policymakers can make a fair choice as, oh, those outputs come from this data. And so if last week, and if it's a tool, all data sets, they can question the quality of the outputs. As it is right now, sometimes it's difficult to question the model's outputs. And it's very auditable. So another issues I wanted to highlight in the fair data pipeline is that although it's a very good start, it's open access, it helps to have a good traceability of the system. It's been developed for COVID, not during COVID, so we were not used at all during the COVID system because it takes time to develop these kind of things. However, it's getting used in the EPIC, so it's the Center of Expertise on Animal Disease Outbreak, which is funded by Scotland, as a base for their system to inform policymakers. And so what is important in that fair data pipeline is that we still need to manage the issues of confidentialities because, okay, you've got open access and not so open access in our world. And how to integrate data, various confidentiality status, but still being usable to most modelers is a big, strong question that we don't yet have an answer. Obviously, you've got protected folders and everything, but there's an issue for validity and repeatability of the science and research outputs. So how good is our work? And also, each data can have a data cleaning pipeline, and each data cleaning pipeline may be different from one modeler to another, and therefore difference may be due to the cleaning of the data rather than anything else. So there's some people, especially in the US, use synthetic data sets to go beyond that, not because it's an issues of confidentialities. It's mostly they don't have the data at all. But there's some issues related to synthetic data sets is that you're losing the fine grain of detail that can inform policymakers. On the right, I highlight when you introduce disease in African swine fever in British industry and it's letting it spread through movement, the patterns of spread of risk may vary where you go. And if you're using synthetic data sets, you may lose that differences of patterns, always showing something similar. So you may not give the right answer to the policymakers. So I will finish my talk talking about cloud computing a little bit because a lot of people will talk about it and have heard about cloud computing. It's actually a very good environment to support data storage, integration, analysis, and communication. However, there's costs and benefits in investment. So starting by the benefit, and there are good benefits. It greatly facilitates communication and ownership in data collection. It allows a very good integration of the data sets. It has, in some situations, a very high computing speed in some cases, so the models involved in cloud computing can run faster in some situations. And it's also a sustainability of infrastructure. So it has a cost, a quite hefty cost sometimes, but it has to be balanced with the cost of maintaining your own servers and everything. That goes with the downside, I think, which is the server location and legal environment around them. So you have to be prepared that if you're working on cloud computing, the data will be somewhere else. It can be in the US, it can be in Europe, it can be elsewhere. And it really needs to have a reflections on where is your data stored, and I am fine with that or not. And there's two last things that are important to think is that operability and currently developed models of the cloud environment. So we might lose some historical information due to not being able to transfer current models that are being used and are quite high tech, but they are difficult to integrate within a cloud system. And therefore, there is also a basic issues due to poor economic settings where internet access and bandwidth is important. So all of that can be important when we're talking about cloud computing. And now we're going to finish on that. Thank you very much.
Thank you very much. We will have all the questions at the end in the panel discussion, so we are going straight away with the next presenter, that is Mrs. Amy Delgado. She is veterinary epidemiologist and director at the Center for Epidemiology and Animal Health is part of the USA Veterinary Services. Okay, the floor is yours, Amy. Thank you.
All right, thank you very much, and good morning, everyone. It's a pleasure to be here. I appreciate the opportunity to get to share with you. When I was given the topic and asked to present on successes and pitfalls, I thought, well, that's easy. I've hit every pitfall you could hit in modeling since I began working as an FMD modeler many years ago. But as I thought more about it, I thought, you guys, you don't want to hear those stories that are really unique to a country. Every modeler has to live within their own data ecosystem. They're integrated and working with their own policymakers. They're using the tools that are available to them at the time. And that creates a unique set of circumstances that often shapes what we can do, how we communicate our results, and how we engage with our policymakers. And so as I started thinking through this topic, I thought, maybe it's time that we take a pause and we look ahead, right, at kind of a bigger picture. So not the story of what Amy's doing in FMD modeling and working in the space, but really the story of where are we going as modelers in the modeling community? What are we learning? What are we seeing ahead on the horizon? What should we be thinking about? And so that's what I'm going to focus on this morning. When we think about the last few years, COVID-19 has brought modeling again to the forefront in a very visible way. There's a lot of things that have been happening in the modeling community, both good and bad. I mean, I think you've seen the full gamut of what can happen as people employ models to support decision making. I'm an optimist. Thibaut and I talk about this often. He's the cynic and I'm the optimist. And so when I look at that space, the diversity of things that are happening, the people who have engaged with modeling, the people, new ideas, new departments of universities that have come on board to say, how can we support, how can we think about the issues of disease transmission within communities and optimal response strategies. I think, what an opportunity. Look at all these fresh minds and these cool ideas that are coming to this space. And so I think we have an opportunity to take advantage of the energy and the momentum that's happened because of COVID-19 as we think about fast diseases. And where are we going? What are the tools that we want to have? What are some of the questions that we should be asking that we aren't? I'm always amazed when I think about FMD modeling, like we're still working on the same questions. I look at some of these posters and I think, I modeled that exact set of scenarios like 10 years ago for the US, and we kind of had an answer and we were like, all right, yep, moving on. And like it comes up again, right? So it's time to maybe think about some new questions. There's still really hard questions that we just haven't answered for particular FMD control and other fast diseases. So there's no shortage of work to come, but it's a great time to sort of sit back and think, are we asking the right questions and how could we be thinking differently? I grabbed a series of papers, and so you'll sort of see these as we walk through. None of this is my work, so I just want to be transparent. These are things that other people have done that I found interesting and intriguing, and so I'm sharing that with you. They were all interpreted through the lens of my perspective. As a modeler, as now a manager, the director of a center, my space is to work and providing scientific support for policymaking. That is the mandate of my center. And so this is all through the lens of how I see the world. This first paper was looking at global research activity on mathematical modeling of transmission and control of infectious diseases. And so you can see here this trend, and it's very similar to a graphic that we saw earlier this week, at this big upshift in publications. And this is a bit broader. It doesn't actually even include agricultural animal diseases. These are human diseases. But if you just look at what happened from 2019 to 2021, the number of people who engaged, the number of publications that were coming out in this space, it's remarkable. These are the kind of trends that we don't often see. And so what does that mean, right? How many of these papers were useful? How many of these papers were timely to actually inform a decision? How many of these papers actually asked practical questions, right, that could impact a response, that a policymaker could interpret and leverage and use to support discussions and engagement around policy? I did not dive into those 1,000 papers to answer those questions, but I suspect that the vast majority of them, maybe not the vast, but many of them probably didn't meet those marks, right? And certainly I understand that by the time you're in a publication, you right? You've missed the window, right? You can't wait till you publish to bring it to a policymaker because they've already made a decision and moved on with their life. And certainly the mark of a publication isn't its immediate impact, right? So a lot of things, we hope, these will go on to have important impacts in our scientific understanding of disease transmission and control. But when we think about emergency response and we think about designing and working in preparedness and emergency response planning for fast diseases, we need to be innovating now, right? We can't wait months and months or years and years for those things to come, right? So we need that integration, we need that work to happen. But nevertheless, I'm excited by this graph. I'm excited by the new minds and the new faces coming into modeling and working in this space and looking to see what comes. What can we take advantage of? What can we be learning? So the next couple of slides, I just pulled out things, again, that I thought were interesting. Some work that's happening that I think maybe leads us to look at the world through a different lens, look at modeling through a different lens, and think about what could we be doing. So I kind of clumped these into buckets, and I'm just going to talk through them with you guys. So this first bucket is a couple of papers that look at sort of this global scale, potential distribution of African horse sickness from 2020 to 2060. That's a time horizon that we aren't often working on. I think that's a really interesting and new way to think about disease risk. The second, potential geographic extent of African swine fever across the world based on risk factors, right? And if you worked on these papers and you're in this room, come talk to me later because they are really interesting work and happy to connect with you. So these two papers, I think, highlight again, right, that we understand that fast diseases are operating on a scale that's independent of our country's boundaries. That's what they're defined for, right? But how do we as modelers begin to engage around that thought in a more meaningful way? And how do we deal with the data ecosystems involved in modeling at that scale? Lots of questions, lots of work to come. And it certainly varies. You know, what you think about within the European context is very different than what we would think about in the US context, right? And the pressures that we're under and the risks that we face. And so each of these have to be taken home and chewed on for a little bit to think through. This next set of papers, I've kind of labeled local complexities and just sort of highlighting some people who are thinking about things maybe a little differently in that space. So this first one is looking at multiple species animal movements and all these different network dynamics, and then how does that impact targeted control actions. In the US, this is a big thing. We are constantly being asked to do incredibly precise types of control actions, right? So no broad sweeps. It's like a very targeted way of thinking about disease control. What can I do on this type of farm, you know, of this size to mitigate disease spread? And those strategies have to be customized because we have very diverse, very large farm sizes in the United States. So targeted control actions are really important for us. And because we're really focused on business continuity during outbreaks, it brings another layer of complexity. And I think we still have a lot of work to do there to understand how do targeted control actions impact different segments of the population, and how can we help fine-tune our response to really get the most bang for our buck. The second paper's preventing multiple infectious diseases, modeling that. That's a complicated space, but I think particularly with fast diseases and others, investments in biosecurity, good management practices, all of those things have a lot of benefit for multiple diseases, and we struggle to articulate that. We struggle to collect data on the value of biosecurity, right, and the preventative effects of biosecurity. It's a big gap for us, and so how do we continue to engage and improve the way that we model multiple disease impacts? Next is an interesting paper that talks about using different models for herds of different sizes. That resonates with me. Working in the United States, we have herds of different sizes. We have massive herds. We heard from Aldo earlier this week. He said, you know, they killed a million chickens in this contiguous coal approach. And I thought, we kill 1 million chickens in one farm. You know, we kill 5 million chickens in one farm. The farm sizes in the United States are really diverse, and we do need different modeling approaches for different herd sizes. That's not true in every country, but in Slovenia, they did this work and looked at and said, we need to think about these things differently and how transmission occurs. The last 3 papers are all related to airborne spread. And some really nice work there. Certainly airborne spread of foot and mouth disease is nothing new. We've certainly seen its impact. We've looked at exhaustively. Graham has built a really nice tool looking at some of these things. But I thought this last paper was really intriguing by Koldrick et al. That talks about dispersion and deposition of respiratory emissions. This filled a critical research gap, experimental gap for our understanding of respiratory emissions, particularly related to COVID-19, where suddenly the mask debate brought all of the scientific gaps to the forefront. And so I think we still have a lot to learn about airborne spread. And it's a space that we need to think about. How do we deal with that in our models? Simply, with complexity, either way, I think there's interesting things happening and more to come. All right, so this next group of papers I've kind of labeled Society, Economics, and Equity. And Thibaut touched on this briefly and the need for equity in what we do. But I think in this space, there's a couple of really compelling things happening right now in modeling, and I just wanted to maybe walk through some of those. So at the top section, I've kind of lumped these into economics, and how do we think about epidemic recovery? I don't know about you, but we've never modeled epidemic recovery within my center. That's not a space that's typically the purview of veterinary services. However, we recognize that disease impacts have disease outbreaks have huge impacts on communities. And how a community recovers is almost just as important as how that community experienced disease, right? So we want communities, we want countries, if they experience a disease, or maybe an industry within a country, to recover in a way that leaves them more resilient, right? That enables them to avoid disease in the future, right? To improve things as they go through an outbreak. Our emergency response folks have a saying, never waste a good outbreak. Learn and adapt as you go and make things better. There's no time like the present, right? To fix something when you're in an outbreak. So it's an interesting paper. Encourage you to check it out. They did an expert elicitation with people involved in community resilience, disaster recovery, public health, infectious diseases. Really interesting framework of how do we think about epidemic recovery and community resilience. The next 2 are kind of more economics interest, maybe optimal allocation of test kits. This was certainly optimal allocation of vaccines for COVID, a very difficult issue, and I would say that optimal allocation of EuFMD vaccines in the United States is a nut that we have not cracked. That remains a struggle for us, right? Because what is optimal? What are we optimizing? I think that's the question every modeler struggles with, right? What are we going to do and how are we going to get there? So again, another space we need to think. The last set of papers are kind of looking at society. So here you can see effects of misinformation, effects of risk perception, population sentiment analysis. This is drawing heavily on social media. And so these are all spaces where I think much of what we need to move into is understanding people, as much as we understand diseases and animal movements and connectivity, right? So there's some great work happening. We heard from Mike Tildesley group. I think Thibaut is also kind of working in this space. We've been working with the University of Vermont in supporting some agent-based model development to understand the effects of information, connectivity, and risk perception and people's choices on disease spread, particularly for EuFMD. So continues to be an interesting space. And then the last is rethinking our approach to inequality and infection risk. And so I'm getting short on time, so I'm going to run through these really quick. But this is a paper that looks at how do we address the socioeconomic divide in modeling. And, how we address socioeconomic status, how we think about that, I would say that most of the time we just don't. For animal diseases. In fact, we're often told, don't worry about the economics, focus on the disease control. But I think we do ourselves a disservice by ignoring the socioeconomic divide. Not everybody has the same ability to implement preventative measures. Not everybody is impacted equally by either the disease or the disease response. And so how do we get how do we advance in those conversations? So this paper outlines this figure which walks through a couple of different ways to address socioeconomic status in our models. They come up with some really neat ideas. They look at shelter-in-place policies and how those have disparate effects on different groups. And they recommend, there are different things you can do between target, duration, and timing of control measures to help mitigate those disproportionate effects. So this is a space I don't think we've really leaned into in terms of animal disease modeling. And so another interesting place I think we need to be thinking forward about equity from the data to the models. Lots to do. And then this is the last paper that I'll highlight. So this is an agent-based model looking at the effectiveness and, again, equity of policy design features. When I was reading through this, these were the highlights from their abstracts, and I'll let you read those. But I thought, oh, that's so true. It's just as true for us as it is for human health and those working on COVID-19. We have to work with real people. It's not the animals out there making decisions. It's people making decisions about how animals are moved and managed. And so when we design policy, when we model policy options, we have to recognize that there are a lot of things influencing the success of what happens on the ground. And when people are making policy, oftentimes what they're juggling internally is this space, right? So we might say, well, the model shows, and they're saying, yeah, but people think, people want, people are able to do all of these different things in these different ways. And so at the end of the day, what you get is kind of a negotiated outcome, trying to navigate through this space. So all that being said, when we're working in modeling for policy, how do we set ourselves up for success? I'm just going to leave you with three little things. So one, build relationships. Modeling can't be done in a vacuum. Policy making is not done in a vacuum. Policy making in each country is unique. The environment by which you decide how you're going to control diseases and different strategies you're going to employ, I recognize is very unique. But almost uniformly, it's not done in a vacuum, right? So in the United States, we heavily engage and negotiate with our partners, our industry, with other parts of the government, with our state animal health officials. We have a very large, diverse set of actors and players who are speaking into this space with their needs and their expectations. And so as a modeler, you've got to have those relationships and be a part of those conversations to be trusted, to understand the complexity of what they're trying to navigate in making a policy. And I think watch those conversations evolve and find the strategic points when you can bring modeling results to the table and help inform that conversation, that debate. And so it really is about relationships. Data is about relationships, all of those sorts of things. But if I had to say one thing, take the time and build your relationships. The last thing that we want to see, working in this space is somebody unknown and unconnected come in and say, I have the answer. And you're like, yeah, no. Nobody's going to listen. I mean, maybe it is the answer, but you're going to have a hard time selling that. So build your relationships, spend that time. Number two, the questions drive the analysis. Be flexible. We had the question, what comes first, the questions or the model, right? I would say for us, it's always the questions. We have models, we maintain models, big, heavy, expensive models, but sometimes you don't need a model, you need an analysis. Or you need to step back and do what's being asked. So some of you may recognize this creature. This is the American bison. They are ranched in the United States, so sort of raised. They are fierce, independent creatures, and so ranching them is a interesting endeavor. But they are ranched across the United States, and so we had a question come, and they say, "Oh, are bison in your national FMD model?" I said, "Well, no, we hadn't put bison in the national FMD model before. We just hadn't really thought about it." And they said, "Well, we need bison to be in the model because we have some questions about how we're going to manage bison in an FMD outbreak." I said, all right, we're going to parameterize bison. So now bison are in the national FMD model for the United States, right? So you got to stay flexible and answer the questions that come and be responsive to those needs. Similarly, this is my one shout out of recent work. So Shankar Yadav, former postdoc fellow with our center, recently we just published together this paper looking at Epi and econ considerations around managing persistently infected cattle. So the idea that in a feedlot, you may not be able, well, if you have a lot of feedlots involved, you can't depop. So you got to find ways to work through when the ideal strategy doesn't work. What's your contingency? And then last but not least, focus on sustainable systems. So how do we build pipelines for talent, pipelines for data, pipelines for experimental data and research, building those partnerships in place, pipelines for model development. They don't fall out of the sky. They take years to build, right? So build your pipelines, get those things in place, and put together a sustainable system so that when the policymakers come with their questions, you're ready to respond. And you have those sustainable system in place that they can rely on year after year as policy questions continue to evolve. So with that, I will leave you guys, and thank you very much.
Thank you very much, Amy. Now, Melissa has some announcements to give to you.
Yeah, good morning, everybody. I hope you can hear me. I've been asked to tell you today that participants in the workshop that's going to begin after the coffee break should meet just outside at 11:20. So in the coffee area in front of the plenary room. So I think that's just out there. Announcement number 2, for those of you that are checking out of this hotel today, the checkout time is 12 noon. And you can put your luggage in the Espace Cultural room. Okay? So I'm sure somebody can tell you where that is.
Change the topic. Yeah. Now we will follow with the next presentation that is by Mr. Bouda Ahmadi. He is veterinary and agricultural economist, specializing in economic and policy analysis of animal health and disease control. Since 2019, Bouda has been working for EuFMD, and he's presenting today Bademos. This nice and interesting application.
Thank you so much, Herman, for this introduction. Good morning, colleagues. It's a great pleasure to present a piece of work that we are doing under current EuFMD program, and that is focused on developing a vaccine demand estimation tool for managing fast diseases. It's very difficult to talk after those two fantastic presentations, but hopefully this is a short example of what Thibaut and Amy were talking about. So vaccination is, as you know, essential component of any fast disease control program, especially in endemic settings. And a very big gap exists between the amount of vaccine required by countries for disease control program and also the amount manufactured and accessible, especially to the countries. Organizations responsible for fast disease control need to understand the size of this gap at national and regional level, and also to better define their policies and implement them. So also, as you know, and many of our manufacturing colleagues are very well aware of it, the investment decision is very much depending on the forecast of the vaccine demand in countries and in the region. Sorry, I wanted to say something about the graph in the right-hand side here. Basically shows the official report of the actual doses of vaccines for FMD used in 2020. This is WAHIS data, and you see a major gap. Of course, all those aspects that Thibaut was talking about in terms of the reliability of data exist for this kind of graphs. So our objective is to provide a bridge for this information gap between FMD vaccine demand for effective control and vaccine production and supply from the other hand. And this again is going to be at national level and hopefully at regional and global level. So to provide also a decision-making tool for countries, especially in stages 1, 2, and 3 of a PCP, in order to better plan their strategy of controlling foot and mouth disease. So what is VADEMOS, vaccine demand estimation models? And this is the first example we started with FMD, and is a decision support tool, as I told you now, to be used for estimation of current and future demand. It's a stochastic quantitative model. It's a mechanistic model, as Thibaut mentioned, the typology of models, and also it's being used to look at the forecast of growing, for example, population, and also considering the progressive control pathway of countries, and a number of other elements, the scheduling of vaccination, outbreak forecasting, etc. So in a way, we are providing these different, let's say, factors in a small calculator that is hopefully more useful than simple calculations. So it uses different databases, WAHIS datasets, FAO stat databases, and also country-specific data on, for example, on outbreaks, livestock population density, and also we ask experts about their experiences on, for instance, if a country moves from one stage of PCP to the next, what will be the impact of that on the demand. So I don't go to the details, but just I wanted to show you on the right-hand side that, on the left-hand side, sorry, that we have two main streams for this calculation. One is prophylactic vaccination, and the other is emergency vaccination. And I should say data on the second one is quite rare, and therefore we need to do more work on that. Here is a small presentation of the online interface of this. We have a production survey. You can choose your region, you can choose your country of interest, and then there are a number of input parameters that you can change and adjust from the default values that we have defined for each parameter, and it's being visualized immediately underneath, and then you can run the simulation, and instantly you get results for specific years, and the results are very stochastic by nature. I come to that in a minute. And then you can aggregate, summarize the results, and also there is a map here to bringing other layers of information, such as serotypes, etc. And you can export these to Excel or whatever format you are interested. So expected outputs, basically number of doses, geographical area, type of vaccination, whether it's prophylactic or emergency, species, large ruminants, small ruminants, pigs, and time horizon. We have now 10 years. You can choose your time horizon, and the sectors, which is dairy, beef, and smallholders at the moment. So these are examples of results in a table format, and as you see, you have prophylactic vaccination, emergency vaccine. In this example, we didn't have any information from Rwanda, so therefore it's 0 at this stage. But I mean, the user can basically adjust the input parameters, as I mentioned. So this is a graphical presentation of the results. It's, as I mentioned, it's a stochastic distribution of the total number of doses needed. This should be by 2026. Apologies for that. And then on the left-hand side, you see the progression of the vaccine doses needed. Here is per species vaccine requirement, large ruminants and small ruminants. And then here we have a comparison between what Vademus predicts and also what is actually used in the field. So we see a little difference, but in most cases we are very close to what is actually used, and also what we think it's needed for effective control of the disease. So this is a regional level estimation. So just to show you that the model can aggregate the results and report the total number of doses needed per region. And again, for emergency aspect, we really need to work on the data side, because we are not very certain about that aspect. So a comparison is quite difficult, because the information available is quite scarce, and also there are missing or inaccurate data from authorities. So we are working on that aspect, and a vaccination scheme, for example, or strategy often not very clear. In general, we have a good prediction for a good proportion of countries, and numbers are always within the calculated range. But most probable estimation is sometimes is overestimated, and the confidence interval is big. So we are working on that aspect. So as conclusion, in overall viewpoints, Vaadimos provides good level of accuracy for a large proportion of the countries. It provides aggregated numbers for vaccine dose at national and regional level over specific time horizon, which could assist business investment, hopefully by manufacturers. We are working on the model to make it better in estimation of the number of doses needed, and also it's more or less ready to be used by countries, and we are planning to basically to bring it to the policymakers. As Amy mentioned, often you publish something very late. Hopefully, we are going to use this model, for example, in roadmap meetings for FMD, and before the meetings, basically decision makers could use the model and get prepared and come to the meetings for their plan. So I want to say that the prototype is available if you want to go and to see how it works, and please provide us feedback. And here are my email address and a EuFMD email address that you can use for that purpose. I would like to acknowledge all Vaadimos team behind this, both present and the past, because this work started 2 years ago, and also colleagues at EuFMD, FAO, WOHA, and other associated institutes who are basically providing help and their expertise. Also, colleagues in countries, especially those who attended our past workshops to validate the model, both from public sector and private sector. Thank you so much.
Thank you very much, Boda. And now we are going to the panel discussion, the first panel discussion of this session. And for that, I give the floor to Melissa, who will be the moderator of this.
Okay, thank you. Thank you very much. So our panelists this morning are going to include the keynote speakers and Buddha. So please join us again on the stage. And we're also going to shift Herman's chair, so he's going to become also a panelist. There we go. Okay. Yeah, I think we stay here.
Shuffle, shuffle.
A bit more horizontal. No, on the line.
Okay, so thank you very much to all of our speakers this morning for a really interesting session talking about some of the technologies and innovations around modeling and how it can be used to inform policy and some of the exciting new opportunities looking forwards. So I'll start with a question. Maybe I'll direct it first to Amy, but then other panelists can also join in. And Amy, I was wondering if you could give us some insights into your interactions that you have with policymakers. What are some of the other considerations that they have, aside from the great models and modeling questions that we can give them? And what are the opportunities for modelers and analysts to really be influential in that process?
Yeah, no, thanks, Melissa. Certainly, that's a complicated question. And like I said, I think policymaking is unique in each country. And so how your policy process goes, particularly for emergency response, there's a lot of, if you get into political science, there's these really complicated models about policy windows and policy opportunities. But for emergency response, most countries are working through, their response planning on a cycle, right? So there's a disease of the day, that is your disease of interest. So in the United States right now, I can tell you between avian influenza and African swine fever, those are our diseases of the day. And so you find that in that moment, you're churning through right, our preparedness plans. So you're running exercises, simulations, you're meeting with our state animal health officials, and really putting a response plan through its paces. And then you're running into all the hard questions, right? So those exercises, those simulations, they start to bring up, well, what are we going to do about this, right? This isn't going to work. Now what are we going to do, right? So these questions come up, and that's the moment when modelers and analysts can really engage and help bring some insights. And they can say, well, you know, here's maybe 3 things that you could try, right? And here's how they kind of play out. This is what it's going to mean in terms of resources, you know, and responders on the ground. This is what it's going to mean in terms of economics. And this is what it might be the epi-benefits of taking these different options. And so you sort of help make progress through that entire preparedness life cycle. And then once the outbreak hits, then all the other policy questions come to the forefront, right? That you didn't realize you needed to answer. And so then you get into a really rapid turnaround of evaluating options and providing advice with modeling and analysis. And it's usually some sort of hybrid of using a modeling tool and then just working with your outbreak data to provide that sort of rapid response. So I think plugging into that preparedness life cycle and being a part of how countries work through and hammer on their emergency response plan is really critical for modelers if you want to have a big impact.
Thanks, Amy. Would any of the other panelists like to give some?
Yeah, my experience with policymakers is very similar to Amy's ones in terms of my experience in Scotland was we had a lot of interaction with policymakers and regular side, and they had questions you have to answer. Sometimes only about data. I think there's confusion between modeling and data. A lot of people, all their questions can be just by analyzing the data sets that we have at hand, the quality and everything. And most of the question actually is data related in data analysis rather than complicated simulation models, for example. But during response mode also, it's actually the modelers are also the data analysts. And so if they ask the questions and depend also the phase of the outbreak, early on is very much a data response is to assess the risk of infection through movement or whatever. And then when the disease is a little bit advanced, they are starting to think about consequences. And this is where you get those big models that are coming in. What is the best strategies? Because at the early stage, all their decision about disease control has been part of their plans already prepared way ahead. And so it's just more, how can we adjust for what we observed on the field that is different from the scenarios that we had before?
Yeah, and that's, I mean, that shift from an emergency preparation to emergency response, I think we could have a whole other session just on that, probably. Herman, do you have?
For me, it's, the most important thing was mentioned presentation of family, what was very interesting in the last slide. I mean, for me, I think that the most important thing is to build this relationship between modelers and the researchers and policy makers. I don't know, directly through other people that are related to policy making. But this is very important because, in one hand, you should focus. Modelers should understand that they focus to the correct answers, to the correct questions. And because sometimes, as you mentioned, there are a lot of modeling, a lot of things that are useful or not. That depends on that. And then try to explain to the policymakers in simple words what are the benefits, what are limitations, what are the possible uses. And sometimes policymakers don't know all these things. So, and in my opinion, that is very important to build that relationship from the beginning, from the start point. Yeah.
I totally agree. I just want to add that all this process creates trust, and when you start from the peacetime and you gather all the basically stakeholders, policymakers, they understand the tool, they understand the data, they understand the shortcomings and limitations. I think that's very important when an outbreak happens or an incursion happens. So we really need to understand what is inside that box. And when they are involved since the beginning, I think that helps a lot for communication and also better decision making at the end.
And just to highlight something is that the relationship and the trust and the work before responses allow modelers and policymakers to understand the questions. So basically, when a policymaker asks a question, a modeler may understand it differently than a policymaker. And so having that relationship helps to better understand what the other mean and initiate a conversation. And that conversation is the most important tool that a modeler can have for impact or for policy guidance, is that you need really to have a conversation to understand what the question is so that we provide the right answer.
Yes, so I mean, I meant bi-directional. I mean, because the language is totally different. That is the thing, you know. Yeah.
Okay, so that's coming back to what Amy was saying in her 3 key points about this building relationships and communication. Are there any questions in the audience? Please.
Hi. It's about the data management systems and communication between them. So I do a lot of meat inspection training in different settings, including different countries. And one of the issues is that the language used is not the same. The terms is not the same. The purpose of the training is normally aiding decision making, but also making sure that data is valid because the language is used. Because even in terms of institutions, people with different backgrounds might use the systems, the recording systems, in a different way, and that makes the data value just. So, what do you think is the best strategy? That was where my question comes in. Is it harmonizing the language so that the data is more comparable, or creating better translators, or both?
No translator effort will help to, because translator or official translators sometimes do not know exactly the field, and it's best to let the control by the people that are doing it. I think the problem with the language things is it's really important. It's core to all the problematic of having that international and global data sets that we all need. And I think you mentioned in your talk, is that if we want to answer a global aspect, we need a one integrated data set. However, it's not only the language issues, it's how you describe a disease.
That's what I meant by language, is exactly the terms you use rather than if it's French or English.
And it's the whole cultural things is that the name of a disease can be similar to another name of a disease in another language. There's the whole cultural aspect of it. There's also the how they structure data sets is different because their mind is different, and you have to cater for that. And nothing is, no structure is based for compared to another one. It's just designed for their own local specificities. So I tend to think that you get a patchwork of data sets, and we need a system to integrate all of that, to combine it together. And the problem of language is we need to work together to make sure we understand what is, and that takes a huge amount of time.
And the problem, I think, is also who sets the standard. So if you're going to harmonize the terms, whose terms are better?
Yes.
So then it's a fight of egos and things.
Exactly.
Okay. Yeah. I think Amy wants in too.
I would just add to that. I think it is a good practice, particularly when it's a big part of when we're in emergency response mode, to visualize your data back to the people who are providing it. So sometimes we'll get, months into an outbreak response, and all of a sudden you realize that everybody's coding the names and types of poultry flocks like different ways. And so you just pull up a list from our emergency management system, and you say, look, this is what people are entering, and this is crazy, and we need to narrow this down. And so it's like a come together moment, like let's negotiate and fix our terms. So I think visualizing that back to the people who are providing information can be really helpful.
A good trick. Steen, please.
Yes, thank you. I think it's a very interesting session, and I enjoyed very much. I would say, though, that to me it seems that we are forgetting one party, and that's the public. Me as a policymaker, I don't only want to discuss with the data analysts and the modelers to understand what they're doing. I want the modelers and the data analysts to have this debate about the quality of the data and what models can do in the public domain. And then I will listen as a policymaker, right? So, and what we saw with COVID was that lots of data were being put out there on dashboards. So it was a public debate. What does those data mean? What can we do? What will be the effect of interventions and so on? So me as a policymaker, I don't want that discussion in a closed space. I want that in the public.
So I take two takes on that. It's first, you're not listening to modelers for your policy decisions. That's good to know. And second is, I agree with you. We need to come with model output and data out there for have that public debate. But bear in mind that public debate means confusion. And so we're going to have to deal with the confusion too, and that creates more uncertainties that during an outbreak response, sometimes you don't have the bandwidth to deal with. So there's another thing is, I mentioned that yesterday in the workshop, is that dashboards are great, but too many dashboards may be not so great, because you will have the same data informed in different ways. They may be analyzed differently. They can be cleaned differently. You will see differences. And therefore, the public opinion will always focus on differences rather than what does it mean exactly. And so not only you have to show the data, but you have a massive effort of communication about differences and uncertainties, and how the public prepare for thinking uncertainties and variabilities and those kind of things. So, and that is a great debate. I think there's a lot of people are starting to think about that, especially in the One Health space when we want to learn on different levels, but it's, I don't know if we can manage that during, disease response.
Would you like to respond, Steen?
You wanted to add something? Oh.
Well, as a policymaker, I can tell you that confusion is brought between me and the decision maker if it's not taken by the modelers and the experts in the public domain. They will ask me, why this, why that, etc., etc. So that's why I prefer that the experts go public and discuss these matters.
Yeah, I was just going to add, I mean, I think this is where, the policy context within each country is unique, right? So in the United States, we deal with a country where the vast majority of people have nothing to do with agriculture, right? There's only a tiny subset of people who are involved in agricultural production who really understand livestock disease and disease control measures. And so most of our conversations are within the agricultural community, right? The people who are going to be impacted by the decisions that we take. And it's not that we're hiding that or not engaging, in more public ways, but we really prioritize communication with our agricultural stakeholders. And that's just an approach that is unique to us and how we develop emergency response policy. So again, you have to be sensitive to the context that you're in, and in some countries, those sort of public, really high-level debates might be useful for some diseases. For other countries, you're just getting a lot of people who have no skin in the game and no context and understanding of agriculture to contribute to the dialogue.
I totally agree that this country is specific. I think that's quite right. But in general, I think basically I agree with what Sten says. I mean, basically, when you bring the debates on the public, I think it adds to the transparency. And then you as modeler or as an expert in that field, you will be more even critical on your own interpretation of what you have. And also, it basically exposes your piece of work to other disciplines. And as Amy shows, there are other experts in different fields who can basically have experience and basically enrich that sort of area. Not necessarily their own discipline or domain, but they can contribute to our agriculture or veterinary problem from their perspective.
And maybe there's different different degrees of public. There's your key stakeholders and the industry people who are going to be very switched on and very experts in the field, and and they need to be part of it. And then there's, the more general public, and COVID, of course, we all have a real vested interest in what happens with COVID, but maybe a bit less directly with the agricultural debate.
Yes. No, well, all depends on the disease and the impact. We are speaking very general, but and depends on the country, of course, a lot of. But when you are in the middle between, we are speaking about policymakers, but we are speaking the decision-making process. It's not just policymaking, because policymaking, I don't know how to define policymaking, because the time is becoming very difficult to define policymaking, because there are a lot of factors that are influencing logically, the policy making process. But the decision-making process on animal health, at least in my opinion, should be a part of this multilateral discussion from the beginning, at least in my opinion. And there could be bridges between the modelers and decision-making processes. That's it. It's my opinion, of course. I try to do it. I am in the middle of these two things.
Thank you very much. I would like to make some comments also to relate with what we've been discussing. I do appreciate all the comments and the information that has been shared with us, with our speakers this morning. And I would like to make some comments before we take the last question from the physical audience, but we also have some questions from the online audience. And I would like to agree that we need to build relationships. We need to understand the actors, do the actor analysis, and really appreciate all the different actors. Some have major stake, others have smaller stake, but they all matter. If we forget or leave out any of the actors, actually it can undermine all the other efforts that are being put together. I was reminded when we had the first COVID-19 lockdown at the beginning of 2020, I was still leading the veterinary profession as the president of the session. And I was a president who had a very good relationship with the farmers. So I got calls from farmers from different rural areas, and they were asking me, Madam President, why are they locking the cattle markets? Why are livestock markets being locked? Do cattle suffer from COVID-19? This is unfair. We've just had quarantine, and cattle markets were closed because of foot and mouth disease. Now, this time, it is not the animals suffering, it is people. Why are they closing markets? We would like to sell. We would like to sell and make money and earn money. So I explained to one of the farmers, I said, the animals so far, what we know, are not suffering from COVID-19. Unfortunately, the animals cannot walk to the market by themselves. They cannot walk to the market and get sold when the people are locked inside their homes. And with explanations, I realized that the farmers then, it's as if they were not seeing anything wrong with markets operating, even if people were locked, because the customers, the buyers were locked, the sellers were locked, but they wanted the markets to remain open. In their view, it was unfair to close cattle markets when it is the people who are sick, and yet when the animals are sick, the people are not locked. The people continue moving. And those were their views. And then another farmer asked me one time when there was a quarantine, asked me, supposing you are suddenly informed that you will not earn your salary for the next 6 months, how do you feel? So the farmers were saying that when the scientists, the veterinarians, we are implementing the disease control in response to foot and mouth disease, we seem not to have a human face. We seem not to understand their plight. We wake up and say, suddenly we announce, from today, this is closed, and the other is closed. No questions, no explanation, no options. So they were asking, supposing it was you, and your salary was just stopped. You're just told from today you will not get a salary for the next 6 months. And your whole livelihood is depending on your salary. What would you do? So they were saying that is the situation where they are put many times. But I realized that is not how I saw it. And that's how many of us, we don't see it like that. We just see stubborn farmers. We see stubborn traders who continue to move the animals when we have told them not to move. So I do appreciate that. I think it's a high time we try to engage all the stakeholders and give them an opportunity to be heard. And sometimes they ask they may seem silly questions, but they open our eyes and help us to actually understand the challenges in a system in a system approach, rather than which helps us to actually address all the pieces, put all the pieces together. So the way I have related and from what I have picked from all the discussions, those are some of the comments that I thought I need to make. And based on the environment where I come from, where we can't finish 6 months without having an outbreak and a quarantine in a particular area. And they are always, then lastly, I want to say actually when we engage, we address the information gaps. Whenever there are information gaps, people fill in their own information. Some people emerge and they start to advise and tell people what to do. So as much as possible, I think it's important for us to address those information gaps. And that's why sometimes engaging with the public is important. We now know from food safety point of view, people are saying we want cage-free birds. We are concerned about animal welfare. We don't want to consume beef from animals that have not been produced in a certain way. So for me, generally, again, from the food safety point of view, I would like to think that everybody is concerned about what we do because we are producing food. And as long as we are producing food and people are consuming food, in a way, almost everybody within the public will have a certain stake. It may not be a big stake, but they will have some stake. Those were some of my thoughts. I would like to take invite one question that we had from the physical audience before we get the slide from I think the online questions are already there. Please go ahead. Thank you.
Meetings. Thank you. It was a very inspiring morning. It seems now we come into an era where the sky is really becoming the limit for modelers. We are now talking about cloud modeling. It's incredible. I don't want to sound old, but when I started my career as epidemiologist, we had these old school books on epidemiology where there were chapters about sampling methodology, how to sample, how to collect observations. And those, we had to be assured that whatever sample we took, what data set we collected was leading to unbiased results. And we had a data set. Then there were databases, now the data platforms. Now modelers can use thousands and thousands of observations where in the old days, we were happy with 100 or 200. We were extremely happy to make a model. We had to validate our model. We had to see what the outputs made sense. There was internal validation, external validation, looking at causality. It seems, and I didn't see that in your FAIR approach, Tipo. So where is this unbiased output still in play when we've been relying on these huge data sets, which are very often collected for other purposes than for your research question? So how can modelers to the public assure that what they come up with makes sense? There have been models made during COVID on reproduction ratios without a proper case definition of COVID. So, and it's going into the public and everybody accepts it because decisions are made based on those results. So how is, in the future, how can modelers assure to themselves and to the public that what they make as a model is really something that makes sense? And I don't want to be bold, but you know what I'm talking about. Thank you.
So just two thoughts. I agree with what you said 100%. One thing is the FAIR approach of FAIR principles is how we integrate data sets, okay? The data sets also, are collected differently. They're collected either from open access or free digital environment, or from business or regulatory bodies, or for researchers, okay? So this is how we treat data sets. So that is slightly different than checking the quality of your data set. And volume doesn't mean quality. And volume doesn't mean And citizen data collection doesn't mean that is an ethical or equitable data set. It only says that it's where people are interested or have access to reporting the information. So the question is more, it's where epidemiologists or data manager or data scientist has to think about how we can make this data collection more valid, more representative of the situation and limit the biases. This system to resample the data and try to make it close to the theories and those kind of things. But it's, again, taking time. You need to know your data. You need to know the methodologies. And that's why the FAIR principle is important. It's because you need to disclose all of this information for each database for people to get an idea of how good is that data set. So how good is the Google movement, mobility movement? How good is the flu track? How good is all those new digital tools that we love to use, love to display on the slides, and actually maybe they give you the wrong answer. And also there's the extraction of the information. You need to know how good is recorded, duplications, maybe same events, different IDs, but you need to figure out those duplications. So there's a lot of time of data cleaning that needs to be there still.
Okay, thank you. So now we can move to the audience online. And a while ago I was told that there was 200 some people online. Probably there's more now as people in other parts of the world are waking up or finishing work and able to join. The first question is also for Dr. Porphyre.
Oh, wow.
Asking about this concern about the location of cloud storage. Can this risk be mitigated using encryption, like PGP? I don't know what that is. But.
The problem of location is related to, and bear in mind I'm not a full specialist on those kind of things, but is who can have access physically to your data. So, for example, if your server is in a country that the local system or the political political background is sensitive, you may lose all your data by hacks or by a coup or by something. So you have those kind of things. There's also some issues related to the difference between American legal system and the European legal system and the African legal system, and many African legal systems. So there's a way of accesses and being able to get into your data. And so that's why the server access is a location has been important. That's why a lot of data moved from the US servers to Ireland. But then also there's a lot of data and appetite in Africa to use cloud computing. The problem is, where does the data go? Is it going to Ireland, or do they have the infrastructure? I know that in Uganda you do have some infrastructure in Makerere University. I think there's also some in South Africa. But therefore, some provide this information, some don't. So you have to think about these things.
Okay, thank you. And a very specific question to finish us off here today, or actually before we go for coffee, we're not quite finished yet, but is Videmos available online, and how can it be accessed?
Well, we are trying to make it online. It has not been launched yet as in public domain, so I shared the access information for colleagues attending this conference. So it's mainly for receiving your feedback, and yes, I mean, I want to make a bridge with what Stan said initially. I think, well, we wanted to make it public, and because it's used as publicly available data. so in WAHIS, in FAU-STAT. So, and basically we are trying to make sense of this data. It's not a very complicated modeling as such. And also, I want to come back to Koen's question. I mean, this exercise, any modeling exercise basically helps you to understand the system better. The aim is not to have, you know, the best answer. I would like to come also to what Amy said. It's more about optimization of decisions. It's not always possible to maximize our basically objective function as such. So we are optimizing, and that's what we want to do also with this modeling. We are trying to estimate. I think we are trying to estimate the number of doses needed, but this may not be necessarily very accurate. So we are constantly trying to update. So this is an ongoing work, and how I see it, it needs to be fine-tuned as we go forward, especially for each specific country, because people and colleagues in each country have much better information and data and expertise about the situation there.
Thanks very much. Any other final words from our panelists? Herman, no? Amy?
Yeah.
Do you want to have some closing words?
I would never stand between someone and their coffee.
Okay.
All right.
Thank you very much.
Thank you.
For desk, posters and coffee.
So please come back at
11:20.
Okay.
Herman.
We are starting the next session, the next part of the session of this morning, and please, I don't know if there are people coming. Just please sit down. Okay. For that, we are inviting to the stage to Roberto Condoleo. Roberto is veterinarian with a background in epidemiology and risk assessment. He's involved in modeling of animal diseases and zoonosis and worked for a governmental veterinary institute in Rome and has been collaborating with EuFMD during the last years. Please, Roberto, welcome and thank you very much for being here.
Hello, everyone. Thank you, German. Thank you to the audience for being here and online. I'm going to give a very brief description about this EuFMD project that is still ongoing, that is called Risk Monitoring Tool for Fast Diseases, in short, RMT-FAST. Sorry, a problem with, OK. What is RMT-FAST? RMT-FAST is a graph. It's a generic risk assessment framework for disease incursion. These frameworks are about risk assessment because they estimate the likelihood of incursion of exotic diseases from a source area or multiple areas to a target country. They are called generic because they are flexible. They can be easily adapted to different diseases, to different areas of interest, to different pathways. Graphs compare rank diseases. So what's the most important hazard? They can rank the areas, so from where does the hazard come from? Or they can rank the pathway, so how does the hazard enter in the area of our concern? As you can see from the picture, some graphs, some frameworks has been proposed recently. Anyway, there is not a perfect graph. Every graph has pros and cons. This is due probably to the fact that they are very different in several aspects. For example, there are frameworks that are focused on the entry, that is the first part of the risk assessment process. Other ones that are focused on the establishment. There are frameworks that deal with one pathogen at a time, others that deal with multiple pathogens at a time. Some deal with only one country, others deal with a group of countries, pooled country. And again, they can deal with different pathways, they can use different platforms. Anyway, most of them tend to use many parameters. So they could be time-consuming, resource-consuming, or they need skill, specific expertise to be used. For this reason, EuFMD thought to create a new framework, a new tool with peculiar characteristics, that is RMT-FAST. This tool has to be able to monitor the likelihood of entry of fast diseases in one target country from multiple neighboring countries that we call source countries. RMT-FAST covers not only foot and mouth disease, but covers also 4 fast major diseases, namely lumpy skin disease, Rift Valley fever, pest, the petite ruminant, and the sheep and goat pox. It considers the main pathways of pathogen introduction, but it's important to say that we wanted something very simple, something that can be used easily by everyone, that can be easily understood by everyone, and above all, very rapid to update. This is a tool that has to be used periodically over the time, routinely. RMT-FAST is a semi-quantitative tool, so a score is attributed for each of the 5 parameters that are part of the tool. You can see in the slide the formula that is applied to calculate the risk score that represents the likelihood of entry of one pathogen in consideration of the multiple pathways that are considered by the tool. The first parameters is updated by the EuFMD team regularly, and is about the disease prevalence. It's about the level of the circulation of the pathogen in the source country. Again, the second parameter is updated by the EuFMD team regularly. It is about the level of the mitigation measures that are applied in the single source country. For example, the proportion of population that has been vaccinated, or the level of the surveillance versus a certain pathogen. The third parameters take in account that not all the pathways are effective at the same way. We see this morning, for example, that for lumpy skin disease, vector-borne pathway is very important, also for Rift Valley fever, but it's considered negligible, for example, for foot and mouth disease. Finally, the last 2 parameters are combined together to estimate the connection strength between the target country and the source countries through the different pathways. The connection source is the fifth parameter. It's the only one that needs the intervention from the user that have to characterize the relationship, the strength of the relationship between his country, that is the target country, and the other countries that are being considered. This is in terms of geographic proximity, in terms of trade, import of live animals, trade, import of products of animal origin, both legal and illegal way. Also, for example, in the magnitude of the transport connection, and so on. Here I present the result from a test, so the result from a pilot, experimental result that we conducted with Spain, with the collaboration of the Spanish colleague. There is no time for going in detail, but it's important to say that it's not possible to compare the single risk score because the transmission way of the single disease is different. But obviously, the higher is the risk score, the higher is the likelihood of entry. The tool provides results by single disease. And it's possible to visualize this result, so to rank the country that are in terms of risk at major concern, and also to identify the pathways from these single countries that are more important for the risk managers. This is not a static picture, so this situation, this score change when change the epidemiological situation or when change the connection strength between the single countries that can be also on a seasonal basis, for example. We try to test, to validate the tool. We perform several pilots with other countries in order to collect a feedback through questionnaires. We have evidence that the tool at the present is still user-friendly, easy to update, it's very practical for a preliminary risk assessment, but we are aware that there are some limitations, as underlined also yesterday during the workshop. For example, data collection is difficult for some parameters. Not always the information are updated completely or in time, and there is still a certain uncertainty around some parameters because the score is attributed by expert elicitation, even if obviously the tool provide criteria for the attribution of the score that we didn't discuss for a matter of time. So the application of the tool, obviously the tool is could be used by the risk manager to assist the risk manager and prioritize the country and the pathways of introduction, as well as can be used to give indication for the border controls. It's useful to monitor the risk and to highlight changing risk in case of relevant epidemiological updates, events, for example, new outbreaks. Can be used to implement periodically report bulletin, and as I said before, can be used for a preliminary risk assessment, so to identify that situation that needs a more detailed investigation. In conclusion, the tool seems agile, is well accepted by the user at the moment. The quality of data is problematic. We spoke about this before, especially for some parameters. And the project is still ongoing, so the version that we have is still a beta version. It's a prototype, so we need to further improve the tool, and especially regarding the surveillance limitation, the information that we have from the Pillar 2 countries and the legal and informal connection. Thank you very much for your attention, and thank you for being here again.
Thank you very much, Roberto, for the presentation on this tool that is starting. Next presenter is Margarida Arede, a PhD student in the Veterinary School of Barcelona, Autonomous University of Barcelona, Spain. Go ahead, the floor is yours. Thank you.
Hi, everyone. Today I'm going to present you the mapping of risk of spread of PPR in the Black Sea Basin, done through a knowledge-driven approach. This project was a collaboration between my university, the FAO Regional Office for Europe and Central Asia, and the PPR Secretariat. Some introduction on PPR. PPR is the most devastating and widespread viral disease for domestic small ruminants. It affects domestic and wild small ruminants. It has a high morbidity and mortality. in the acute phase of naive populations and threatens the food security, nutritional security, and sustainable livelihoods of rural populations. It's It's widespread in Africa, Asia, and Middle East, but it has had some outbreaks in the past years, in the past decade, in previously free countries as Georgia and Bulgaria, which made us brought some attention to the need of contingency plans for in case of incursion of disease. Towards this objective, there's the global strategy for control and eradication of PPR as the next livestock disease to be eradicated. And it's a collaboration effort between the OWA and the FAO. Also towards this objective, so in the same scope, our objective of this study is to identify areas at higher risk of spread of PPR and domestic small ruminants in countries surrounding the Black Sea that are specifically these 9 countries pictured here. To do this, we decided to use spatial multi-criteria decision analysis. That is a methodology that integrates available knowledge about PPR in this case and the use of free spatial data. This method has a sequence of steps here shown above. The problem definition, starting with the problem definition, that is similar to the objective of this study. Then the identification of risk factors. For this, we consulted 2 PPR experts and chose the most relevant risk factors already studied in the literature and that were most relevant for the region, and that we had the availability for spatial data for all these countries that we are working with. We ended up with a sheep and goat density with like these 5 risk factors: sheep and goat density, proximity to PPR outbreaks, smallholder farming, proximity to markets, seasonal pastures. For the data collection, we did data collection through a questionnaire with 9 focal points of these 9 countries through 2020 and 2021. The weighting of risk factor elicitation. So as I told you, this method uses the elicitation of value judgment, of opinions of experts, in this case, of the relative importance of each identified risk factor for the spread of PPR in the Black Sea Basin. These experts could be experts that had knowledge about the ruminant production in the region or about the PPR disease, knowledge about the PPR disease. The participants on this project were FAO project consultants, our focal points, country PPR focal points, as I said, PPR secretariat, also some nationals that worked, that participated in a training for PPR management, and the PPR grant, and international PPR experts, of course. For this, we did a pairwise comparison matrix, and we asked these experts to compare risk factor 1 on the column to risk factor 2 on the row, if risk factor 1 was less, equally, or more important on this scale, and filling in this pairwise comparison matrix. We obtained a total of 21 consistent responses. Each one of the responses gave us 5 weights for each one of the selected risk factors that we multiplied for the risk factor standardized map, and then that were averaged to obtain a final suitability map of spread of PPR. The final suitability map was is this one here shown. We saw that the suitability of PPR spread was higher in most of Turkey, the border of Thrace with Bulgaria, and most of Romania, and some regions in the Caucasus. And the suitability of PPR spread was lower in Ukraine, Belarus, and northern region of Bulgaria. We validated our results with cases from 2020 and 2021, and the capacity of our model was of around 75%. So for some conclusion, we had limitations regarding quality of data, different spatial resolutions for different countries, some use of proxies for animal movements, since this data was not available, and uncertainty of expert opinion that is intrinsic to this methodology. We see our suitability map, despite the limitations, as a good application of ease and effort that is easy to apply and affordable as well. In the end, we had like a moderate capacity of distinguishing cases from pseudo absences. And thank you, everyone, for listening. Our acknowledgments. Thank you
very much, Margarida. Now the next presentation is online. Will be given by Alexis Delabouglise. He's a permanent researcher in CIRAC, research unit Astre, based in Montpellier, France, since 2019, and he is currently involved in several projects in Africa related to animal health. Alexis, are you there? Yeah. Yeah. Okay.
Yes.
Yeah. Okay. Thank you. Good morning. Or good afternoon. Where you are.
Good morning. So thank you very much for giving me this opportunity to present this work, which actually was conducted as part of a more general work which we conducted with the support of EuFMD actually on innovative tools to better monitor and predict livestock mobility in different settings. So this specific work is about predicting the dynamics of livestock mobility in Senegal. I'm using the internet clicker, but okay, it works. Thank you. So mobility of ruminant herds in West Africa is a very important, has a very high economic importance since farmers, pastoralists, actually specifically use transhumance as a way to optimize the use of their scarce feed resources. And also long-distance commercial trade networks enable supplying livestock meat, livestock products to major centers of livestock consumption, which are located primarily in big cities like Dakar in Senegal. But of course, livestock mobility is also, as you all know, a strong driver of fast diseases like FMD or PPR. So it's important to find new ways to better predict, better understand what are the determinants of the temporal dynamics of livestock mobility over time, not just work with a static network. Okay, next slide. Okay. Okay, so our hypothesis in this work is that mobility can be predicted on the basis of several variables, which include the calendar, which reflects the importance of religious festivals in the trade of livestock, and also with environmental variables, and also some economic indicators, which are actually market prices. So next slide. So we used several data sets to conduct this analysis. We used rainfall data at department level. Next slide. We also used biomass production also at the level of the department, so which is the basic administrative units of Senegal. Next slide, please. And we also used a livestock mobility data set, which was provided by the veterinary services of Senegal, who monitor the mobility of the movement of ruminant herd by collecting sanitary permits. Normally, so theoretically, according to the legal framework, each farmer who wants to move a herd of ruminants from an administrative unit to another needs to request a sanitary permit from the veterinary services. Next slide, please. And of course, we also collected data on prices, which actually were supplied by the WFP, specifically the Vulnerability and Analysis and Mapping Office based in Dakar. We conducted a regular monitoring of small ruminant and cattle price in several markets of the country. Next slide, please. Okay, so in order to link the likelihood of, actually the number of recorded movement of herds between departments, specific departments of Senegal at specific months of the years, which were actually, so sorry, I forgot to say that we worked specifically on the year 2019. We used a generalized additive model, which allows us to model nonlinear effects of these variables, as these effects are supposedly nonlinear. And we also incorporated the constant terms, which are actually the specific effect of the root connection, so the link between pairs of department, and also the calendar effect, so the specific effect of calendar month as an additional spline term. Yeah, okay. Next slide, please. So one issue that we faced is the high number of missing observations in the data set. There are a lot of instances where no movements were registered during specific month. And to address this issue, which creates over dispersion in the data, we use the hurdle Poisson regression approach. Next slide, please. So it actually worked pretty well. We produced quite good fit using this modeling approach, so a generalized additive hurdle model. As you can see with the graph showing the relationship between predicted value and observed counts of herd movement for cattle and small ruminants. Next slide, please. And regarding the results, we could actually show a strong effect of the calendar month, which is actually related to some religious festivals like the Magal de Touba for cattle, which is a religious festival where cattle, the slaughtering and consumption of cattle is highly praised, and Tabaski for small ruminants. So Tabaski is the most important Muslim festival of the year, during which it is traditional to slaughter a sheep. So there are about 1 million sheep slaughtered in only one day in Senegal at this time of the year. Next slide, please. We could also demonstrate the effect of environmental indicators, so rainfall, for example, which actually seems to differ between cattle and small ruminants. Next slide, please. And also the effect of an effect of biomass. So actually a change in biomass, either negative or positive from 1 month to the next, seems to stimulate the departure of both cattle and small ruminants. Next slide, please. And finally, an effect of market price, which actually differs between cattle and small ruminants. For cattle, there seems to be a decreased likelihood of departure when price increases, while it is the contrary for small ruminants. An increase of price at the department of origin, I need to precise this, seems to increase the likelihood of departure. Next slide, please. So to conclude, so the used model seems to be adapted to this kind of data set. And we showed some evidence for the influence of seasonality linked to reduced calendar and market prices and the biomass and rainfall. This work has some limits. The limit is that, of course, the data set of livestock movement is incomplete due to the incompleteness of this data on the livestock mobility. Not all farmers actually obtain this sanitary permit before moving their herd. And also the availability of price data, which was actually inconsistent across the country. And yeah, we encourage also to conduct further studies to understand the difference observed between cattle and small ruminants in the motivation to move the herd, specifically regarding the effect of market price. And next slide, please. With this, I would like to thank all the collaborators to this study, and also EuFMD, we supported this work. Thank you very much.
Thank you very much, Alexis, for this clear presentation. And now it's time for the next keynote and last for these presentations of this session. Mike Tildesley is Professor of Infectious Diseases Modeling at the University of Warwick, UK. Okay?
Thank you very much.
Thank you.
Okay, so that's a little bit loud. Okay. That shocked me. Right, so I'm going to talk a little bit today about real-time decision-making. Now, there's been a lot of discussion over the last couple of days on the use of models during outbreaks, and I think there's quite a lot of polarized views in the audience as to whether or not we should do that. So I'm just going to give a little bit of information on my experience of doing real-time decision-making during the COVID pandemic, and then I'm going to loop back to thinking about FMD and show you some kind of implications of that. I think the UK really have a very long history of using models during outbreaks, and I, along with many others, was drafted into the COVID response in early 2020. And I'm going to sort of talk through a little bit about what the structure of the government modelling team was, and then we're going to talk about some of the kinds of things we had to do. There's a group in the UK called the Scientific Pandemic Influenza Modelling Group, and in sort of peacetime, as it were, outside a pandemic, they would maybe meet once a month, and it would be about 10 fairly senior academics who would look at kind of what's circulating and then advise regarding the risks. During the pandemic, this grew significantly to about 30 to 40 modelers. We were meeting every week, and the idea was we would feed in our model predictions to the Scientific Advisory Group for Emergencies, which was around about 20 very senior scientists, who would then feed through key modelling outputs to the UK government and the Cabinet Office. And then what would happen, there was this kind of feedback loop where the government would communicate scenarios back to the modellers that they wanted them to investigate. And this all happened in a very short period of time. Now, this guy, every day, would get up on the podium and then communicate that to the press. And I realised I haven't checked my email for about the last 20 minutes. We might have a new Prime Minister by now, the way that we're going. Okay, so just to give you an example of some of the things that we looked at, every week we were producing weekly R number estimates. Now, typically this would be multiple different modeling groups would provide their predictions. This was put into an ensembler, and of course, critically, you're looking to see whether R is greater or less than 1. And then the ensembler would come out, and this was what was communicated to the press every week. We did things like model fitting on a weekly basis, so you can see kind of this is one of our model estimates. And then very specific tasks like whether we should reopen schools, and if we should, what year groups we should send back, and what impact that would have on the R number. And then things like relaxing lockdown, and again, what the implications would be. So I'm not going to talk about those in any detail, but I'm going to give you one example of how that worked. So typically what would happen is the UK government would give us a commission. Very annoyingly, these often arrived on a Friday afternoon and they wanted results by the Monday morning. So we had some fairly unpleasant weekends during the pandemic. They needed very rapid turnaround because they had to feed into decision making. So I'm going to give you one example and I'm going to talk very briefly about it. So in May 2021, UK was in lockdown, but was gradually relaxing and coming out of it. And the UK government developed what they called a roadmap to unlock the country. So we were tasked with producing a report on the impact of vaccination and the impact of that upon relaxation. So what we did, we produced documents that looked something like this. Again, I'm not going to talk about this in any detail, but we had an executive summary, which was really kind of very high level. These are the key points that you needed to get out of this. And then things like tables of assumptions, lots of tables of assumptions and our uncertainty around them. The key results. And then very, very importantly, huge detail on sensitivity analyses. We know that these parameters were uncertain, and so it's very important that we looked at the implications of different parameters on our results. All of these papers are available on the UK government website, so there's a nice QR code there. If anyone wants to have a look, you can look at these. They are in the public domain because it was one of the things that was made very, very clear. We needed to be transparent, so these documents were written up and pushed out on the government website. So, what have we learned from this? Well, these models were used in real time, and they were continually refitted as new data were available. Now, very clearly, one thing we did learn is no two outbreaks are the same. So we need to be very, very careful not to use off-the-shelf models in real time without fitting them to what's going on during an outbreak. Now, after an epidemic is concluded, in a sense, with the benefit of hindsight, we might be able to go back and look at fitting our model and determining the best control policy. And I say best dependent upon what the government's objective is. That's very, very crucial. But what I want to ask is really how useful are models during ongoing outbreaks? And I think there is a lot of debate about how useful they are. So having put that into context with COVID, I now want to loop back to FMD. So in a sense, in real time, what we're doing is we're taking a bunch of data, we're taking some suggested control actions, we're putting them into a model, and then we have various predictions of what the impact of each of those controls will be on, say, the number of infected premises in an FMD outbreak. Now, after an epidemic is finished, and this is the UK 2001 epidemic, we take all that data and all those control actions from the end. And, we can do that and say, well, okay, we've seen the outbreak, we can fit it all, and we can see what happens. But of course, in real time, what we're doing is we're doing that here. We're doing it at the start of the outbreak when we have limited information. And so we want to look at what the impact of that might be. So, in big writing, what I want to ask is, at what stage are our model projections and control recommendations resilient to this information gain during an outbreak? And I think that's something we really need to think about. So, what we're going to do is we're going to take our model and sort of every week during the outbreak, we're going to advance through and we're going to look at how well these control actions work. And so I can advance through, and if I do this all the way to the end of the outbreak, every week I can say, well, okay, this is what I think each control action will do. Now, of course, every week, what I have is accrued knowledge. I have information only based upon what I'm seeing on this day. And of course, there's a lot of uncertainty around that. So, I might have a state of the outbreak where I know farms that are infected, I know farms that are susceptible, but of course, there's a lot of uncertainty under the hood that I won't know until the following week. And what I can do is I can compare that with what I would do on that day with the benefit of hindsight. So if I've seen the entire outbreak and I can fit to the entire outbreak, I can say, right, well, on the 26th of February, this is what you should have done based upon kind of perfect information, as it were, with the caveat that no information is ever truly perfect. And I can do the same thing, and I can say, well, based on perfect knowledge, in inverted commas, I can get another set of predictions as to what the outbreak would do. Okay, so what I'm going to do now is say, right, well, what would a retrospective model advise? So if I do this after the epidemic, then I can get kind of every week, I can say, well, this is the impact of various different controls. And I don't want you to get hung up here on, this is not me saying 10-kilometer vaccination is the best. I don't want to get you hung up on the different actions. What I want you to think about is the variability between the different interventions and how that changes. So sort of from this, we can really clearly see that right at the start of the outbreak, there's a big spread in terms of which control policy is optimal. And from this, and I say don't get hung up on the fact this is vaccination, but vaccination, according to my model here, always wins. It's always the best thing to do. And only culling for animals on infected premises is always the worst, according to this model. So in the benefit of hindsight, if I'm fitting to the entire outbreak, I should know what to do. The problem is, and I'm now going to compare this with what I would do fitting only to the information I have in real time. So the one you've just seen is on the top, and my new one there, only fitting to the information I've got during the outbreak, is on the bottom. And this is really concerning, because in week 1, all of a sudden, all the control policies look like they're doing kind of the same thing. Whereas in reality, of course, with the benefit of hindsight, we know what the best policy is. And again, I'm not a policymaker, but, if you present this to a policymaker, is this a suggestion of, well, we know vaccination's really costly, we know ring culling is really costly, so they all look about the same. I'm probably going to do the least severe thing because there's going to be the least damage. Now, by about week 3, we're pretty confident which is the best policy. And I think this is just an element of caution here that we need to be really careful using models in real time during the very early stages, because there's so much uncertainty that we can't really resolve that. By about week 3, we have enough information. So, very quickly then, I've tried to sort of delve into, well, what is it that's going on between week 1 and week 3 that means I don't know what the best policy is. So what we did is, we fit our parameters, and this parameter here is the risk of onward transmission by sort of by distance, and we can look at where the model thinks cases are now. And so the darker the county here in the UK, the more cases you see, according to the model. Now, the complete information tells me where the true cases are, but in week 2. The accrued information is giving me loads of cases in Scotland, in the north, that actually don't happen in reality. And by week 3, actually the model's doing a lot better. Spatially, it's all of a sudden realizing, right, we're not seeing those cases in Scotland, and it fits much better to the true data, as you can see there. Okay, so this spatial uncertainty really affects our predictions at the early stages. There's a lot of uncertainty in the system, and it influences how well our models are going to do. Okay, so I don't want to end on a real negative, so I'm just going to end on a slight positive. So what we then did is said, well, okay, what if we rank our control policies from best to worst using our real-time and our hindsight model? And again, there's a QR code to the paper here if anyone's interested. And actually, it's slightly more promising, because even with this underlying uncertainty, from the very start of the outbreak, we can see consistency between these two models in terms of which ones are worst and which ones are best. The vaccination strategies tend to win. The only culling infected premises tends to be the worst. So there's an element of promise here that at least we are getting some sort of viability with the real-time model. Okay, so let's loop back. What have we learned? it's important as modelers that we're responsible and that we're responsive during ongoing outbreaks. We can't always capture complex individual behavior or nuanced intervention policies, and this is where communication with other experts is really, really important. I might suggest a control policy as a naive modeler that is just not implementable in the field, and I think that's really important that we are aware of that. And I just want to give one example, and I'm going to loop back to the COVID pandemic now. This is some work we did in 2021, looking at various different sort of scenarios as to what we expected to see as lockdown was lifted going through the summer of 2021. And all these coloured lines here give you our different predictions. That's what really happened. And this was a government decision that they were trying to sort of run hot and keep the R number basically at 1. So actually it meant that sort of it swept through the population and we had kind of this low-level infection that actually in a model we just couldn't capture. But that was a government policy decision, and I think this is really important that we are aware of this. So I suppose to conclude, I say models can play a significant role in supporting decision-making, but, and I really should put this in bold, they should only form part of the evidence base, only part of the evidence base, because there are other experts who can really, really help with this. Very final thing, we're now applying a lot of this methodology to endemic settings, where we know for FMD often we don't have access to quite such rich data as we do in say, places like the UK, where all animals are tagged and we have detailed information. And what we're really interested in is looking at how we can maybe target surveillance and monitoring to reduce uncertainty and use these data in these environments to kind of improve our model predictions. This is very much a work in progress, but it is something we're looking to move on to. Okay, so I think that's me done. Huge acknowledgement to a long list of collaborators at various institutions around the world who've helped to inform this work. So thank you very much for listening.
Thank you very much, Mike, for this interesting presentation, to be honest. I like it a lot. Now it's time for the next panel session.
Yeah, so the panel members again will be the speakers in the session, and so, but we're going to keep Herman here with us, although he should also join the discussion as he wants to. So Mike, Margarita, Roberto, please.
Put myself too, no?
Sure,
no,
yeah, why not?
Okay.
Please come back up.
I want to be in.
So thank you all very much for preparing the presentations and for sharing your work with us. I think it was a really nice, continuation and examples in some cases of the themes that we discussed this morning about data and limitations of data. Oh, we also have Alexis on the screen. Hi. And also the interaction with policymakers. And so just to kick things off, I would like to ask the panel, maybe starting with either Alexis or Margarita, you showed some research work that gives insights into animal mobility and risks in different countries. And I'm wondering how those research results can be practically applied, to the policy-making environment in those settings where you were doing the research. So you said, both of you concluded, I think, that this can be helpful to inform disease control. How do you see that might happen? It's a bit awkward with Alexis behind us on the screen. So Alexis, why don't you start? Because we can see you there, but I don't know if you can see us.
Yeah, I can see you. You can see the audience, the back of the audience, actually.
Okay.
I see the panelists. Okay. Maybe I can try to answer this question. As you said, indeed, it is research work results, and like predicting how it will be used and whether it will be used in the future is a little bit tricky. However, we can see that this work I mean, we can clearly see that this kind of work would have practical application, since the variables that we tested in the model that I presented are very easy to collect in the field. So biomass, rainfall, and even market price could be metrics that could be monitored on a regular basis by government agencies. And on the basis of this variable, make, I mean, not very accurate, I wouldn't say that, but at least like, have some signals of that could enable policymakers to predict the future extent, the future intensity of livestock movement in the coming month, for example, which could be used to inform to allocate more effort in surveillance, for example, or in the control of like, the sanitary monitoring or inspection of live animals being moved from one place to another, for example. The question here is more this is a model that we did on one place, Senegal, at one time. The question is whether this model is very replicable on other years, in other places. And I think to address this question, this work should definitely be replicated everywhere where it is possible, where we have good data on livestock mobility. In Senegal, we have the chance that the veterinary services have a very good data collection system on livestock mobility. So wherever it is possible, in Africa or in Asia, we will encourage to replicate this kind of work, specifically because animal mobility is really dependent on the motivation of farmers. Why we decide to move livestock. It could be for transhumance, or it could be for commercial reasons. And the effect of price as an attractor of animal herds is quite complex. Not necessarily farmers moving animals or traders moving animals to places where prices are high. Actually, we could see based on the result of our model, we suspect that actually the effect of price is much more complex than that.
Thank you.
I don't know if I answered the question, but
you've given good thoughts and good insights as to the nature of your work and how it contributes, I think. Do any other panelists like to input on this question about the applying research results to the decision making and practical control of diseases? Mike, it looks like you have something to say.
Yeah, I mean, I think it's a challenge, and I think I've already alluded to this in the presentation that I've given. I think that sometimes there's a danger when we're working directly with policy that, for example, as a modeler, that the models give all the answers. And I always, one of the things I harp on about a lot when we're communicating our results is communicating uncertainty, and I think that is one of the big challenges we have. We know we can't give definite answers. We know there's uncertainty around that, and there have been high-profile incidents in the past where scientific results have been reported when they've gone into policy, and that uncertainty has been missing from the work that's gone out. And then, of course, that then has long-term potential implications on the scientists themselves, because you report that uncertainty, you're generally, hopefully, you have integrity in doing that, but then that gets omitted. And then when your prediction does not necessarily come to pass, then there's an issue in terms of your credibility. So I think it's something we need to be aware of, how we report our scientific results, but also how we report our confidence and our uncertainty and what we don't know. I think that's just as important as reporting what we do know, but it's a huge challenge.
Yeah, that's a really good point. And Margarita, in your presentation, you mentioned that one of your limitations was the data quality and uncertainty.
Yes.
And how do you think that impacts your results, and how did you communicate it?
Yes, of course, brings uncertainty, and it's what Mike was saying, that we have to communicate with the decision makers to. It's just preliminary results of what they can use to implement these this control and surveillance strategies. But also, their data is also always improving, and these countries are in high development, as for example, the countries of the Caucasus that are implementing identification systems right now. Georgia just implemented this year identification and registration systems for animals, and this is going to change the data quality a lot. And when they start using it more for movement, for registration and movements as well, like we'll have much higher quality of data, and we'll be able to update this kind of model. Also, it's one of the advantages of our suitability map is that can be updated with more with higher quality data and more recent data. I think that's a little bit.
That's great. Roberto?
Yes, if I may, I feel a connection between this presentation and also the workshop yesterday regarding the collection of data. This is a topic that always we speak about, because in modeling you want a lot of data, you want the best data, but you don't have. And also, coming back to your previous question, this research have the scope of the aim also to try to manage this situation. Anyway, the methodology is not enough, and also my presentation was very clear, but I like also his message full of hope that despite the fact that we didn't have the best data at the beginning, but the choices that would have been done are the best one. They also, when we have perfect knowledge, the choices that we have done probably are anyway the best. So I think that we shouldn't stress too much the fact that we don't have data, because science is this to serve to give advice for the best option on the basis of what you know. Science is not perfect. It's like we said, what we saw with COVID, but it's the better of our knowledge, and the models, apart from, I like the methodology, has to be used for this.
Yeah, I think maybe that's the trick, is how do you make decisions in this, with this uncertain environment? And I guess, models can help with that. Herman, do you have any comments?
Well, yes, a summary of what they said, that they are desperate modeling anyway. But yeah, I think that data quality, uncertainty, how to communicate uncertainty. So I think uncertainty is systematically estimated also by statistical methods, and how to communicate to people that don't know about these models, and uncertainty is difficult. It's a challenge. It's a total challenge. And then the responsibility of the modelers, the people that are using these model outputs, because models at the end is just another thing. You have to look at the field. You have to see what is happening in reality. I mean, this is a representation that you can take it into account, of course, and it's a very good help, probably, if your data are good, but still, the old traditional way of looking at the diseases on the field, and I think or the decision making, or the decision makers, and that never have to forget this. I mean.
Do we have any questions in the audience, please?
Hi, Daniele De Meneghi, Department of Veterinary Science, Torino. A comment, a question for Alexis. I really like the presentation and the tool they use, but you mentioned that movement certificate of animal is a critical aspect, even if Senegal has a good veterinary services. But in your model, being the area, a pastoral area, how do you deal with the movement within and amongst the different countries, let's say Mali, Mauritania, and that area? So that is a component that has to be taken into account to have the whole picture.
Well, thank you for this very good question, actually, indeed. So, actually, in Senegal, the sanitary permit is also compulsory to move animals across the border, across the international border of Senegal. So whenever there is a movement coming in, traders need to request a sanitary permit from the Senegalese authorities. Of course, that doesn't mean that we all do so. There are a lot of, I think most of the movement go unregistered. In this work, we focused indeed on domestic animal mobility, but some of the movement, actually, some of the herd movement we worked on were movements coming from departments at the border with Mali or Mauritania. And the animals, actually, the origin of animals were probably from these two countries. So that also capture movement of animals coming from outside Senegal. Also, we only focused on the movement between departments. We did not work on the movement inside each department. So, for example, in a restricted pastoral area, we did not work at we did not work at the very say micro level in this specific study, focused on the long distance network movement. This is because of an issue of data availability and making a trade-off between model precision and completeness.
Thank you. Are there other questions from the audience? Case?
Maybe I can ask a question. I can ask one question as we wait for more questions from all our speakers. One comment almost came out from all our speakers about the quality of data that they are working with. And my question is, what are the modelers doing, or what can they do to support us, the veterinarians, the epidemiologists who are often involved in the primary collection of this data? First of all, what are you doing to give us the feedback so that we know the quality we are collecting, and also to support us to improve in terms of the tools that we use so that we collect better quality data that can facilitate you to do the modeling that you do best, so that we also do our part best when we are collecting this data, knowing that it is very important data that can be used to support decision making and decisions that could influence even the control of these diseases that we are eager to actually not only control or prevent, but we are eager to eradicate them, but we keep talking about them from one decade to the other. I hope you can get the summary of my question. Any of you can be able to respond.
I can have a go. Now, I will say somewhat flippantly, there is a saying in our field that you should never ask a modeler what data they want, because they will say all of it. But the joking aside, I think there is an important point here that we're very aware as modelers that there's only a limited capacity to collect data. And I think actually one of the really important things, I alluded to this at the end of my talk about the work we were starting to do in East Africa, is thinking about are the ways that we can target data. Because if we're trying to fit a model to an outbreak, it may be that there are some data sets that are really, really crucial. We really need to fit a model, and other data sets that actually are less important. And I think this is quite important for us as a community of modelers to maybe almost say, right, well, okay, this is kind of our perfect list of data that we would like, but however, these are the really crucial data that we need. So that if there's a limited capacity to collect data, this is how we should prioritize that. Unfortunately, the problem with that is that's very dependent upon the system that we're trying to model, that those data requirements may change. But I think it is something we can hopefully help to inform by looking at how model predictions change when we have different data sets available to us.
In short, if I get very, very short, I think that's key also communication. So to communicate to our colleagues the importance of this data, because sometimes they're not aware, and sometimes they think, as I think in my experience, that it's a lot of bureaucracy. It's something that they have to do because by law or because it's something that is decided. But indeed, they are producing a lot of important information that maybe are not important at the moment, but that can be important in the future.
I was in the UK outbreak in 2001 as one of the field epidemiologists, collecting these data that we're still talking about, a long time later, more than 20 years later. But the feeling in the field among many of the vets working there is, why do we bother with this? We just want to kill the animals, get rid of the disease, and go home. Yeah. But look, we're still talking about it. It's remarkable. Case, you've been patiently waiting. Do you still have a microphone?
Thanks, Melissa. I have a question for Mike. Real-time decision support. Very interesting talk. But when you have a new pandemic and you have a new prime minister, you will tell him, please wait for 3 weeks. That's what I...
New pandemic, new prime.
Yeah, and I think my point there was in the very early stages, what you really need is sort of what I would call muddy boots epidemiology. You need on-the-ground experts really actually going out and doing surveillance, find out what's circulating. In a sense, we have these models that we've developed and we can fit to the data as they come along, but they're only part of the process. And I think what I didn't say when I was talking about the structure of SPI-M, SAGE, and the UK government is that there were multiple different groups that fed in. There's a group of behavioural scientists, there's a group of public health experts, there was the economists, and all of these different groups fed into that decision-making. So I think really the point that I was trying to get across is not to wait 3 weeks to make a decision, but think very carefully at the start of an outbreak about using a model and a model on its own to make that decision. And I think that's where there's the element of caution. There are other experts that we need to make sure we tap into throughout. And of course, I think as modelers, we need to be humble and aware of that models should not provide the answer. They should only form part of the answer.
Okay, thank you very much. And one comment or question to all of you, also relating to discussions yesterday. We have experienced I think, also in the Netherlands, the discrepancy between sending a message and receiving a message, especially for the lay public. And when you talk about R0 and confidence intervals and so on, the interpretation of that sort of information by the general public is quite difficult. Would you consider to hire real experts in the future to bring across that sort of messages, like social scientists or communication experts? Do we have to take that more into account as let's say, more beta scientists?
So basically, is the question sort of recruiting more people to assist with science communication? Is that the point? I think that's extremely important. I think we are aware that it's really, really important to communicate uncertainty, but trying to determine what level we should set to communicate that uncertainty is really, really challenging. I take your point, but I also slightly disagree that I think actually during the pandemic, we actually did the public a disservice in terms of assuming they couldn't understand uncertainty. And I always draw a parallel with weather forecasting, right? We are really comfortable with the idea that tomorrow there's a 10% chance of rain and we know what that means and we make decisions based upon whether we think it's going to rain or not. And sometimes the weather forecasters are wrong. But I actually think we could explain uncertainty. I agree with you that we need to find a way to do that. But I think that actually the public would understand that we can't predict with a high level of certainty how many people are going to be infected tomorrow, how many are going to be infected next week, what the impact of this control would be. But we need to work harder to work out how we communicate that. So I don't know what the answer is, but I think it is something we need to think about because communicating that uncertainty is crucial to what we do.
Thank you.
Any other comments on that or perspectives? No?
I think our online audience, we have one question. I don't know if the slide is ready. If the slide is ready, we can have it uploaded.
But maybe just while there it is already. Here, I can read it. To Mike, you've worked in 2001 where EuFMD was spread to large parts of the UK. Would you have had the same result if you looked at the more conventional 2007 outbreak?
Okay, so we did look at the 2007 outbreak. So I mean, the problem with the 2007 outbreak is there were only 9 infected premises, and they were all sort of in Surrey or around that area. Very low livestock density area. I did actually do a little bit of work with DEFRA during that outbreak to look at potential impact of vaccination. But I think that actually was one of the examples where there were so few cases that actually surveillance was really, really important. That actually the models can only really have a limited role because it was such a small outbreak. And absolutely, we undoubtedly would not get the same result, which is kind of one of the points that I was making right at the start. No 2 outbreaks are the same, and we need to think very, very carefully. We can learn lessons from the previous one, but when the new one comes along, we need to think, well, okay, how is this different? How am I going to amend my model to take account of that?
It seems a little bit at odds with the advice, especially the question to vaccinate or not to vaccinate, which Amy pointed out this morning. We've been having the same question for 20 years. But you need to make that decision very early in the outbreak. You can't wait, or you shouldn't wait. Models have told us we shouldn't wait 3 weeks to decide to vaccinate or not to vaccinate. So those seem a little bit at odds.
And in 2007, that was the big question that we were looking at, should we vaccinate? But ironically, I mean, I remember I spent about 2 days adapting my model to build in the latest data and run some simulations to get the answer that I knew I would get purely by looking at the outbreak, that actually vaccination was going to have such little impact, with the caveat that if there was transmission that we hadn't seen to somewhere else in the country with a higher livestock density, then it could have had an impact. But again, that's something that we can't predict. Yeah, we do need to respond, but it's really hard because we can only do it with the data we've got available. And in 2007, there wasn't very much.
Okay, so I think that concludes our panel discussion and our session for this morning, where we've heard about innovations in data, but still that there's big challenges and gaps in terms of making these data accessible, talk to each other, equitable. We also heard about let me just get my notes. About the time that we need to think about new questions, as well as, still continue to work on the old questions, but new questions, and how can modeling help us to address these new questions, and how can we better communicate those results. So we really appreciate the in-depth research presentations we've had, the more oversight keynote speeches about some of the horizons ahead of us. And I think it's just been a fantastic session. So I'll hand over to the chair.
Okay, thank you very much. And that's just a remark. What we were saying today, I mean, I like the word responsibility, but all part, not just all the pressure on the modelers, because they do as what they can to do a good job. But of course, models are a part of the decision-making process, and that should be explained along with the limitations, assumptions, and around these modeling techniques, and never forget to have an open eye on what is happening in reality, because and this responsibility should be shared by all parts, and that is my conclusion. It's important. So now I think I don't know if
I think the only important thing to note is that because it's our last day, that we have a shorter lunch break today. So we'll come back here today at 1400 at 2 p.m. instead of 2:30. So please, let's go and have lunch.
Maybe let's appreciate our panelists.
Thank you.
Thank you very much. Very nice. Okay.
Thank you.
Thank you too.
Okay.
history that it might be in FMD.
And I think we've done a brilliant example of that this week.
What you need to know about foot and mouth disease. Foot and mouth disease is a very contagious viral disease which affects cows, sheep, goats, pigs, and other animals such as wild boar. The disease causes serious production losses, even if not all animals die from the virus. An outbreak in a country can result in a major block to international trade. Humans cannot become ill from the disease, but they can play an important role in spreading it to animals through dirty clothes, vehicles, and other equipment. The signs of FMD are variable and depend on the animals. In cattle, the disease can be severe, and you may notice depression, high fever, not eating or difficulty eating, decrease in milk production, increased salivation, blisters followed by lesions on the muzzle, inside the mouth, on the feet and teats. Occasionally, it can cause sudden death in young calves. In pigs, the disease can be severe, and you may notice an inability to stand, with some animals in a sitting position, lesions on the feet, sudden death in piglets. In sheep and goats, the disease is not that evident, and you may notice small blisters just above the feet. Sudden death in young sheep and goats. FMD can be found in all fluids from infected animals. The virus can spread very easily through any of the following ways. The most common is through direct contact between an infected animal and a healthy one. Contaminated materials such as hay, feed, water, milk, urine, and others. Dirty clothing, footwear, or equipment. Contaminated pens, buildings, or contaminated animal transport vehicles. Meat or other products which come from diseased animals, if fed to animals when raw or improperly cooked. And in certain weather conditions, it can spread through the wind from a nearby farm. Contact your veterinarian or local authority immediately if you suspect your herd is infected with FMD. Do not move any animals from the farm and limit any movement of people to and from the farm. A veterinarian will come and look at the animals and take samples to send to the laboratory. The laboratory will examine the samples and confirm the presence or absence of the virus. Once FMD has been confirmed, measures will be put in place to stop the disease spreading to other farms. Farmers whose farms or animals have been in contact with the farm where the disease is present will also be contacted by the local authority or veterinarian. Everyone has a role to play in ensuring that your community and country remain free from the virus. By staying alert and reporting suspected cases of FMD, you can make a difference. Thank you for your attention.
Good afternoon. I hope you had a good lunch. I would like to thank this afternoon session by thanking the participants of the workshop for the nice and fruitful discussions that we had. And we will now open the 6th and last sessions of the Open Session 2022, which is focused on surveillance and control. The session will be chaired by Giancarlo Ferrari. He is a member of the Standing Technical Committee. Giancarlo is a veterinary virologist and epidemiologist working with the FAO and also with the EuFMD. Giancarlo has been assisted in the preparation of this session by Karsten Poch. Karsten is an epidemiologist working mainly with EuFMD, mainly in the Southeast European neighborhood. Karsten, together with Sylvia, Buddha, will moderate this session. So, Giancarlo, I hand it over to you.
Thank you very much for all of you being here. Thank you very much also for all those attending remotely. So good morning, good afternoon, good evening, wherever you are. This is the last session of the last day, which is always very, very challenging because people are getting ready to go back home, including myself. And I just like to frame a little bit the content of the session. You know, the name of the session is surveillance and control. And I was thinking, you know, these are actually two distinct activities. Obviously, you do surveillance because you want to improve your control methods, control options. But that leads us to consider two different scenarios maybe that we have to take into consideration. And they will come out, I think, through the keynote presentation, but also at the oral presentation, but also if you look at the posters, you know, these different objectives. Obviously, in free settings, you want to strengthen surveillance because you want to reduce the time between the introduction of an agent and its detection, assuming that, you know, earlier you will be able to detect and more effective your control measures will be. In endemic settings, you may have different objectives. So sometimes surveillance is utilized, for example, to gain a better understanding of a specific disease, which is known to be present. And this data and information generated through the surveillance system would lead you to implement control measures and also to evaluate the effectiveness of these control measures. So these are two distinct things. I would say that in addition, in those settings where certain diseases are endemic, at least this is my experience, there is an additional challenge that whenever you are able, and I think this is a possibility that is there, to collect relevant information about the specific disease, then when you have to translate this into the implementation of control measure, that's a challenge. Because then you will face many issues like, well, we cannot do this because there is, I don't know, religious festivity, so we cannot close the live animal markets. We cannot do this. So there is an additional challenge. So there is I think there is a gap that should be considered, especially in endemic settings. So, having said this, I would like to present the first speaker of this session, that is Sumption. All of us know Sumption. I have the same difficulty when I have to say WOA, I have the same difficulties, you know, I still keep saying OIE. The same with Keith. It's difficult for me not to associate Keith with the EuFMD. So Keith, I think you have a presentation on lessons learned from SARS-CoV-2, surveillance and control and implication for FMD fast surveillance and risk reduction. So please, Keith, the floor is yours.
Thank you very much. It's a huge pleasure also to be here on the stage with Giancarlo and with Carsten, because I think it was in 2008 we helped develop the progressive control pathway for FMD, and a lot has developed since then. Also, massive pleasure to be here. I remember 2 years ago when we planned this open session in Marseille, and it was March 2020, and the session was supposed to be in October 2020, and I said, yeah, we can do it. Of course, in 6 months, you know, things will have returned to normal. So you may take with some degree of skepticism some of my projections as we go forward, okay? So I think we all have been acutely aware this is not the situation. We were not in the same situation as we expected to be 3 years ago, 2 years ago, with FMD control. Why? Because this has happened, COVID-19. Maybe we expected it for years, but it happened, of course, and we've all been affected greatly in our way. So I'm not going to say that much about our current situation, but in this 2 years, there's been a tremendous change in some of the associated measures and tools, which are probably relevant also to think about FMD. Now, just about 2 years ago, we published a paper comparing COVID and FMD, because at that stage, 2 years ago, we could say, we're in an epidemic curve. How did we do for FMD when we've had it in 3 countries? You know, and there are many similarities: preclinical viral transmission, the importance of awareness and early detection, movement restrictions and biosecurity, which we'd never really used in the human population, choices for eradication compared to living with infection, mass diagnostic screening. All these things were battles that we've had with FMD epidemics, which were applied also here. And I got in trouble in my own house of FAO for saying, well, vaccines, you know, will be good until we have variants arising. And you know, what's the evidence for that? People said. Well, I said, anyone who works on FMD knows that's the story. Do we really expect with coronaviruses to be any different? Some of the maps, now look at this map. This is taken from a group looking at the global landscape of genomic surveillance. And if you look at the scale here, high availability of sequencing of it, you'd expect that to be all OECD countries. But actually, there are countries here where you might be surprised. They're shown as having high availability of sequencing now. Why? Because sequencing strategies were elucidated for countries, depending on whether they wanted to sequence every case, which has been some countries, or they take a fraction of the cases so that they can follow to monitor the changes. As Giancarlo was saying, it depends on your purpose. That's not a map we would normally see. We would normally say good sequencing only going to be in countries which are higher income countries. Whoops, go back. Okay, this one, thank you to Don and colleagues. You know, a lot of attention to how the virus of SARS-CoV-2 is varying and what extent that will change or need change in vaccines. That's something that you guys, we guys, everyone on FMD has been thinking about for some time. How do we predict and project when we're going to need a new type A vaccine, for example? So can we learn from the scale that they're doing things in COVID? Hopefully some things will. So lots of things in terms of control measures I could talk about. I'm not really going to talk about that in this presentation. But just to a shout out to some of the, I think, spectacular things that colleagues here have done over the years, which have also been applied and of value in SARS-CoV-2. So how long ago was it we started working on lateral flow devices as a way of transporting samples? Well, it took several years, and thanks to Labib, thanks to Don, thanks to all those teams working on that in the field as well, showing it can be done and done safely. And then the team in Ireland, and I think Donal, you're here and you were involved in this, took note of this and then applied it also to SARS-CoV-2, so that from lateral flow devices, you could also pick up virus and you could do sequencing, right? So I won't say everything is heard first in EuFMD, but there are some, I think, some really good things which is a crosswalk. Diagnostic reserves. This is really the slide from EuFMD with the diagnostic banks for the Balkans. But I'll come back to this later. Diagnostic supplies in the first weeks and months of an outbreak, they were limited and became limited. So we're now thinking very much, what about pooled procurement for countries so that they have enough for their routine use as well as emergencies? Thinking of that for the future pandemic preparedness, not simply FMD. Okay, communication, having a clear exit strategy. Do we have an exit strategy for SARS-CoV-2? I think we've sort of exited into a world of perpetual vaccination, monitoring, and keeping an eye on it. And it's becoming one of those things that we have to keep under control. But that maybe is our exit strategy. But it wasn't clear, was it, when we started 2 years ago, what the exit strategy was? Okay. Modeling, you've already heard enough, or you've heard a lot on the modeling side. We can't have enough assistance on this side, but in the end, decisions still have to be made on the basis of a combination of modeling and expert advice. But perhaps the biggest thing with this model is that it brings in the resources which are limited, and that's good. Okay, let's sort of thinking further and thinking beyond. There has been a huge impact of the pandemic on agriculture and food security, and now we have new implications that come from the global economic crisis in this year. So the surveys that have taken place a couple of years ago, and thanks to EuFMD and the EMC and FAO, did really look at the impact, and there were significant impacts on veterinary service activities during the pandemic, and we maybe should look at them now and to see what extent things are still affected, if at all, by the COVID situation and the way that we can get to the field. Pirbright's, the number of samples, Don, correct me if I'm wrong, you had a plummeting number of samples during the pandemic, and that is in complete contrast to the amount of sampling and sequencing taking place on the human health side at that time. So, what do we learn from that? Where do we move on? Well, this is just some sort of thoughts on how we might be sort of changing and moving. A number of us were involved with what I think was a groundbreaking piece of work in a goat market in Kathmandu, and thanks to the Pirbright team that came out for our real-time training, picking up foot and mouth and peste de pitti ruminants from the samples. But for some related viruses, like hand, foot, and mouth disease, they've been picking it up in sewage and effluent for a much longer time. So, where are we now? And I tend to lower the tone of any conversation. So this is to talk about COVID Poops 19. COVID Poops 19 is a network of 283 universities, 70 countries, 152 dashboards, 3,800 sites where poop is being tested for COVID and putting the data together. And that shows the number of countries and sites. That's all taken place in the last 2 years, right up to the minute. And you may be familiar with looking at national dashboards. This is from New Zealand. Those two curves are the amount being detected in wastewater and the cases being detected. And the scale on the left-hand side is genome copies per person per day. So we've got about 1 million viral particles equivalent per person per day on much of that time through 2022 in New Zealand. So congratulations if you're from New Zealand for the amount of virus that you managed to put into your wastewater. Now they connect that to sequencing as well. So that's actually looking at variants picked up in wastewater over a timeline. But I think the other thing to notice is through monitoring wastewater, they estimate 73% of the population is kept under surveillance by testing on the wastewater from those populations. And 96% of the sites they picked up SARS-CoV-2. Okay. So another thing that's, I think, relevant, and there's a crosswalk between the PQ system. Don't need to say more on that because David and others will have done so. But how does that relate to COVID? Well, the huge effort of WHO with a massive number of partners setting up the COVAX scheme, which is an advanced market commitment scheme to ensure that they can leverage the higher income countries to be able to create the amount of vaccine needed for the longer term for low and middle income countries. So that scheme, flawed as it might be, was one of the things which has really been a revolutionary thing. I can't imagine setting that up, but that was set up. And I think, as Pascal was saying in a meeting this morning, this is bringing the big financiers, big thinkers into the room, working out how to create the kind of advanced market so that producers are de-risked to produce the vaccines. But you need a qualified vaccine to do so, so that links to the pre-qualification. I think the only equivalent we've got in our field, and it was mentioned yesterday, is the AgResults Vaccine Challenge, which is an advanced market commitment of its type by saying that they're willing to pay for vaccines, to pull vaccines into production relevant for East Africa. We need a lot more of these types of mechanisms because the vaccine needs are still huge. And this relates to COVID here, and African Union intends to develop, produce, and supply more than 60% of the doses it needs by 2040 by a set of partnerships to achieve that, to work for vaccine security. Because at the moment, less than 1% of the vaccines used on the continent are locally produced. What are the other things we've had to do across COVID which are relating and relevant to our area? Well, vast scale-up of training for competencies. The virtual learning centers that EuFMD, FAO has been pioneering in our sector. I think they're an example of what you can do very quickly and how you can provide a service. For COVID, the number of frontline healthcare workers that needed to be trained was practically the entire global population of healthcare workers. So you just imagine the efforts taking place to be able to rapidly scale up training. So virtual means they had to use and are being used. I don't think we can compete as FAO with WHO's 7 million course enrollments, but maybe their courses are not quite as, deep and sophisticated as ours. Okay, so what do I see as really relevant to fast disease management? And if some of these things sound future, well, we have to think about that. Wastewater and environmental sampling as a means to monitor viral prevalence at population scale. Has anyone tested wastewater coming out of infected farms? I'm not sure many people have tried it. Maybe it's not the route. Maybe the environmental sampling that Simon, Gubbins, and others have been doing in Cameroon, shows us a way. It's not difficult to pick up FMD in environmental samples. Maybe we can use that more. The modalities to bring new vaccines into use in a short time, all these advanced market commitments. There was a huge need on the side of COVID. I'm not sure how we can convince people there is a need on the FMD side, but there certainly could be a need for things like vaccines for Rift Valley fever and other zoonotic diseases where we need to be able to really rapidly scale up at times. Scaling up sequences. I'm told that over 30 million full genomes are now, for COVID, have been sequenced in 2 years. That enables real-time tracking. So bringing these things together, scaling up our sequencing centers. with new strategies for variant tracking. So when more and more use of next generation sequencing is being used, we'll generate information that is also potentially on FMD and other related diseases. A lot of that is happening for antimicrobial resistance, to look for genes, antimicrobial resistance genes in samples. At the same time, the sequence information, if FMD is there in the sample, nobody is looking at it. So there's potential to link up across these initiatives. There's a potential for much more integrated surveillance in low and middle income countries. A lot of talk about how we're going to do that. Pooled procurement is one of the routes that potentially reduces the costs of reagents for everybody. And if we consider that now we're very much looking at how we do this to ensure that for zoonotic pandemics, we can pick up in the animal population viruses of risk. Maybe pooled procurement. We have the idea of a national procurement providing to the veterinary services, their laboratories, academics, in networks, human health facilities, the sampling that they need, the reagents they need to be able to do that. It's a thought. It's possibly a way to go. And of course, high throughput, point of use genomics for field epidemiology. Some of you have been talking about doing that for years. Surely we're going to be using that for future. And virtual learning and training management for healthcare worker training programs needed across the human health, the community animal health workers also need to be part of that build-up. So with that, I thank you very much for your attention.
Thank you very much, Keith, for this enlightening presentation. We will leave all the questions to the panel session, so if any one of you from the audience, or also for those attending remotely, if you have questions which you want to direct for Keith, during the panel session, that will be the right moment to raise these questions. Okay. As you see, I'm not very much digitalized. I still need papers with me. It's age effect. It's called in epidemiology, I guess. So with this, I would like to introduce the next speaker, Angus Cameron. I think he's well known. He's the director of AusVet Europe. Angus has a vast experience in designing and implementing surveillance and animal health information systems. So please, Angus, the floor is yours for your presentation.
Thank you. Thank you very much, Giancarlo. Absolutely delighted to be here. I was talking to others about other meetings I've been at, and obviously it's been a pause, and so it's great to have a chance to come back and see everybody face to face. I'm going to be talking about about surveillance and data in surveillance, and this is a passion of mine, probably since I started much too long ago. But it's an area that I've been working in, and it's been evolving with the various needs of the world. So one of the features I'm going to be talking about is this market-driven approach. So let's start with a fundamental question that a lot of people designing and doing surveillance are asked, which is how much surveillance is enough? And this is something that I asked myself, and when we think about surveillance, it's been mentioned or referred to a little bit. There's various reasons why we do it. We do it for diseases that are present, measuring the occurrence of disease, and Kern's comment, when we measure the occurrence of disease, things like prevalence and incidence, we've got well-established ways to work out sample size. This is classic sampling theory. We know how much is enough if we set our targets. When we do case finding, it starts to be a little bit more complicated. Looking at diseases that are absent, if we're trying to quantify the quality of our surveillance for diseases that are absent, we now have ways of doing that, and we have had for, goodness knows, about 20 years. Early detection. This is something that we talk about a lot. WOA has it in the code as an absolute basic requirement for every country in order to make any statement about its disease status. You need to have an effective early detection system in place. But what is an effective early detection system, and can we measure it? So this is something that I've been thinking about. And so I want to start my talk with a few comments about that. Some of you may have heard me bang on about it in the past because it's something that I care about. But then I want to go on to another topic, talking about a project that we're working on at the moment, which seeks to try to address some of the challenges. So just going back, when my brain was young and agile, this is the sort of work that I did with the help of some much wiser heads to try and measure the quality of surveillance, in this case, surveillance to demonstrate freedom from disease. Anybody who's been involved in international trade or proof of freedom or the eradication of rinderpest, the demonstration, will obviously have memorized this formula, who knows exactly what's going on. So this is the answer to one of our problems. When it comes to quantifying the sensitivity of early detection surveillance, this is what I'm capable of these days. And it's really pretty simple. It's not rocket science, as they say. We need to understand what's the coverage of the population. What percentage of the population is our surveillance looking at? What's our temporal coverage? If we're talking about early detection, it's meant to be early, and sampling once a year does not give you early detection. And finally, how good is your test? The detection system that we're using, what's the sensitivity of that? That's it. That's the secret. That's fine. The problem is, if we want to measure our performance, and we've got this really complicated calculation, and you pick out your 3 numbers and you multiply them together, you need to be able to then say, is it good enough? And so we need to set some sort of standard. And when it comes to early detection, we're talking about the first incursion of an exotic disease into a population. Or we're talking about, in the sense of COVID, emerging diseases, and being able to get it early enough, detect it early enough so that we can do something about it. Without ending up with a global pandemic. So we've got plenty of examples of failure of early detection. We've got some good examples of successful early detection, but we need to understand what is this target. And I'm afraid, for me, the answer is relatively easy. In the future, how many COVIDs are we happy to miss? Is it all right that we let them turn into global pandemics? What proportion of those new emerging diseases do we think, that's okay, we'll let them go through? Or, in the context of FMD, if you're working in a country trying to eradicate disease, or you've been successful in eradicating disease, how many of those new re-incursions are you happy to say, oh, well, those ones don't matter, we'll just let them happen, but the other ones we'll try and find? My conclusion is that's our target. When we're talking about early detection of diseases that matter, this is what we should be doing. And if anybody disagrees, you can argue with me later, but I'm the one with the microphone at the moment. So that's fine, but there's a few little problems come in. We've heard some mention about the issues of risk-based surveillance and how we can use that first formula you saw there for freedom. That was all based on some quite interesting probability theory and hypergeometric distributions and so on. When we're using probability theory, I think it's just magic. I just love it because it allows us to predict the future. We can actually know what is going to happen within certain confidence in the future if we understand those probabilities, which is great. But probability theory is based on repeated events. We can predict the future in terms of rolling the dice or flipping a coin, and we know that if we do it 1,000 times, we will get within this range of heads or tails or 6s or 2s or whatever. Early detection. We want to detect every event. Events are not repeated. It's a single event that we're trying to detect, which means probability theory is out the window. It doesn't work anymore. And I was just talking at lunchtime about the African swine fever in Germany. And the latest detection near the Dutch border occurred in a village where there were very high biosecurity farms. And if you've done a risk-based approach where we should be doing our surveillance in Germany to detect new spread of African swine fever, these would be the last farms you would look at. These are miles away, and they have, they're the best farms. But it occurred there. And if our performance standard is we want to detect 100%, the answer is then simple. We need to, for early detection surveillance, our target is that we need to have the entire population under surveillance. We can't use these risk-based approaches and say, here's high risk, here's low risk, forget the low risk, we'll just concentrate here. It doesn't work anymore. We need to have them under surveillance all the time. And we need to use a really sensitive detection system. So, at this point, generally people say, well, that's it. Very interesting, nice presentation, Angus. Now let's get back to the real world and do what we can. But I'm a little bit more ambitious than that. And so this challenge is something that has been playing with my mind. I've introduced this in the context of early detection, but we've just been hearing presentation after presentation from the modelers saying, we want more data, we want better data, we've got problems with the representativeness of our data. And so we just don't have what it takes to do a really good job. And most papers say further work is necessary, improve data. Some of the conclusions are, or the limitations are, data quality or data access. It's just happening in everything. I'm talking about early detection, but in most of the work we do, this is a problem. So is there a way we can actually overcome this type of challenge and think about data, think about surveillance differently? Now, I have been absolutely very, very fortunate to have an opportunity to work with a consortium in a USAID-funded project that has given us an opportunity to do this. This is, it's a $34 million project over 5 years, working in 4 countries. We're working only in 2 countries, but you can see the rest of the consortium there. In our team in OSVET, we've got between 20 and 35 people actively involved in this all the time. So I'm just going crazy. This is big and complicated for me. I've not done anything like this before, but it's just so much fun because we get a chance to really grapple with these problems. And the reason we've got a chance is it's a research project. It's not a traditional development project. We're trying to look at effective risk mitigation. Okay, that's what we all do. That's our bread and butter. We need to do things about transboundary animal diseases, which is why we hear fast diseases. Also zoonoses, also antimicrobial resistance. Keith just mentioned that trio. But here's the fun bit. Market-driven sustainability. They had this idea at USAID that things aren't working with donor-funded projects, with government-funded projects. Can we explore whether the market is able to provide the sustainability that a lot of these other systems seem to struggle to achieve? Others in the consortium are using various areas. Our role in the consortium is data. We're fascinated with data and how we can achieve these things. So, breaking it down, analysing it a bit, the need is comprehensive full-time surveillance. We want to get every disease captured. And that's wildly ambitious, but that's okay. I am wildly ambitious. Where this information comes from, if we're talking about whole of population full-time surveillance, well, it's coming from the producers and the farmers. But here's all the problems that people cite, and they are major, real problems. Why would producers record every disease event that occurs? It costs a lot of money just to capture and manage that information. Who's going to pay for this? And how can we manage all of this data? So I'm going to just jump through really quickly, some of these points. Let's look at the first one, motivation. And this is something that we do a lot of work with, we think about a lot. And we look at it in terms of the benefits, the costs, and the risks. And individuals, if they're going to be involved in anything, they balance it up. They do their own benefit cost analysis. Is it worth participating in some sort of crazy surveillance system that these people are talking about? Or should I just go in and run my business? So, okay, let's look at the benefit side. And what we are working on in this project is trying to get census-level surveillance from the producers. And the idea is to provide enough benefits so people say that this is a good thing, that people participate not out of obligation, not out of legislation, but out of self-interest. And so we're focusing very much what are the potential benefits. And obviously, at the farm level, we can provide various benefits through information. And it's not just disease information. We were talking about integrated information. This is very broadly integrated information. It's to do with farm management. It's to do with farm economics. It's to do with disease. It's to do with all sorts of information that farmers can use to run their farms better. So this is nothing new. People have always produced farm management software to help people get better information to make better decisions about their farms. So that's a good start. But a lot of farmers don't use these systems. A lot of farms are making management decisions in the absence of good information. And we have to ask why and what we can do to change that. But where this starts to get exciting is the other bit. What we are actually producing is not, well, it's a national or even above national eventually, but let's start with small ambitions, a national level data integration system, census level data integration system from producers. So rather than just dealing with data from one farm at a time, we believe that if we integrate that data, then that collective data set containing all of the disease events, all of the production information, all of the drugs used for treating animals, all of the cost data associated with this, if this is all in one place, maybe this could be useful. So some of the potential value that we see from this are things like benchmarking. The research opportunities would be amazing. Surveillance, this is why I'm here. Market access, demonstrating status, demonstrating compliance. Policy development. We've had some great discussions about policy, and I'd like to have an opportunity to say some things more about that if I only had time. Production forecasting is a major issue for some countries to stabilise price volatility. So these are the benefits we can promise, but that's only one side of the equation. Farmers don't participate in surveillance because of risk, because of cost, because of the dangers, and we've heard about some of those. So how do we address these? And this is another area we're trying to look at very closely. Costs, keep it cheap. Privacy and security, keep it safe. And so what we're talking about here, I've used the word data integration. I've never said data sharing. And so we're never talking about revealing anybody's production data to anybody else. We're talking about integrating production data, analysing it in a secure way, and producing the results in a de-identified way for the benefit of the industry and for the individual. To make that work, we need to have strict rules about ownership, and we need to have effective governance so that collectively the owners of the data are setting the rules. It's not up to government to set the rules about how industry uses its private data. It's up to the data owners, the producers themselves. The other problem is, how do we pay for this? And this is the question that USAID has asked us to answer. And here's where we're having a lot of fun playing around with various ideas. But basically, we're dividing the population into data generators and data users. And obviously, the data generators are also data users. But they're investing in the system already with their data. That has huge value, and so their effort and their data is payment for the value that they're getting. Whereas other stakeholders, and we can take various groups, but one that we've found to be particularly interesting is input providers. These are drug companies, feed companies, genetics companies. They get huge value out of understanding how their products are being used in reality in the field, and how they can provide a better service to their clients. By understanding the situation on their farms. And so having a cheap, rapid way of accessing detailed information about their clients and the performance of their products is hugely valuable to them, and they're willing to pay for it. So suddenly we start to get an economic model that could work to achieve this sustainability. And so the model that we're exploring is that we've got the data providers and we've got the data users, and there is an exchange of value between them so that everybody gets benefit through the exchange of data and the exchange of value. Okay, data management, there was comments about this before. if we're talking, for instance, we're working in Vietnam and Indonesia, we're talking about hundreds of thousands of farmers. And we're talking about daily individual events on all of these farms. We're talking big data now. This is serious stuff. And so cloud computing, big data systems, we've got it now. It's under control. We can manage it. We're using the same platform as we developed for Indonesia. Omniscient is its name. The name sort of says it all. We want to know everything. It comes from omniscient. So that's what we're trying to achieve. A couple of quick snapshots of the system that's being developed. As I said, it looks like farm management stuff, but it's actually national data integration for the benefit of producers and provision of public good at the national level. Working with pigs, working with poultry, soon hoping to work with ruminants, aquatic animals, and more. This is just an internal one. This is very much underdeveloped at the moment, but the message here is a quick look at the menu system to give you some idea of the breadth of integration of data that we're talking about. Certainly disease, but if we're going to provide benefit back to the data providers, we need to answer their problems. And their problems, they've got many more problems other than disease. If we provide them with benefit, because we give them a tool to manage their own data, then they will use the tool. That's the hope. And it's actually what is starting to happen as well. Here's one of the tools we are providing. And this is embedded in the system. This is something that has been, I'll credit, way back in the late 80s, Pauline Breitling, Mike Larkham, and the team in Victoria and Australia for starting this work with a Bayesian computer-aided diagnostic system for dairy cattle. And we've, Richard Shepherd, PhD student of mine, years ago, picked this up and adapted it for cattle in Northern Australia, and now we're extending it and using it for as the basis of what we're calling an antimicrobial stewardship system, a real-time, live antimicrobial stewardship system. So where people can describe the signs and get a differential diagnosis or a ranked list of diagnoses. The next step is then to get treatment recommendations based on those which are probabilistic assessments of the likely efficacy of different treatments and advice ultimately on steering them away from the inappropriate use of antimicrobials. It's a little bit of magic because it's providing very practical use. It's packaging up a whole lot of expert knowledge into a very simple system. But in the process, it's collecting surveillance information. We're finding about cases of disease and what signs these cases are demonstrating. We're collecting drug usage, and particularly antimicrobial usage information. So for surveillance, it's lovely. And at the same time, we're nudging them in the direction of more responsible use of antimicrobials. So that's just one element. I am almost completely out of time. So a couple of quick conclusions. I've been talking whenever I get a chance about this concept of big surveillance, and so this is what we're talking about. The potential to have big data, and we're not talking about citizen science collected data. We're not talking about accessing available data sets. We're talking about generating properly structured, well-organized, high-quality, census-level information covering the whole population and using that for our studies, for our surveillance, for our analysis. That's our dream. The missing bit is, hang on, you're saying census level and it's voluntary participation amongst farmers. Why do you think they're all going to be involved? And I don't think everybody's going to be involved, but if we design it properly, if we focus on the direct benefit in providing services to the producers, then I believe we can get very high coverage. The experience from Indonesia in the other system I mentioned, e-sickness, has shown that that's actually been quite successful. Market-driven approach. What we're trying to do is address these problems of donor-funded projects or the challenges, particularly in low and middle-income countries, with competing demands for budget, which means that great systems, great approaches sometimes fall over because the funding just is not sustainable. And so if we can go to the people who are actually getting financial benefit from this type of information and from the data and the decision support advice that's being generated, if they're making more profit, then they will invest in having access to these systems, which starts to get the sustainability wheel turning. And as I said, it's an exchange of value. And I'd like a chance to elaborate on that, but I've got 46 seconds left, so I probably won't. But the principle is that if one sector has data but doesn't have a motivation for disease control, and another section needs data but has a motivation for disease control, let's get this exchange of value going on. So the opportunity for a high biosecurity, highly intensive production sector to get surveillance information about low biosecurity backyard or village level production systems that are generating risk in their neighbourhood. If they can understand the diseases present around them, they can use that data to increase their own biosecurity, to prevent those problems, change their vaccination schedule. If they're getting that benefit, they're making money from it, that money can then go back and support the improvement of surveillance in the smallholder systems. That's a very simple example of this exchange of data concept that we're talking about. We're using it in multiple areas, but that's what we're trying to do. Just so you know, the project is entering its second year. We've got 5 years, and if I get a chance, I'll keep you updated because there's some really exciting work going on. Thank you to the moderators. Thanks to all of you, and I'll answer questions later. Thanks.
Thank you very much, Angus. I think it's a very fascinating point, is trying to align interests of stakeholders and those who are responsible for disease control. But we will leave the questions, as I said before, to the panel session. So the next presenter is Dr. Simon Gubbins from the Purevsuren Institute. He's going to present on longitudinal animal and environmental samplings for FMDB in northern Nigeria. So Dr. Gubbins, please, the floor is yours.
Okay, excellent. Thank you. Welcome to everyone in the hall and online. So I'm going to talk about, as was introduced, this a project that we've been doing in northern Nigeria, looking at longitudinal sampling for foot and mouth disease virus. And although it's me up on the stage, coming to, and actually getting a nice trip to Marseille to actually present the work, I'll begin by actually acknowledging everyone who actually did the really hard work. So these are the people at the National Veterinary Research Institute in VOM, who actually went out collecting the samples for us. For people at Purevsuren, who were involved with the study design, some of the training, and then actually processing the samples. And then thanks to our funders, so EuFMD funded this study with additional funding on the UK side from DEFRA. So to introduce the background to the project, and Keith's already alluded to some of this. So environmental surveillance is a potentially very useful tool for fast diseases. So environmental samples, they're simple to take, they're non-invasive, and it's relatively cheap, so cost-effective. And to give some examples, we've done this for foot and mouth disease virus, and this is actually on the top picture. If you look carefully in the middle, that's actually me taking environmental samples to prove how easy it is actually to do. And that's at the Nepal, in the Kathmandu goat market in Nepal. And then the bottom is the study Keith mentioned in Cameroon, where we were sampling at livestock markets for FMD. So we've used the techniques, and we've shown that we can actually find the virus quite easily within samples. But the question is, well, how does this actually relate to other surveillance results or surveillance methods? And in particular, how does a positive in an environmental sample relate to virus circulation? So are you always going to find positive samples, or if you start having outbreaks or more virus circulation, are you actually going to see an increase in the number of positives? And then there's also, again, something Keith alluded to, is having a positive sample. I mean, obviously, an FMD positive sample in a disease-free setting is very important, but it's of less value in an endemic country. So, what additional information can we get from an environmental sample, and in particular, can we actually generate sequence data from these? So, to address these questions, we're working in northern Nigeria, and as a result of an earlier EuFMD project, we identified 2 high-risk areas in northern Nigeria based on serological surveillance. And this is Bassa, which is the browny area, and Jos South, which is the greeny area. And we undertook monthly sampling within each location between March and July last year and September, October. Unfortunately, due to the security situation, the sampling teams weren't able to go out in August. So we don't have any results for August. And in terms of the sampling design, in each area, we were sampling at 5 households, 1 livestock market, and 1 transhumance location. And in terms of animal sampling, we were taking 9 samples, and ideally that was 3 sheep, 3 cattle, and 3 goats. But if there weren't enough animals of each type on a farm, they were made, or location, they were made up, sort of the other type. But the emphasis here was actually on small ruminant surveillance, so sheep and goats were preferred. And we took serum samples and oral swabs. And I'd say for the sampling at the households, we were sampling the same animals at each time if we could. But obviously that wasn't possible at the livestock markets. And then we're also taking about 10 environmental samples. And this is kind of examples at the top. So sampling places that have come into contact with the animals, so troughs, sort of fences, herders' sticks, boots, ropes, sort of anything that's potentially contaminated with virus. Okay, and what did we find? Essentially what we found is kind of, in the first sort of sampling period, so between March and July, we got very few positive samples. We got some, so kind of maybe kind of sort of 2 positive environmental samples in April, and then kind of between 1 and sort of 1, 2, or 4 positive serum samples, and obviously no positive oral swabs. But then we saw the switch in September and October, when we see kind of a marked increase in the number of positive samples. So kind of going up to sort of almost a quarter of the samples are positive for the environmental samples, and 10% of the samples are positive in the oral swabs. And we can look at this in a bit more detail for the actual longitudinal results. So here, on the left-hand side, we've got the results from Bassa sampling, and on the right, just south. And then the top line is the environmental swabs, middle is serum samples, and the bottom is oral swabs. And the colour represents kind of what proportion of samples were positive. And then each row is for the households, the livestock market, or the transhumance location. And what you can see is between March and July, kind of we have very sporadic, we have occasional sporadic positive results, but we don't have kind of sort of high proportions of positives. And then that pattern persists in Bassa in September and October. But in just south, you can see that the proportion of environmental samples increases markedly at kind of most of the households we were sampling and the livestock market. And again, sort of a similar increase in the number of positive oral swabs at those locations as well. And to put that in context, we know that for the March to July sampling period, there is at least 6 months since the last outbreaks had been reported in the areas. Whereas for the September to October sampling period, we know there are ongoing outbreaks kind of within the area. Okay. And then I kind of mentioned what more information can we get from these samples. So we selected a proportion of the positive samples for sequencing, so 16, and we actually managed to generate sequence data for 5 of those samples. And here we're using kind of a probe enrichment technique implemented on an Illumina platform. But if you want more details of that, I'll have to leave you to someone who actually understands the technology, which isn't me. And what we found was 4 positive environmental samples and 1 cattle sample. And these are all serotype O, East Asia 3, or East Africa 3 typotype. And they're closely related to other viruses isolated at the same time. And you can't actually read the identifiers, and the laser pointer's not going to work. But essentially, kind of the bottom 3 highlighted sort of sequences, there are environmental samples which were taken in September from the households. The next one, sort of the second one down, that was a cat from a cow sampled at the same time as one of the environmental samples, so it's slightly differently sort of diverse from the environmental samples. And the very top highlighted one was an outbreak at the same time, but further away. Oops, I'm right over time. I know that's 7 minutes, isn't it? Anyway, so in terms of conclusions, so what we can show is kind of FMDV RNA can be detected sporadically in environmental samples when there's no outbreaks occurring, but it's generally at a relatively low level. And then when we start seeing virus circulating and causing outbreaks, we see a higher number of positive samples at that time point. So environmental sampling does have the potential to actually reflect kind of what's going on within the field, rather than just kind of you always find positive samples. But obviously, kind of if we're thinking about using environmental sampling as a means of early detection, we'll need to sample more frequently than monthly if you want to actually be able to sort of catch up with the virus. And obviously, we've shown that samples can obviously generate sequence data, or we can generate sequence data from environmental samples, but it requires more sensitive approaches than we would normally have for the standard sort of next generation sequencing protocols. And as I've still got a couple of minutes, and we're not asking questions, I'll mention, as well as, so we were focusing obviously in the talk on foot and mouth disease virus, but as we were sampling, taking the environmental samples, we also looked at for what other fast diseases we could detect within the samples. And what we found was that we also got positives for PPR virus as well. That was about 9 positive samples. And we also found positive samples for capripox viruses. There was actually 2 each of sheeppox, goatpox, and lumpy skin disease virus. So kind of, although we haven't looked in detail for sort of these other fast diseases, we can actually see that environmental surveillance definitely kind of has the possibility as a monitoring tool for those as well. And with that, I'll say thank you.
Thank you very much for presenting this very interesting piece of work. Again, questions will be during the panel discussion. And so now I would like to introduce the next speaker. Presenter is Dr. Vasily Basiladze. He's the Chief of Veterinary Officer of Georgia, and he will present on the PCP FMD progress in Georgia and how it advances pest control.
Thank you.
The floor is yours.
Thank you.
Yeah.
It's a great privilege to make a presentation on EuFMD open session. It's probably a bigger privilege to make a presentation about the success of your own country. So thank you very much. I will not take too much time, and I will try to be as quick as possible. I have a short presentation regards activities about FMD in Georgia. So this presentation, I would like to start with the good news that we received not long ago from Regge of Western Eurasia, that they accepted Georgia's advancement to PCP FMD stage number 3. In order to achieve this success, several steps forward were taken, such as FMD prophylactic vaccination in large and small ruminants, FMD risk assessment through ARC-Gs, FMD, SP, and NSP serosurveillance in large and small ruminants, FMD clinical investigation in candidate zone, evaluation of vaccine quality, awareness campaigns within the farmers, regional simulation exercises, workshops and trainings, and OCP has elaborated. For FMD prophylactic vaccination in Georgia, it's done by risk-based, and in eastern Georgia, we are vaccinating total number of animals, large and small. In western Georgia, animals located in high-risk area. For visibility purposes, I put this diagram on the bottom of the slide, and you can see in blue color indication for vaccinated animals for large ruminants, and green color indicates animals, small ruminants. This is the map of Georgia, and in blue dots you can see vaccinated villages and points where we vaccinate animals against FMD. Many thanks to EuFMD team. Together with them, we trained our veterinarians, and we taught them how to create risk maps. And together with them, we determined the risk points, such as animal density, seasonal migration, live animal markets, veterinary inspection points, and etc. and etc. And on a map, you can see above map is risk map for import and export, and below risk map for internal migration within the country. FMD, SP, and NSP serosurveillance. It's done by 4 risk categories. NSP serosurveillance, such as villages with high-risk categories, villages low-risk categories, migrating animals in eastern Georgia, and villages in candidate area. For NSP serosurveillance, each year we do 5,000 samples, and it's followed by 500 samples as a follow-up. For SP serosurveillance, we do 500 samples each year. You can see results for SP and NSP in orangish color in columns. You see NSP results throughout the years, and as you can see, pretty successful. And the blue line indicates results in SP. And as you can see, past 3 years, prevalence been more than 70%, or at least around there. Also, NSP surveillance by each risk category that I mentioned earlier. And as you can see, the numbers are pretty successful throughout the years. NSP numbers, for example, in high-risk zone, it started 2016, it was 18 points, and it dropped down to 5.1, and same in each risk category. Candidate area. Me and Karsten, we've been talking about candidate area, and before we go to a discussion, I will I will continue how we selected candidate areas. Certain criteria were set, for, such as geographically isolated territory, only few entrances to the region, selection clinical, strict clinical conditions, no seasonal migration. In 107 villages, 3,074 animals were investigated, and all the data was entered in electronic system. And on the map up there, you see that's the area we're talking about. Evaluation of vaccine quality and immune response in naive animals. Together with the EuFMD team, we did certain concrete research, and in order to conduct the research, following criteria were set. Same for to choose the region, and also to set a protocol for the research. For selecting area, we did low NSP circulation in the area with non-vaccinated animals, area existing with both species, large and small, and the toughest one was to convince the animal owners to allow us take the blood samples from the animals, because each time we had to take a few samples from each animal, and it was really tough. For the protocol, species, large and small, like I said, status, animals with free from FMD and antibodies, not vaccinated against FMD, age 6 to 12 months, totally 20 sheep and 20 cattle. And identification by ear tag, individually identified animals, and housing animals should be kept in local area. As for results, seroconversion, post-single vaccination, it was 60–80% responsive animals, but very fast antibody decay, which leads to low antibody levels and insufficient immunity. For booster vaccination, is necessary to elect a strong and fast rise of antibody, and duration estimates should be planned. Species, small, better immune response in small ruminants than in large after single vaccination. And particularly for type A and Asia 1, opposite after booster vaccination. This was the results. Information campaign. I will not talk about it much, but I will say it's very important. Simulation exercises. First one, we did it in 2016. Second one was in 2019, right before the COVID. And next one, we are hoping that we will do it in 2023, together with EuFMD and all the countries, all the neighboring countries and other stakeholders involved. A few words about fast disease and control. First outbreak of LSD in Georgia, we had 2016, and the second one was in 2018. I mean, that was the last one. We vaccinate animals against LSD, 200 doses of Nisling strain. We received donation from European Union, and many thanks to European Union. Other than that, yearly we purchase vaccines. It's mostly sheep and goat pox vaccine, And on the bottom, you see vaccination against the LSD. PPR. Last outbreak was in 2016 in Georgia, and on the bottom you see vaccination statistics. FAO donated 900,000 vaccines to Georgia. First donation was in 2020, and the second one was this year, 2022. They donated 500,000 vaccines. Thanks to FAO, we are keeping it under control. This year, we vaccinated almost 300,000 animals. Serosurvey was conducted by DITRA funding, and 3,200 animals have been tested. And on a map, you see prophylactic vaccination chart. Cold chain. It's very important to vaccinate animals, but it's also very important how you do it. And when you keep it where it's supposed to, I mean, you do it where you're supposed to. So this is the cold chain units that we have in Georgia, the transportation unit, the cases called cases for veterinarians to carry vaccines in the field, and the stationary cold chain unit. A couple of words about One Health approach. We have, and especially nowadays when everything is digitalized, we have a so-called AID system. It stands for Electronic Integrated Disease Surveillance Systems. Both veterinary and human health laboratories, together with the NFA, are integrated into this system. And the system ensures quick exchange of information and creation of database. Couple words about identification and registration. I tried to change this slide because on the other slide I had the FAO logo there, because FAO supported Georgia in creation of NIDE system, which stands for National Identification Traceability Systems. This is the interface of the software. We made it in 2018, and since 2018, all the information about large and small ruminants are into this project. Into this program. It has 22 modules, such as vaccination module for slaughterhouses, module for border control, and et cetera, and et cetera. So what I'm trying to say is that all the information is in NIDE system, and it has quick access. You can access this program from any computer that has access to internet. So it's very modern and very digitalized, I would say. And once again, many thanks to FAO. This is it from my side. Thank you very much, and I'm ready to answer your questions. Thank you.
Thank you very much, Dr. Basiladze. So with this presentation, I think we are now ready to shift into the panel session. But before we set the scene for the panel session, I will leave the floor to my colleague, Carsten Poch, that is the moderator. I think you have something that you have.
Hello. And before we start with the panel session, we want to present to you a video on an initiative called the Statement of Intention. That is an example of a regional collaboration that has started in 2016 in the Trans-Caucasus and neighboring region. And Vasily, who just presented to us the situation and successes from Georgia, represents one of the most active countries in this initiative. But please see for yourself a 3-minute video, and then we start with the panel discussion.
Statement of Intentions. A mechanism for FMD fast risk reduction and improved disease preparedness, surveillance and control. Hi, my name is Alexander, and I would like to explain more about the Statement of Intentions. In 2016, under the Global Framework for the Progressive Control of Transboundary Animal Diseases, a Statement of Intentions was established between the veterinary services of Armenia, Azerbaijan, Georgia, Iran, Turkey, and the Russian Federation for an intensified collaboration in the prevention and control of FAERS diseases, and capacity development for surveillance and improved control programs. The EuFMD assists the implementation of the Statement of Intention activities through its work plan. Countries agreed on cooperation in a series of actions aimed at improving the confidence in the effectiveness of control programs and at reducing the risk of introduction and spread of fast diseases in the region, such as sharing of information on FMD outbreaks and vaccination, testing of emergency preparedness through joint FMD simulation exercises, FMD post-vaccination monitoring to better assess the quality of vaccines and effectiveness of vaccination programs, improvements of epidemiology and laboratory networks in the West Eurasia region, risk-based surveillance for FMD and other TADs to improve early warning. To account for the often observed move of new FMD viruses from Asia to the European neighborhood, which then pose threats to Europe, Iraq and Pakistan were included into the Statement of Intentions activities in the last 2 years. The Statement of Intentions data platform facilitates monthly updates on FMD outbreaks and vaccination progress. Turkey, for example, immediately notifies outbreaks in the border vicinity to its neighbors. Prices of live animals and meat, which are important drivers for animal trade, are also reported. This data sharing platform, together with the quarterly reporting of fast diseases, are the important features for continuous fast risk assessments and risk reduction in the European neighborhood. To improve regional risk assessments and early warning, the activities under the Statement of Intentions will be further extended to other fast diseases and increasingly include risk mapping. On a larger regional scale, the Statement of Intentions also supports the improvements of epidemiology and laboratory FMD networks in the West Eurasia region under GFTADS. This Statement of Intentions cooperation agreement can be a model for other regions to enhance early warning, build confidence in the effectiveness of control programs, and reduce the risks of fast diseases. Thank you.
Thank you, Alexander.
Okay. I think we are, yes, please, Keith. Yes, one chair more. Yeah. Okay. Okay, guys.
Yeah. Okay. Let's start with the panel discussion. Thank you very much to the presenters for these really inspiring talks. It's always nice to hear from a range of backgrounds, global policy, from a country view to research. And so we have, I think we have a lot to discuss. Keith presented on SARS-CoV-2. The global epidemic and similarities between FMD and this pandemic disease. I don't want to go on the similarities, but I think there's a lot to discuss on this. On the other hand, also surveillance and early detection plays a big role, and here probably things like the early detection and the view of problems and difficulties, like in endemic countries, in terms of what consists an expected or an unexpected epidemiological event with resulting responses are probably topics which will come up in this discussion. So this panel discussion is open. And yeah, if I may ask the first question to Keith, with all the similarities he presented, and also what we heard in terms of new developments in FMD research, like environmental sampling, like also the increasing importance of socioeconomic aspects. Is there a need? Do you see a need to bring the global strategy closer to the One Health paradigm?
You meaning the global strategy for FMD?
Global strategy for FMD control, yeah.
Yeah, a really interesting question. I think what's our global strategy for pandemic prevention? I suppose that's one of the biggest topics at the moment that's occupying a lot of the international space. So I think what Angus has brought in is where people view what animal health services are and what they're for. And now, FMD, we can say, you know, some people would say, well, that's not One Health because it's animal health, it's not zoonoses. But actually, we've changed our view really in a big way, I think, as international organizations now, and viewing that animal health, you know, without healthy animal production systems, which includes FMD, you know, we're not going to make that much progress in some of the other areas that we have to make progress upon. I'm not quite sure global strategy at this point is maybe I think there's a lot that's still very, very good in terms of understanding what's valuable to countries, what their aspirations are on FMD, and what is an optimum position for them. I think and I probably stop at this point, but I can see some crosswalk across, and that's why I was trying to sort of suggest that we need to make sure that the gains that we're going to get from more investment in pandemic prevention, including the animal health surveillance side, do genuinely lead to system transformation in the way that we think about other diseases, and FMD being one of those.
Okay, thank you. Maybe following up on this, looking at COVID control, we have seen that like zero COVID strategies come with a lot of problems. And on the other hand, we have FMD and FMD freedom with taking a similar approach. Do you see views and developments kind of reviewing, softening this approach?
Is that one for me as well? You know, I think we know from country work that many of us have been involved in. One of the most difficult questions we ask to a country is, what's an acceptable level of control to you for FMD? And they usually find that extremely hard to answer. It's surprising. Yeah. So what do you consider is acceptable? And I think that's where we are with COVID. What do we consider is an acceptable level of this balance between vaccination, revaccination, the rates in which people enter into intensive care? You know, do we accept this as being the only way of going forward? Which is actually what the case of many of our countries are for FMD. They've essentially accepted that there is a burden. They're going to live with it. They're going to spend a little bit maybe on vaccine. The rest they leave to the livestock owners. If livestock owners can access vaccines, not all can, then you have that balance. Yeah.
Okay, thank you. Maybe others who want to talk about one or two of these questions?
I can make a comment is because, you know, initially when the pandemic started, I think many countries around the world, they adopted a zero tolerance policy. So we wanted to eradicate COVID-19. I heard in my country, Italy, you know, saying, we have to vaccinate 90% of the population, 95% of the population. Obviously, if you want to vaccinate 95% of the population, it's because you want to interrupt the chain of transmission, which is not possible for me with, you know, vaccines which have a very, not a long-lasting immunity, so you have to revaccinate every 3 months, basically. So that's impossible. So there are some similarities with FMD in this regard, because also in the FMD world you have vaccines which are usually not providing long-lasting immunity. You have the same challenges as RNA virus that can frequently change. So and also I think what has happened is that many countries, I think, they shifted toward a more, let's say, a policy to cohabit with the COVID-19 and choosing a more focused protection for elder people which they suggest, to be vaccinated because it's the segment of the population that is at highest risk. So that
risk-based strategic plan, then.
Exactly. Yeah.
Sorry.
And there are a set of indicators, which have been built. Going to your comment in saying what is the acceptable level of now in the field of the COVID-19. I think some indicators have been built, like you don't have to exceed the intensive care unit occupation by a certain number of. You don't have to have a certain rate of hospitalization. Maybe that's a challenge, for us to try to build this indicator to define what is the level, the acceptable level of.
Yeah, what's acceptable.
Yeah.
Could I maybe just make a short comment on that as well? Looking at it through this market-driven lens that I've been talking about, and I suppose there's government policy, there's assessments of impact on the population and so on. But one way of responding to this is to understand the perspective of the producers of the market. And so if the disease is causing impacts which limit the capacity for profitable production, or particularly because the producers in a country are very diverse, and you'll have quite different segments. And so, for instance, there may be a segment of the industry that is looking at export opportunities, and for them, eradication to achieve freedom to allow export markets is a priority. For other smallholders, it may not be. If the economic incentive to achieve freedom is adequate for the intensive producers, in order for them to support eradication amongst those sectors for which there is no particular benefit, then there may be a mechanism by which the eradication can be funded and achieved from an industry perspective, from a market-driven perspective. If, on the other hand, as appears to be the case in a number of countries, that exports are not a major concern, or the export markets are not FMD sensitive, then it's a matter of, do we live with it? How do we manage it to a level where it's impacting our production? But there are going to be, at a certain point, there's going to be certain market forces which help make that decision for us. It's not solely the responsibility of government to make that call. The producers are actually dealing with these problems themselves. And what I was describing was actually a means of developing systems to help that negotiation between different market players, including the government, because we actually consider the government to be a market player as well, to negotiate those types of solutions and to negotiate the funding for those solutions.
Yep. Thanks for this addition. And maybe I have a question to follow up on this. Early detection, early reporting are key to implementing control measures. You have worked and are active in endemic countries. So disease like FMD, which does not have economic implications so drastic for small ruminant production. What is the incentive for and what are the mechanisms for reporting or not reporting the disease in such systems? And maybe as another segment of where we get under-reporting from is like self-reporting. We're talking about farmers as stakeholders and how to take them on board. Reporting often brings a stigma with it in the community, bringing in measures, bringing in the government, bringing maybe depopulation or any drastic measures. I mean, market-driven approach. Focus on finances, on money, but there is more to it. So what are your solutions? What have you encountered in your work on this? And how can this what are ways to overcome it?
I think these are very, very complex and difficult questions, which is why somebody's paying us money to try and solve them. And whether we'll come up with solutions, I don't know. But the framework we're examining these questions is a little bit, as I said, if we have FMD in small ruminants, which is very difficult to detect, and there's very low motivation amongst producers to report it because it's not causing significant economic loss. We're either in a context where this is not an economic disease and eradication is not feasible or worthwhile. Or we have a industry sector for whom it's not worthwhile, but there are other impacts on other industry groups. So if there's intensive dairy production in the same country and there's spillover from sheep and goats, but it's causing major impact for dairy, then it does matter. So what we are looking at is, can we negotiate, can we work out an exchange of value so that if the intensive industries for whom there is an impact, the economic impact is adequate to motivate them to try to do something about it, they will make more money by controlling it in the sheep and goats, then there is a motivation for them to invest in that. How can we produce a mechanism whereby they can invest in better surveillance and better control in another sector? Well, there's the challenge. But I think maybe Simon gave us an example of how that can be done. If there is a mechanism for the exchange of funding, so subscription to a service that gives them good surveillance data or gives them the capacity to assist with better control, then the types of environmental sampling, having structured projects which are funded by other sectors of the industry could be one of the ways forward, so that there's an opportunity to improve our surveillance even though the sector itself doesn't see much motivation in it. Collaborative effort across the whole industry can provide the same outcome. Yeah, Keith?
Yeah, I mean, I guess the question is are we here to improve surveillance, or are we here to have a sustainable system for better animal health? And I think what Angus is saying is, in the end, we want the sustained system for animal health to be in place, and at the same time to generate co-benefits in terms of better information. So I think what you're focusing on is how to make it work for the people who need that information, and ensure at the same time we harvest other things that might be valuable. So examples for it, how do we reduce the level of antimicrobials used on farms? We could take an individual disease by disease approach and say, there's a lot of FMD that's driving antimicrobial use. Or we could assist livestock owners who are interested to know whether their health is actually better or worse than someone similar to them in the same environment, their neighbor, perhaps. And if they find they're using more antimicrobials, which presumably your system will say, then you have benchmarking and you compare yourself with others. So this is, I think, what we're challenged to do now is how do we result in changing the system and with a co-benefit to do the things that you are talking about.
Can I also answer the second part of your question, which I momentarily forgot? But this issue of stigma, I think it's an interesting one. The lack of motivation for reporting, notifying disease because of social context, because of the repercussions, and I think that's an important issue. It falls into the area that I was talking about, looking at risk and balancing consequences of being involved in a reporting system. And social stigma is one of them. Draconian government response is another, that even though it might not be an appropriate response, there are some situations where depopulation will occur, and so there's a risk of that. Other negative consequences of disease reporting, quarantine and so on. So all of these things are real, all of these things are having an impact on reporting, and managing those is really complicated. But I suppose, and I haven't got an answer, but maybe I've got a couple of ways forward. One of them is related to negotiated rules around the way we deal with disease data, and so this is why we're talking about having a governance system, having data oversight committees that set the rules for the use of private data. This is what we're talking about is private production data. It's not official reported government data, and so the owners have the right to use it, apart from official legal systems for official notification. But if within those settings, communities and producers can decide whether, we actually do want disease to be notified, we recognise that there's stigma, so within our own reporting, we will maintain anonymity, for example. That could be one outcome. there's obvious challenges to implementing effective control in that situation, but at least you get better reporting. So there's a range of options that could point the way forward to dealing with that. The other side of it is to think, okay, why is there stigma? Let's look at the sort of sociological studies to say, is this stigma because of social attitudes towards the disease, or is it stigma because of the government response to the disease, or are there other things involved? And so we need to understand that better in order to effectively respond to it.
Yeah. Do I get another chance to come in?
How can you not?
I mean, now we have these sort of debates between the sort of human health side surveillance, where there appears to be vast amounts of information, and they see the problem as being data exchange in a way you can understand, you know, the metadata behind things. And when I say in animal health, it's completely different. We actually don't have vast amounts of laboratory-based data for the reasons of there are trade-associated issues. Now, one of the things that could change that is disruptive technologies, you know, such as Simon is working on, which is going to potentially change things. So Simon, with a whole network of environment sampling and universities and other people contributing data, people won't be able to control all of that, will they? They won't be able to stop it. It'll be out there.
Yeah, I mean, sort of, when we were chatting before the session, Keith told me to think big. So basically, he's convinced me that I now have to set up a global wastewater FMD surveillance system. So that kind of watch that for sort of 2 years' time. We'll have it kind of up and running.
Good.
But seriously, I think kind of these are obviously approaches that we can use. I mean, thinking about systems level surveillance, this is environmental surveillance. We've been looking primarily for FMD, but obviously any virus or pathogen that's shed into the environment, you can go sort of use environmental surveillance for that. And we've been thinking especially about livestock markets, because these are obviously areas, sort of places within sort of the farming system where animals always come together. So if you want to monitor what's going on within the system as a whole, you could potentially go into sort of the markets, take the environmental samples, and kind of use those to kind of work out, well, then perhaps even in the longer term metagenomic approaches, to actually kind of, what pathogens have we got circulating, both kind of human, sort of animal, sort of pathogens, and sort of take sort of that approach. And that's kind of perhaps a kind of a longer term vision, and getting away from kind of, we still tend to think about, or seem to think about, single diseases in isolation, rather than thinking about kind of almost the pathosystem as a whole.
Okay, thank you. We have a lot of, I mean, there were quite a few questions on environmental sampling piling up from our online audience. Maybe we flip around and give Buddha the chance to allow one or two questions from the online audience, and then we see how much time we have left for our discussion here. Sorry.
Thanks very much, Carson. I'm being told that we are running out of time. But there are a number of questions. We will see them in a minute on the screen. But maybe one to Vasily, perhaps. That's a question. Interesting, these veterinary inspection points. Would you see any value of environmental sampling for fast surveillance?
Sure. As you know, Georgia's been advancing, and we've been investing a lot into veterinary profession and into veterinary services. So yes, why not? I mean, every year we take several samples. We take to laboratory. International organizations such as you've been involved in progressing Georgia into different level. And I also would like to mention that progressing to PCP stage number 3, this is very important for Georgia, and hopefully soon we'll be able to advance to the 4th stage and finally receive the free status, to encourage farmers to produce more, and that way we will be able to access also European market as well.
Great. Thank you so much, Vasily. I'm being told that we don't have any more time, so I do apologize for those who ask questions, but I will refer these questions to Simon and to Angus to answer those remaining questions posted online. Thank you.
Thank you.
Back to you, Carson.
Yes.
Thank you. Please stay there because I think that we have online our director, the director of the livestock production and health division in FAO, Dr. Tanawat Tiensin. I have to, well, introducing Tanawat, I should say that not long ago, I think a couple of weeks ago, during the FAO Science and Innovation Forum, he organized a great event on how science and innovation can support the sustainable livestock sector transformation. And I think it's very much in line with this theme of this open session on digitalization and innovation to control FMD and similar transboundary animal diseases. So, Dr. Tanawat, if you want to say a few words, we are in the middle of the last session of our Open Session 2022. Over to you.
Thank you very much, Fabricio, and also very good afternoon to delegates and also participants, speakers, and also experts who are working on FMD and also others similar transboundary animal diseases. And actually, I know that my colleagues, Keith Sumption, the Chief Veterinary Officer, is there, and also follows your discussions on these particular issues, and also actually last 2 or 3 weeks when the Chief Veterinary Officers of the European member state came to Rome and discussed about the work plans and also exchange views on EuFMD and also others transboundary animal disease. I think it's because some of them I have met most of them before when I was the deputy delegate of the World Organization for Animal Health, or OAE, when I worked at the Department of Livestock Development in Thailand. We know that the digitalization and innovations is a tool that can help us to improve our disease surveillance. And also this device is change the way of our work. This small device, the mobile technologies, is really changed the way that we perform our surveillance system. And you can see that. I can show you very simple. You can see that in these applications, I can see the report of the disease reporting system in Thailand, or so in some other countries in Asia easily. We can keep our eyes, we can keep our ears to get all reports from the field. We can get all the reports from our farmers, producers, or even though now general public also can report to us when they see any suspected cases of animal disease, dead animals, sick animals from the field. And digitalizations and also innovations from other issues, whatever in terms of laboratory diagnosis, that we need to increase the capacities at the country level to make sure that we can prevent, to have improve the better preventions and controls of transboundary animal diseases, including FMD, because FMD is still, I think, is one of the most important endemic diseases that cause socioeconomic impact in most of the countries, in not only in European member states, but also in Africa, in Asia, and so on. I'm sure that you are discussing the technical issues, new development, new tools, techniques. And that's why when Fabrizio just mentioned earlier, just last week at FAO, we organized the FAO Science and Innovation Forums, and also at FAO Animal Production and Health Divisions, we also have the sessions on science and innovations for sustainable livestock transformation that we brought here the experience of EuFMD to share with our member states, to share with our audience how we using or applying epidemiological modelings to support the member states to improve your prevention and control policies and strategies at the country level, and also how the epidemiological modelings provide support in terms of decision makings. And I know that during this week, I think you started your discussions on, I think it's on Wednesday and Thursday, and today is Friday. You will get a lot of information from different experts and also scientists who are participating in the meeting. And then once you go back home to your home countries, you bring the new ideas, you bring new innovations, and also all the knowledge that you gain during the meetings to make sure that we can scale up, we can replicate some of the good work from other countries and can implement and can do more work in your countries. And FAO, as we are hosting the EuFMD and also FAO Animal Production and Health, will continue to support our members. And also, we will continue to make sure that right now, when we're talking about livestock sectors, we are in the times that we have a lot of challenges that we need to move forward for transforming livestock sectors to be more sustainable, resilient, inclusive, and efficient. Because in each regions have a different priorities. I had an opportunity to visit some members because today is my almost 60 days in the office. But last month, I had an opportunity to visit some farmers in European member states. And we have learned from our farmers how they are struggling with the situations. And that's why the way forward, that we need to join our effort to support our sectors, to support our livestock farmers, to improve the way that they produce, to improve in terms of livestock productions, animal health, and so on. And that's why the work of EuFMD and also all the scientists who joined this meeting today will support our farmers. And also the work that you are working in the scientific communities will make an impact once the science and innovations and technologies that all the scientists are working can be scaled up, can be applied, can be replicated at the country level, and we can make more impact. And also we'll see more result. And And I'm sure that I would like to inform you on 4th of November, just next year, FAO will also launch the second phase of the pestis ruminant or PPR global eradication program that we work closely with our partners, World Organization for Animal Health, to make sure that by 2030, we will achieve the eradications of PPR. And we need your support, and we need your engagement. And also, we would like to raise the profile of the livestock sectors, the work on animal production and health. On 7th of December, we will have the high-level special events, FAO development and emergency in actions, sustainable livestock transformation. We will echo our voice that the work on animal production and health will make an impact at the country level. And we will also bring the work of the EuFMD to showcase at that event as well. Thank you very much. And also hope to see you in person sometime. Thank you for your invitation to talk with you today, and wish you all the best.
Thank you. Thank you very much, Tanawat. I know that you want always actions from us, and the action that I propose now is really to bring to your attention, to the attention of FAO, the attention of the CVO, and to the attention of the EuFMD work program, the outputs that we have out of this meeting. So how we really can apply the lessons learned from the different research outcomes that we heard during these days. So thanks for your time again, and thanks also to the panelists and the chair and the moderators. Giancarlo has probably to leave now, I think. Yeah. So thanks to you.
My apologies.
Oh, that's your apologize. And we have our last coffee break now.
Okay, so hello everybody. Welcome back from the coffee break to our last round in the surveillance and control session. I hope you are still alive and energetic enough to follow these remaining 6 presentations. At least our online audience is energetic and interested. We had more than 500 viewers just before the break. Okay, so we will start with Sarah Mielke. She's a biological scientist and works on data design and analysis, and will present to us a talk on is FMD virus serotype C extinct? What can data tell us? Sarah works as the US Department of Agriculture, Animal and Plant Health Inspection. Please, the floor is yours.
Thank you. Good afternoon, everybody that's present, and also good day to everybody that is joining us virtually. I'm happy to be here to share a project that was completed with Doctors Donald King, Melissa McLaws, and Marta Remenge, in which we tried to tackle a tough question about the potential extinction of foot and mouth disease virus serotype C. So in this study, we focused on regional virus pools 1 through 6. We did not include regional virus pool 7 because within the data set that we were using, we did not have data points. And also, there have been some descriptive analysis about the presence and absence of serotype C in that region. But that doesn't mean that we wouldn't in the future be able to incorporate that into this framework. So what I want you to look at here, we're going to look at the data sets that we did use. I'm representing the regional virus pools with the large circles. Within each of these, you see the small dots. Those represent foot and mouth disease virus detections. We pulled 2 data sets, one from the Purbright World Reference Laboratory. This data set spanned the years 1942 to 2021 and had nearly 22,000 total samples. We ended up using this primarily to look at the historical detections of serotype C, which occurred in all of the regional virus pools except for pool 5. The next data set we pulled from the FAO Foot and Mouth Disease Dashboard. This set spanned the years of 2012 to 2020, and we had nearly 19,000 total samples. We ended up using this for our analysis because of the large number of samples within the span of years that we were looking at. So using this data set, we scaled it in 3 different ways. First, we have unstratified data, where we applied 2 assumptions. One assumption was that the prevalence of serotype C would remain the same from year to year. And the second was that a country within the region could represent the entire region with its data points. Next, we stratified by country so that we looked at each individual country within the regional virus pool. And lastly, we stratified by both year and country, allowing us to look at the data for each year and for each country within each of the regional virus pools. The last thing I want you to notice is on here we're using a livestock density map, and this is showing our cattle livestock density, with the bright yellow indicating the higher density of cattle. Now we'll go and look at some of our data output. Today I'm only sharing with you for regional virus pool 3, just because of the time constraint. So we wanted to look at 2 detection measures if serotype C were present. Our first one is detection probability, and we wanted to see when the data would support a 95% detection probability at specified prevalence thresholds, which we set at 1, 2, and 5% for our analysis. We did this across each scale of our data. So this would mean that we would be 95% sure of detecting serotype C if it were present at one of these prevalence thresholds. So moving from the left to the right, our first plot is unstratified data. So this is where we have applied both of our assumptions. And you can see on here that at 1, 2, and 5%, the data is supporting a 95% detection probability, which is indicated by the red line across the top. Moving into our second plot, we have stratified by country. And starting in the top row, that is our 1% prevalence threshold. And we can see there that we just have a couple of countries that have data that will support that 95% detection probability. And as we move down the plot and the prevalence threshold is increased, we see that there are more countries where the data will support that detection probability. We also start to see that there is gaps in the data. So you can see where there are countries that never have data that will support detection probability at 95%. In our last plot, we have this stratified by both country and year, and we're seeing the same pattern. So on the top row at 1% prevalence, we have one country with a few years where the data can support that 95% detection probability. And as prevalence threshold is increased, again, we start seeing more years and more countries where we're having 95% detection probability based on the data that we're using. We also start seeing a lot more in detail about those gaps, where the information is coming from that actually ends up feeding into that unstratified plot at the far left. So the other measure that we used was detection capability. So this was where we wanted to see, we know that we are achieving that 1% prevalence threshold for some of the countries. So we want to see what is the lowest prevalence we can detect with the samples that we have with 95% confidence. So this information is being shared with you through the Trello board, and you can also access it on the QR code there. It's our interactive document. In this interactive document, you'll be able to look through all of the results, how we set up the analysis, look at our assumptions that we applied for each scale of the data, and what our interpretations were. We also have questions that we've directed at you. So when we started this analysis, we knew that the data would not be the only thing that would inform our understanding, but that other factors within the country or within the region would also provide us information for how we want to apply our assumptions and how we might want to interpret that data. So here, this is the main reason that we came to the conference. We wanted to share this with you as subject matter experts in foot and mouth disease virus in these different regional pools. We'd like you to go in, explore that document, look at our questions that we have proposed to you, and if you can provide feedback back with, and make sure that you indicate which regional virus pool you're speaking about and what your name is so we know where that information is coming from, we would really appreciate that. So those are the questions that I've set forth for all of you, and during the panel, I'll take your questions. Thank you.
Thank you very much, Sarah. Our next presenter is Rosemary McManus. She is a postgraduate student at Glasgow University and interested in application of drones, machine learning, thermal images in animal health. So please, looking forward to your presentation. Thank you.
Thank you so much for the introduction. I'm really honored to be here today to discuss my research about investigating gaps for novel animal health surveillance data within Scotland. So my research focuses on what we can use thermal and drone-acquired imagery for in animal health surveillance and how that may be useful not just at the farm level for disease detection, but also at higher policy levels. Today I'm going to speak to you about the qualitative research we've been carrying out to ascertain what issues and problems may arise if a surveillance system using such data was to be implemented. This is an ongoing project, and I'm presenting preliminary results about our investigation into gaps for novel animal health surveillance data within Scotland. This project had two main aims. The first was to map the data journey of current animal health surveillance in Scotland to identify stakeholders and gaps for novel data. And the second was to introduce this novel data into a gap in this data journey and identify areas of potential data friction through stakeholder interviews. So what do these terms, data friction and data journey, mean exactly? They're terms that come from the field of critical data studies, which has evolved to explore the unique cultural, ethical, and critical challenges posed by big data. A data journey maps data as it flows between different sites of practice, and data friction is a term that's used to describe the complex factors that come together to slow down or restrict data generation, movement, and use. I hope to show you today why this methodology is a useful tool to have in our arsenal when it comes to dealing with large data sets. I've used the terms data consumer and data producer to categorize stakeholders, and I've also provided some information about data governance, which aims to ensure the availability, usability, consistency, and regulatory and legal compliance of data and data systems, which ultimately means that more data can go further. While this project has mapped data for both peacetime and outbreak situations, given that this meeting is concerned with FMD and other fast diseases, I've supplied you here with our map illustrating the data journey of animal health surveillance from farm through to policy in Scotland and beyond that would occur in the event of a foot and mouth disease outbreak in Scotland. Data moves through multiple sites of practice, and arrows show the direction of the data movement. When there is suspicion of FMD in Scotland, the person who discovers it informs the Animal Plant and Health Agency. The APHA then send a veterinary inspector to the site of suspicion, where samples are taken and sent to the National Reference Laboratory at Purbright for confirmation of disease. If disease is confirmed, results are communicated to the Chief Veterinary Officer for Scotland and the CVO for the UK, who then notifies international bodies, namely WOA and the European Commission. Local authorities, private veterinarians and farmers are also informed of results and of control measures to be implemented. EPIC, which is the Scottish Centre of Expertise on Animal Disease Outbreaks, advises the Scottish Government and other stakeholders on the risk of spread of the disease and on how to control this risk. Data on animal movements are collected by Scot EID and are used by EPIC and beyond. So, having mapped out the current data journey that exists, we inserted novel syndromic surveillance data into an identified gap, with data collection being carried out by an industry stakeholder, an agri-tech company. This seems to be one of the most likely ways this data will be collected. So using this projected map and stakeholder interviews, we identified potential areas of data friction that could pose a problem if this data was to be used not just by the farmer, but further along the data journey for disease detection and policy. So, from a veterinary and epidemiological perspective, we identified areas of data friction, moving data from the technology company into government institutions. Veterinary stakeholders at EPIC and the APHA expressed concerns about sensitivity and specificity of thermal and drone technology, the cost of such a system and who could bear the cost, data quality and trustworthiness, and how many participating farms would be necessary to enable meaningful data analysis. Speed to detection also emerged as a question. Could a system like this truly speed up our detection of FMD, whether by identification of suspicious FMD lesions or by detection of herd or individual animal temperature increases. Some stakeholders thought that a system like this might be helpful in providing data about spread of disease, but that it wouldn't be likely to beat the current surveillance system for early disease detection. The second area of data friction identified relates to data management and curation. I focused here on data flowing from the farm to the technology provider, but many of the points are applicable at multiple stages of the data journey. So, how do we get this data off the farm? Internet access can be an issue in rural areas. While cities tend to have fibre broadband, this is yet to make it to rural areas in Scotland. And where and how do we store this data when it is collected? Will it be stored locally, or is it possible to upload it to a cloud-based service provided by the industry partner? Image files tend to be quite large, so will it be possible to keep this raw data for historical interrogation, or do we convert it to CSV format for ease of transfer, thus losing the original raw data? Which leads to the last concern. Will it be possible to collect this data in real time or near real time? And what infrastructure would need to be put in place to facilitate this and real-time data analysis? The final areas of data friction we identified relate to legal and regulatory issues. Starting at the farm, who's going to collect the data? If it's to be drone acquired, will it be necessary for the farmer to gain the qualifications necessary to fly a drone? Or can we create an autonomous system to collect the data? The current UK Civil Aviation Authority regulations forbid flights where the pilot doesn't have the aircraft within their visual line of sight. So truly autonomous flights are out for now. Drone regulations also require a person to obtain certification to demonstrate adequate knowledge and skill with regard to flying, drone regulations and safety. Next, who has access to the data and who has the rights to the data? Many stakeholders suggested that the farmer would own the data collected on their farm. But database rights in the UK denote that intellectual property rights would lie with the agritech company if there has been substantial investment in obtaining, verifying, or presenting the contents of the database. Will personal data be collected? County parish holding numbers are used in Scotland and across the UK to identify farms. And in the UK and Wales, this number is considered personal data. And so a farm or farmer, sorry, excuse me. In England and Wales, this is considered personal data, so it's not shared. But in Scotland, it's not considered personal data. So will we be able to protect the farmer from being identified? Finally, stakeholders expressed concerns about data sharing. Surveillance institutions in Scotland and the UK have data sharing agreements in place, and these could be put in place, but will these adequately protect the farmer? Agritech companies in the US have been sued in recent years by farmers and consumers using their technologies, as they were allegedly using collected data to aid producers in inflated price fixing in the poultry and pork industries. So, I want to thank you for listening to me. Hopefully I've given you a good overview of our work to date. I think the underlying theme of data governance is hugely important and can foster data sharing and use, both nationally and internationally. I've left a lot of open questions for your consideration, and I'd be interested in hearing your views on potential solutions. And lastly, I just wanted to extend my thanks to the University of Glasgow and my collaborators. Thank you.
Thank you very much, Rosemary, for this really interesting presentation. As usual, we will have the questions at the panel discussion. So we will follow this nicely in thematic ways by Michelle's talk on the potential use of drones in surveillance for fast diseases. Michelle is the Deputy Director of the Qimron Veterinary Institute in Israel. Please.
Thank you very much to the organizer. I'm very happy to be here. And we are going to talk today about the potential use of drones in surveillance of fast diseases. This work has been done with cooperation with Ezra Rosenblut, Dr. Ezra Rosenblut, which is sick now, and I hope that it will come back to us swiftly, and Boris Eventov, which is the director of the Golan Galilee district in Israel, in the veterinary services of Israel. So we have a lot to talk about, and we have very few minutes, so please fasten your seatbelts and be prepared to take off. with a drone. Well, how can drones assist in fast disease surveillance and prevention? We have taken the lumpy skin disease as an example. As you know, surveillance is very important, and our duty is to keep animal and public welfare, and animal and public health, and animal welfare, and avoid economic losses. So when we are speaking about stable or a pen, it's very easy to have surveillance because you can put animals on a line and to see if they have clinical symptoms. But it's another story when we talk about remote herds in the field, like you can see there. And the use of the drones are mostly with remote herds. This is the map of lumpy skin disease in Israel. Between 1988 and 2007, we had 3 outbreaks that came from the south. 2012 and 2013, we had a huge outbreak with more than 160 farms that came from the north. And in 2019, that's when we have done this survey. We had the lumpy skin disease that came from the east. Well, what are the advantages of the drones, the using of the drones? There is a possibility of scanning large areas, as you can see here in this video, in short time. This gives reduction of working time searching animals in the field. We have the possibility to find and reach remote hidden herds or remote and hidden animals. And here you can see that the animals, they have no fear of the drone. They are observing the drone at first, but afterwards they are continuing their activity, as you can see here. It's like a big fly for them. Well, other advantages are to overcome obstacles like water, as you see here, or fire, or impassable areas that we cannot reach, or to overcome a dense vegetation that we cannot pass. And also we can overcome minefields that are common in the Golan Heights in Israel. Well, we can use a drone for counting animals in herds, including the morbidity of the animals or the mortality of the animals. And it's very important when we are speaking about a fast disease, when you put a quarantine on the herd, we can do census or surveillance to be sure that no animal escaped from the quarantine. We can track animals in movement. We can see if they are limping or not. And it's very useful in surveillance in an area with a large number of farms. So you keep biosecurity. You don't have to jump from a farm to another, and you can keep the biosecurity. Like you can see here, a lot of farms, and you don't have to enter to the farms. You can just send a drone. Or surveillance in areas where the cooperation is missing, like in some villages that the farmers don't want you to come into the village. His grandmother is in the hospital, or he doesn't feel well, etc. So you can, he can tell you, come tomorrow, come the day after tomorrow. You can tell him, okay, I'm sending a drone now, today, and I am not entering your farm. We have to think also about the safety and welfare of the staff. For example, physical difficulties to reach the herd, or in hot or cold weather, or in snow, or in rain. It's the welfare of the staff is important. We have some, in the desert, we have some issues of dust and air pollution, so it can be useful. And we can avoid animal aggressions. We can avoid arthropods, like flies, like mosquitoes. We can avoid snakes, scorpions, and other dangers in the field, and also, as I mentioned, mines. Well, here we can see that we found an animal with lumpy skin disease. You can see very well the lumps and the shape of the animal, and we can close up, like you see here. And you can see very well the lumps on the neck, the size of the lumps, etc. And you can see, we can have the possibility also of aging the lumps. Here you can see old lumps. They have a crater in the middle of the lump. And in the top, you have new lumps. So you can know the age of the lumps. And you can also identify the insects. Like here, it's stable flies, Stomoxys calcitrans, that are passing the lumpy skin disease. We have the possibility to identify the animals by the numbers. We can zoom on the number, and it's very, very useful to recognize the animal by their ear tag. And as I mentioned, when we have a foot and mouth disease, we want to know if there are still symptoms in the farm. We can send a drone. We are not obliged to enter the farm and to corrupt the biosafety. And we have high quality documentation by photos and video, which is important. There are some disadvantages of the drone. In exceptional weather conditions, it's not possible to use them. In typhoon, in very strong wind, we cannot use it. Short time of battery. A drone can be very expensive. And if you lose it, so it's expensive. It can enter a tree and you can lose it. You need license. You need permits in some areas, like army areas or airports. And you need skilled operators. Other examples that we thought about were eye lesions or episodic hemorrhagic disease, FMD, as I mentioned, PPR, POX, and also HPAI, as we can see here. And also, we can locate wounded animals in a war zone. We can locate wounded or dead animals in war zone, and we can avoid hazardous substances or aborted missiles due to the drone. Other ideas like poisoning, following rabid animals, maybe thermic camera, maybe. And I want to thank Ezra and Boris and farmers who cooperated, and also the animals. And Ezra highlighted that no animals, aircrafts, or veterinarians were hurt in the production of these films. Thank you very much.
Thank you very much, Michel, for this very interesting and entertaining presentation. The next one will be Antonello Di Nardo from the Purphyre Institute, and he will talk to us about the OpenFMD, a data sharing and analytical portal to enhance genomic and epidemiological surveillance of FMD. Please, Antonello.
Thanks, Carsten. I'm going to touch on the most discussed topic of this open session, I believe, data sharing. And I'm proposing a solution that might not be the solution, but something we are working on, we have been working on in the last couple of years, and is moving on that direction. So as we all know, either during peacetime or during disease emergencies, the devising of control policies, the effective control policies, really rely on accurate data collection. And as timely and openly, those results are shared between or within the research community and between international organizations and governmental agency, as fast and more focused those control policies can be applied. And of course, these control policies can be more cost effective. From Angus and Kit today, we have learned that there has been a lot of flourishment, development of analytical dashboard, platform for data sharing, and this has mainly been done for human diseases like Ebola, seasonal influenza, and COVID, for example. And I was quite interesting to know that even a network of poop dashboard has been developed. And the thing is that because investment, huge investment, a huge effort has been put for human disease on data collection. Unfortunately, this has not been translated to veterinary science. And for FMD, for example, we are still relying on biased data. And not only the data are based by the sampling, opportunistic sampling, but as well, if data exist, those data, for most of the time, are shared between or from different organizations, and data management, the data integration is really difficult. And of course, in this case, the problem is that most of these data are underutilized or are not reachable in a way. As Angus proposed previously, our approach is try to integrate in a most comprehensive way, in a centralized approach, all the information for FMD in an online repository. And this online repository is pretty much focused on and widely targeted for a range of expertises, from people that want to discover about dynamics of evolution of the viruses space-time, someone that really want to know about vaccine protection and efficacy, or mainly for a government and agency organization that are really focusing on the risk management and policy making. Our approach is basically using a modularized system where we integrate and we integrate all the information in the database, and we divide application through modules that are pretty much focusing on bioinformatic pipelines and dashboard that are all integrated through APIs. This modularized approach is quite robust because it enhances data management, enhances the expandability of the system, and of course can rely on the security and the stability of the system. I'm gonna present a little bit of what we have done on 3 apps that we have been developed. I'm not gonna, for the interest of time, I'm not going to show you a demo of this app, but just a brief discussion. The first one is pretty much the most comprehensive database of FMD genomes that we have at the moment, and pretty much including all the public data that are available in GenBank, plus the data that we collect through the FMD surveillance work of the FAO in the WRL, and those are unpublished data. It's fully curated sequences and include as well metadata, and is linked to the dashboard where the user can filter whatever information they want to have and see the information in the dashboard, and as well download the sequence in itself in either FASTA file or CSV. And at the moment it's just updated up to 2018, but we soon update the entire database up to 2022, of course. The second one is an app that is linked to the database directly, and is an analytical dashboard that is pretty much provide external users the capability of generating what we generate in the WRL as genotyping reports. It's a simple and guided interface where basically user is guided through the entire pipeline, from the uploading of information of the sequences in the FASTA file to selecting those sequences. The pipeline is annotate, curate the sequence that is being inputted by the user, genotype then annotating, so pretty much discovering which kind of serotypes, strains, and whatever lineages is included in the sequence that is being inputted by the user, align the sequences, generate the maximum likely phylogenies, provide the interactive tree as a visualization interface, and of course has the capability of generating the report in PDF format for the purposes of the user. And yeah. The third one is is an app that is more oriented on the, let's say, risk policy maker or risk manager. Is a pretty much a what-if scenario that is basically combine expert opinion with empirical data on vaccine matching to try to provide some parameter of risk and vaccine coverage from evaluating vaccine strain and identify high priority vaccine from a list of vaccine that is present in the dashboard. Is the pragmatist, that I'm not sure if anyone knows about that. We port from the Excel file to the more interactive dashboard in that way, and the user can modify whatever is present in the dashboard as regional risk of FMD incursion from the data that is present in the vaccine matching format, and the output is pretty much a ranking risk of the prioritization of the vaccine and the coverage of the protection conferred by the vaccine that is going to be tested in the app. So the idea behind those resources actually the one of democratization of the information through new technology and providing this new technology to the benefit of all the entire community, regardless of the expertises that those users are, well, the user using the app, from modelers that want just download CSV file or connect the database through APIs, or a risk manager that just want to understand the risk of their country and if the vaccine that is protecting, that they're using in the country is going to protect the next incursion of FMD. Just two things is that the entire portal is still under development, so we are put the entire system in a staging environment, and we hope to migrate to the production environment in the last couple of months, hopefully to release to public by the end of this year, beginning of next year. And I want to stress that the system that we develop is generic by design. So with minimal modification and a little bit of parameter tune, it can be adapted to any fast of disease, any disease, transboundary disease similar to FMD. And it's quite powerful because, I mean, so the same system can be ported for PPR, for example, ASF, or lumpy skin disease. So this is a work in progress, and watch this space, watch that space. This has been a quite collaborative work that has been going on between different groups in Purevsuren, for example, because we sequence a lot and we curate the data and see the sequence data. We develop the database, we develop the APIs bioinformatic pipeline, and the system in itself. App Interactive, a company in New Zealand that pretty much did a fantastic job for developing the dashboard, the visualization part of the dashboard in a very, very simple, intuitive way. And of course, EuFMD that give us financial and technical support, and of course, feedback through the development phase of the system. And I'm going to thank you for listening, and that's it.
Big thanks, Antonello, for this really interesting talk. At least I would have a lot of questions, but we'll have to wait until the podium discussion. Our next talk will be from Dr. Bolotuya Purevsuren from the WAR office in Thailand, and she will talk to us about the digitalization of FMD datasets. We can already see you.
Oh, thank you. Thank you, Mr. Chair.
And hear you.
Can you hear me?
Very well. Yes, perfect. Thank you.
Good afternoon, and it's my pleasure to participate in this event. I'm just greeting from Bangkok. And I'm here today. Would like to present about digitalization of the FMD dataset using the interactive visualization tool on CCFMD portal. And very few words about CCFMD. As you may aware that CCFMD, it's FMD, Prevention and Control Platform for Southeast Asian countries. And we have 12 member countries, 10 of them is ASEAN, and plus China and Mongolia. Actually, this platform established in 1997. Then recently, we launched CCFMD portal, which is web accessible in digital resources, and it consists of different digital resources for FMD, and also the FMD situation dashboard and CCFMD roadmaps and other events. And I will try to demonstrate about this FMD situation dashboard because CCFMD portal, my colleague has already submitted this session, and it's in poster. Maybe you can explore that one. What we have done here is we just aimed to analyze our archive data because we have our own regional information system, and also we have a lot of country reports from countries and manuals and other stuffs. And I mean, we have a quite of the big data in this region, and we just wanted to gain insight about a huge amount of data, and wanted to present the CCFMD data in interactive and dynamic way. And of course, it can be extended. And also, it's not for only FMD. And also, we are planning to extend it maybe LSD and ASF for the further development, and encourage to use available data intelligence tools. For starting point was we just created the simple Excel data sheet, and we all mined data from ARAHIS, WAHIS, country reports, and also national and reference laboratory reports. All this based on the FMD. Actually, this data set covered 2019 to 2021. However, recently we updated in 2022. And you know that they have many tools available for data intelligence and analytics, which we use in an interactive environment to explore the big data set. However, we just choose this Power BI because it's a Microsoft platform, and also it's, I would say, the sort of integrated Excel, and it was really easy to use and to link some query among our data set. That's why we just developed our dashboard by Power BI. And this is the interface of the Power BI desktop. And you can see this right side gives you a lot of visualization tools. And also there is the fields that I imported and linked for this. And let me try to demonstrate. I hope it will work. And this is the first page, is our dashboard. We've developed several pages, and we give them to allow filtering the data, report the year and the country, and other different panel options. And let me do some example here. I hope it will work. Does it work? And for example, maybe 2021, Indonesia, we had the new incursion in, sorry, it's 2022. And Indonesia has a new incursion. It was big epidemiological change in our region, and was, okay, oh, sorry. Then, at the next page, recently, Power BI also developed the heat mapping, and you can see this is heat mapping is already in Indonesia. And in this page also, we just added one more slicer to filter the information by monthly cases. And then you can easily to filter what kind of the information you needed or which month. The next page, yes, we just simply use the FMD temporal distribution. And you can see from here easily to observe some seasonal pattern is observed in the member countries. For example, in here it is 2022 and 2021. Then those, we just put two page for the genotype information. And those information is mined from this reference lab and also the national laboratories. And it gives also the arachis in our data. And how was the lab performing those samples and how many of them is typed and not typed, and the kind of the classification we already have in our data set. For here, I just would like to emphasize that those is only the available data. Of course, I should confess that still we have a lot of missing data from member countries. This is FMD genotype information. I just want to emphasize here, you can see the one is toolkits. It gives you information about the scatter plots, and also here this genotype and which subtypes or serotypes, I would say, the dominating in our region. And you can see this India 2001 is still dominant in our region, and A type is still reporting in some of the endemic countries. For the left side, also the isolated, or maybe it's the confirmed cases from the lab, and we try to present here the special distribution of the confirmed cases. This is the affected species, and also it's based on the data from the member countries, and mostly cattle and buffalo effects in FMD in our region. And I think this is the last page, PCP stage, and you can see here there's Indonesia is free from FMD. Unfortunately, FMD free status is suspended in this year. I'm coming to the conclusion. I'm just looking at the time I have to finish, maybe. You can see that's the quite interactive dynamic visualization tool, and its end users is very friendly and display epidemiological patterns and trends and range of the filter options. You can make your classification, and also I would say that the CCFMD portal is only developed for the member countries. It's the secured access, and whenever you update your Excel source of the data, Power BI will be automatically updated. That means it's the real-time visualization tool, and also you can access any devices, even it has the mobile application. And I think that's all from my side, and thank you very much for your attention.
So thank you, Dr. Bolortuya. We are looking forward to having you on the panel in a few minutes. And the last talk of today and of our open session is from Polly Compston. She is a PhD student at the Royal Veterinary College, and she will talk to us about barriers to effective FMD vaccination in Nakuru, Kenya.
Thank you, and thank you to everyone here and online for lasting until the end. I'm really glad to be talking today about my PhD project, which has been looking at vaccination in Nakuru County. And I have to recognise that this work is not just mine. I've got many co-authors, and I have to acknowledge everybody in Kenya, conscious of being a European in Europe talking about Kenya, whereas so many people have been instrumental in this work, and especially during a pandemic. I'd also like to acknowledge Farming Systems Kenya and the University of Nairobi for collaborating, and of course my funders, the RVC, the Purevsuren Institute, and EuFMD. So this work's based in Nakuru County in Kenya, where FMD is endemic. There's a large dairy industry, and most of that dairy comes from smallholder farmers. So they're really important stakeholders in any sort of disease control program. And I'm always coming back to this cow, how do we get the right vaccination to the right animal in the right place, delivered by the right person, at the right price point? So I wanted to have a look and do some mixed methods research. First of all, some qualitative work, well, they were done at the same time, but some qualitative research looking at the vaccine value chain, describing it and identifying challenges within it, and then also some economic analysis to understand the value of vaccination in these smallholder farms, hoping to bring together those two streams in order to develop recommendations for disease control. I'm going to whiz through my methods. The qualitative part of the project was through key informant interviews, which were done iteratively over quite a long period of time with a wide range of stakeholders. These were then used to describe the vaccine value chain, and a thematic analysis done to identify challenges within the chain. And then a cross-sectional survey in smallholder farmers who were in areas who had experienced FMD in the previous 12 months. And the data that I'm going to talk about today was from farms that had experienced FMD, mainly. These data were then delineated into household profiles using multiple component analysis. And then from there, some economic partial budget analysis and a cost-benefit evaluation. So I'm going to present the results of the economic analysis first. And I mean, please, this is slightly preliminary results, so I've put a, please don't take a photo, badge on this. But designed a 10-year stochastic model, modelling 3 vaccinations a year, public vaccination, essentially scaling up the vaccination programme that already exists in Nakuru County, Kenya. And from there, came out with a benefit-cost ratio of about 4. So for every Kenyan shilling spent over those 10 years, you'd get a benefit of 4 back. And although that's preliminary data, I'm fairly confident that that's an appropriate range. But if we start to look at this on a household level, I said earlier that I delineated 3 household profiles. And so profiles 1 and 2 were quite similar in terms of number of cattle, in terms of their production, but different in terms of the importance of cattle to that household. So profile 2, compared to profile 1, gained much more of their household income from their animals. Profile 3 were much lower input systems. The people who self-identified as pastoralists were in this 3rd profile. And there's really 4 things to take away from this graph. The first is which shows the partial budget analysis. So this is the marginal benefits on a household level of vaccination. The first is how variable these distributions are, and that shows how different it is by household, the impacts of an FMD outbreak. So some households will be much more affected than others. For profile 3, the median benefit's much bigger, but that's partly a function of how many cows. So if you do this on a per cow basis, it comes down a lot, and that will influence how much they've got to spend more because they've got more cows to vaccinate. And then looking at profiles 1 and 2, the median value is quite similar for both households. But if we put a gross margin on that, so that's an annual indicator of how much the household is making from their animals, you can see that the benefit in profile 2 compared to that annual income from the animals is much is much greater. And I think that's important when we're thinking about motivation to vaccinate. So you'd think, great, let's vaccinate. It seems like it's profitable. But looking at the vaccine value chain, and we can see that multiple challenges exist within it. So this is the result of the thematic analysis. On the left-hand side of the screen, you can see the challenges, the themes that came up from the challenges, and these sort of lead to this nexus of difficulties that result with vaccination. But of course, it's not this simple. It's a big, interrelated, complex web of different cause and effects, and these things are iterative. So I'm just going to pick out a few of these. So firstly, a big theme that came out was devolution. Kenya has a devolved animal healthcare system where surveillance occurs at a national level and animal disease control occurs at a county level. And budgeting for animal disease control occurs at a county level, whereas the strategy is defined at a national level. And this decoupling means that it undermines the vaccination strategy, because local political interests can determine how much budget is given to vaccination, and that means that it can be less than is, which means that it doesn't necessarily align to the strategy. It also means that each county does something different. So you may get counties vaccinating at different times, even when communities are really close together. And so again, you've got an undermining of an effective vaccine strategy there. Unclear processes and roles. So in particular, we found that farmers struggled to tell the difference between paraprofessionals and vets, and also public and private services. And this meant that although they were seen often by other stakeholders as passively waiting for free vaccine to come, actually it was because they trusted the government vaccines. They knew that when the government came to vaccinate, it was the right vaccine, it was part of something official. And this difficult infrastructure of different sorts of animal health provider, they found difficult to navigate, even though some of those health providers could provide effective or, could provide vaccination. And so this means that the sort of private sector wasn't as well integrated as it could be. And then finally, and I think this is important as well, this project did not look at vaccine quality, and that's not something that I have any comment on. But sometimes the vaccine didn't appear to work, and there are lots and lots of reasons why that could be. But what undermined farmer trust in vaccination wasn't necessarily that it didn't work, but it was the way in which investigation about it not working was communicated to them. So either they told somebody, but no one took samples, or maybe someone took samples and they didn't feel that that information had been communicated adequately back to them, even if the people who had done the investigation felt that it had done. And I think that's sort of an important thing to think about. So just to sort of try and draw quite a lot of things together there, vaccination programs definitely have beneficial impacts for farmers, and definitely for some farmers it's really important. But this economic isn't enough. It needs to be situated within these socio-political systems, and there needs to be sort of process change within them in order to make vaccination programs effective. Budgets need to be consolidated and align to strategy. We need coordination mechanisms across geographical areas and across different sectors. I think aligning, it's everything we've been hearing about, aligning disease information with vaccination and making sure that all stakeholders are involved. Information often flows away from farmers. It's really important that it flows back. And that things are transparent. It doesn't always matter if a vaccine doesn't work, as long as we can all work together to understand why and make it better next time. So with that, I'd like to say thank you, and thank you for listening till the end.
Thanks a lot, Polly. And yes, we will immediately start with our panel discussion. for which you also have to put up with me because I'm not only the chair of this session, but also the moderator of the panel. After the contract break, I'm asking. Okay, so thanks a lot to all of you for these really interesting, inspiring presentations. Maybe the first question from my side to Michelle and then also to Rosemary. I think the drones were we all liked the presentation and the use of it. What I had asked myself, was there any issues with privacy and data protection in the use in Israel? You said if farmers refuse access, we just send drones above the farms.
Can you hear me? Yeah?
Yes.
The veterinary services are responsible for the health of the animals. And if the herd, of course, you cannot enter to a house or to a private place, but if the herd is remote and is in the field, so you can send a drone, there's no problem with this, no, with privacy or something, there's no problem.
Okay. That's interesting, and so this is public
yes.
Domain, public property, okay. Have you explored other uses of drones, like in wildlife surveillance, maybe sample transportation or vaccine transportation in remote areas, things like this?
Yes, there is, the wildlife is under the responsibility of the Natural Park Authority in Israel, and they have a section with working with drones, and they have a special presentation about this. Maybe next time you can invite them to give the presentation. They have a very nice presentation about how to, but it's not diseases. It's how to count animals or how to track animals, or rare animals, etc. But not diseases.
Okay. Okay, very interesting. But I'm sure there's more questions about this from our audience. So maybe leaning on to Rosemary about this. Like based on the stakeholder interviews, could you kind of depict to us a scenario of a Scottish farm using thermal imaging and sensor-driven surveillance, and how is this accepted by the farmers, and also how does it feed into the larger system of the veterinary services? So we've seen you have involved them. So how, what can be, what will be next steps, and what will maybe you present us at the next or in 10 years open session on this?
So I think we can see that there's already sensors on farms. Many people don't think about it so much, but we have wearable technology monitoring animal movements and animal behavior to inform about maybe estrus, things like that. So they already exist on farms. So I think farmer acceptability is quite high to use these types of technology, but we need to, for thermal imaging particularly and for drones, I think we need to check the validity of whether these technologies can produce data that we can actually extrapolate decisions from and make decisions from. There are some technologies being used in Sweden to detect subclinical mastitis using thermal imaging. So it does exist for maybe production diseases, but not quite yet for exotic diseases.
And probably the question is also to make available the farms. They might be used for like on farm or in the sector, but not for
so
for
at the
wider
moment
use.
this data stays on the farm or with the agri-tech companies. So we need to figure out how we can share this data and who can have access to it and what type this data needs to be, what kind of format it needs to be in so that it's actually usable at multiple levels, I think. Yeah.
Okay. Yeah, very interesting. To Sarah, maybe a question, what can we learn from your initiative for disease eradication schemes like Rinderpest and PPR? So what needs to be observed in terms of how sure are we about the results and kind of these uncertainties on the global level?
Well, I think for using the FMD as an example, we can look at how we want to apply any assumptions, and look at those factors within different areas that will help kind of inform how we can interpret if we're looking at eradication. Because we know kind of trying to prove that the virus is not there or that one serotype is not there is a very difficult task. So looking at the whole picture and adding in some of that contextual information into the interpretation and how you will apply assumptions is going to be very important. So you can identify where gaps might be and where you might want to target more surveillance or more sample gathering.
Yeah, feeding back those information and try to fill the gaps. Yeah. Okay, interesting. To Polly, maybe question. What is so? This is a research project. I don't know if it's come to an end. What is the next step, or is there a follow up? And I mean, really interesting results. It would be probably very nice to work with the stakeholders to maybe look for interventions and how things can be improved and follow those up. Is there any plans?
Yes, so it's, I mean, it has been a standalone research project, which is currently nearly a thesis. And once it is a thesis, the plan is to develop I mean, I've been working in collaboration with the government vets in Kenya, both at a national and a county level. And so we're going to develop some resources together for different stakeholders, mainly the county level vets, to help to disseminate these recommendations. And I think as well, though, there's something I would like to take forward is quite a lot of these things are more universal. There's some things that are quite context specific, but some of the bigger principles are more universal. And I think there's opportunities to take that forward, even just by encouraging them to be articulated. I think often these more process-led things are not well articulated in our scientific literature, and therefore we don't always learn from them as much as we could.
Yeah. Always nice to see the interaction between our research and application in the field. And it's good if research projects don't stop at the end of a PhD, but are followed up. Yeah, I think that's quite important. We hopefully increasingly learning. Maybe I don't want to take up all the time and ask all the questions. And the last time we have not had the opportunity to ask the audience. Maybe it's we also give you the opportunity. We have 2 more presenters that have not been addressed. So the floor is open for questions from the audience. And I see we have no online questions, so please be active. And Aldo, please.
There are many hands. There are many hands. There are 7 hands, like 4 hands.
I'm sorry, could you maybe repeat that a little bit?
So you looked at detection of type C and you analyzed the data, but in most diseases we would look at antibodies against the disease. Are you aware if there are any studies specifically looking at type C antibodies?
I am not.
Yeah. Thanks, Aldo. I mean, I think it's an important component of this type of work is to think serologically, but I think for certainly the ELISA test approaches, if you're going to use an ELISA test, we know that the tests are quite cross-reactive, and I think we've discussed this previously within the reference laboratory network. Looking for absence of a serotype based solely on serology is difficult, and would require that you would have to analyze the data with quite some level of scrutiny to look for clustering and to give some evidence that the disease is here. And I mean, I think people are probably aware of previous studies that have been undertaken in Africa and elsewhere looking for serotype C, where they find evidence of serotype C circulation, and we don't believe that those are correct results. We believe that they're probably results that they're attributed to the cross-reactivity of those assays. But I think it's a fair point that obviously serologically you get a wider range of samples, and perhaps if we felt that there were high-risk locations where serotype C might exist, I think serology would be a good place to start to target some surveillance activities that would hopefully lead in virus samples so that they could then be tested to confirm the presence of serotype C. But while I've got the mic, maybe just to say another point that stems, I think, out of Sarah's presentation, which is something we don't really talk about that much, I think, regarding FMD, is this concept of the critical community size for the virus. So we know that for FMD to be maintained in an ecosystem over a period of time, there's an expectation that animals are infected, and then the virus is passed from infected animals to susceptible animals, and that's a continuous dynamic that's maintained year on year, and that's how the virus is maintained. And I suppose a question that we came to quite a lot when we were looking at the data was, the assumption is there's a certain amount of mixing in these systems, and if there's mixing, then perhaps it doesn't matter too much. But if there are discrete compartments within any of the endemic pools where we are, for one reason or another, not able to sample, then it's still possible that serotype C exists. And I suppose that's in terms of a remaining gap for FMD, it's understanding whether or not some generic approaches could be put together to proper estimate what the critical community size is for FMD. And it might also have some important impacts on vaccination and control elsewhere as well. It's not really a question, it's more a comment.
Okay, thank you, Aldo and Don. There were more questions. Melissa?
Thanks. Hello? Oh. Yeah, now it works. Thanks very much, Bellaiche and Antonello, for your presentations on the dashboards. Antonello, you called yours OpenFMD, but Bellaiche, I understood from your presentation that the data are secure and confidential. And so I was wondering, what the reason is for that? Are these data really confidential? And would there be a possibility to have, some part of those dashboards publicly available? Nice to see you, by the way.
May I answer, Melissa? Thank you for your question. Sorry, I have some, the reason this time we have a security access for member countries because those all data and other the information is not validated from some of the information is need to be validated and follow active with member countries to finalize. And also the last time we had the NC meeting in Indonesia, national coordinators meeting in Indonesia. There was a lot of partners and other like institutions collaborating with the CCFMD, and we already provided them the access to CCFMD portal, but we are still working on the development, and I hope if there is declared like data protection policy, and also the members are cleared the data, and I hope it would be the public the dashboard.
Okay, thanks a lot for this clarification. We had a question from over there. Yep.
Rita Popola-Pereira from the University of Liverpool, and it's about the drones. So I assume that the drones just collect the images. In terms of diagnosis, do you have trained software to aid you in the recognition of the conditions, or is it solely operator managed? And if so, how close does that drone need to get to the animal, or do you kind of scan the herd and is it fairly easy to detect, or do you need to go like scanning individual animals fairly close to it?
Okay, you can do both. You can scan a very large field to see where are the hidden herds, or if they're carcasses or sick animals, etc. But you can also take the drone pretty close to the animal, maybe 10 meters. But if you have a good drone and a good camera, the zoom, you can zoom very, very close to, and you can get, as you have seen, you can get images, even the number of the animal and even the insect. You can recognize the insect that is on the animal. It's not, the drone won't be very, very close to the animal, but if you have a very good zoom and a good resolution, so you can do a lot of things.
At the moment, you don't have a trained software to aid you, so it's all kind of manual. But
yes, it's manual. For now, it's manual, but I hope that it will be better.
I would probably say that another part of my project tries to look at this. So we're developing kind of an algorithm to try and detect temperature change from thermal cameras, or to try to quantify movement of animals from aerial images as well.
I know, for instance, at least, sorry, I keep speaking about meat inspection. I know at least there's meat inspection software that compares, so by having a lot of standards in terms of the presentations of certain conditions in the software, you can aid in the detection of altered features. So I wonder if it's something like that you could use, where you program how something looks on the outside, so it modulates how the software can aid you in detection, because I can imagine big herds might be a bit hard.
Depends on whether you're looking at individual animals or herds. So, yeah.
Thank you.
Thank you for the question.
Thanks a lot. We had a hand raised here in the first row. Zarko. Okay.
We're about to close.
Stereo.
Two ladies coming. Well, thank you very much for the talks, and they are wonderful. My question or comment is challenged by again by the drones, but also the presentation by Rosemary, and the discussion that we had this during lunchtime, that we have, for the example of Australia, with so many animals and so less vets, and how we can have a good surveillance, how we can cover this. And my question is, have you considered, now we live in the world of modern technologies that are developing every day, and we have something new tomorrow. Have you considered use the thermal visions, both in drones or in big farms, so that you can really scan the herds or the whole farm and animals to detect animals with fever that can contribute for detection of animals with fever, past diseases, or some other transboundary diseases that are relevant for Europe. That can improve early detection and, from the other side, provide confidence of disease freedom. Thank you very much again.
Thanks for the question. So part of the project I'm undertaking at the moment does exactly this, so using these thermal vision cameras on farm, on stationary, looking at dairy cattle as they exit the parlour to try and see if we can correlate their rectal temperature with their eye temperature. And therefore, can we establish whether there's a herd level change in temperature or an individual level change in temperature that might be an indicator for FMD, where animals do develop pyrexia. Yeah, so yes, is the answer.
Okay, we have time for a last question from the audience. James Wood.
Thank you very much. What a fascinating session. I really wanted to ask Polly about the use of drones in Kenya, but I'm not going to. What I'd like to do is ask Polly about how she sees her results being used by her main stakeholders in that part of Kenya to see if that can drive vaccination programs, given the clear benefits, or how can the work be taken forward? Now you've done, come up with a really nice economic analysis, which is so often lacking for vaccination programs, despite the fact that we deal with economic animals so much.
I think there's probably two answers to that question. I think a lot of these, especially sort of quite processy challenges, it can be helpful just to know that they exist. And so I think that, I'm always wary of saying that, if you tell farmers that they're going to save money, they'll change what they do. There's lots of things that I know would save me money, and I still don't. So but I think that by sort of sharing this information, I think it's important for especially policymakers and vets in Kenya and everywhere. Often we group groups of farmers together when they're not as homogeneous, sorry, it's the end of the conference, as we think they are. And I think that's something that comes out of it, this big variation, both in terms of different farmers and in impacts. And so sharing that helps, especially public vets, design strategy, that's more effective. I think as well there has to be some acknowledgement that these political systems there are aspects of these political systems that are difficult to influence. There's other interests at stake and other interests that influence how these programs do develop. So sometimes, and it reflects some other conversations that have been had this conference, let's understand the best that we can do. And maybe some aspects of this, it's good to shine lights on them, because it means that we can say, well, maybe that's an intractable problem right now, and we can concentrate our resources somewhere else. And I definitely want to emphasise, I don't think this is a Kenyan problem. Coming from a country where I think there are also some political obstructions to long-term disease control policy.
Okay, thank you very much. Unfortunately, no time for more questions. Give the floor to Buddha. No?
You conclude.
Okay. Just some concluding remarks from this session. It was really nice. I think we had a good mix, and a refreshingly good mix of papers on surveillance and control. We saw practical use in endemic countries, free countries. We saw the potential application of novel technologies like thermal imaging, drone use, smart farms, 24/7 surveillance on farm level, but also larger sector, maybe countrywide level. We also Evin presented a large scale of different tools, and we saw increasing willingness to share these tools, to make them publicly available. Maybe this is also thanks to the COVID pandemic and the climate of sharing, because quick exchange of data and information was necessary in this situation. So a lot of tools. And we at EuFMD, I think we have a lot to digest from this, and we got a lot of inspirations and probably also ideas for further work and collaboration. So with that, I would like to end the session and our panel discussion, and give the floor to Fabrizio for the final closing remarks. So thank you, everybody.
Thank you to our last panelists, chair and moderators. Ooh. So, happy? How did it go? Good. We have two more things to do. One is the poster awards. You know that we had this voting system for the best poster. And the winner is... The title of the poster is Emergency Toolbox, Digital Solutions for FMD and Similar Transboundary Animal Diseases. And the authors are Costanza De Laurentiis and Ludovic Canela. I have to say that considering that they were those counting the votes, I had to double count, but I confirmed that they were the winner. And we have also a little award for you. Thanks a lot. Now, I want to acknowledge the work done in the past months. And I want to thank some people that managed to make this possible. Before, I just want to thank all of you, the participants here in the room and the participants at home, for your inputs. Then I want to thank the Standing Technical Committee that act for this open session as a scientific committee. Thanks a lot. And I also acknowledge that technical work done by a few colleagues that didn't have the opportunity to come on the stage, but had worked a lot for the preparation of the session, and in particular Kirill, Charlotte, and Sviatko, who is also helping me a lot in other things. Then, of course, the presenters, the panelists, moderators, the authors of folders. I would like to thank France for hosting us in this beautiful city of Marseille, and with a nice pastis. And of course, our partners, and I think that you had the opportunity to meet in person them on the area where we had coffee, and also our colleagues following us online had the opportunity to meet with them or see their space in our app. But now I really want to thank the operational team. So those who manage to find this nice location, to get you here, to follow the testing of the online part, look after the colleagues following online this session, They worried about the bandwidth, but apparently it worked very well. They look after our very nice design, beautiful design, and nice hub. So I would like to ask them to come on the stage, and I will call them by name. Starting from two colleagues, our technical colleagues, two veterinarians that really helped in the organization of the session, following us after the member of the Standing Technical Committee, and looking at the abstract and organizing all sessions. So I really want to thank Etienne Chevan and Mattia Begovoeva. They are behind the poster, but they were liaising with the participants. Come on the stage, please. And I'm really thankful because they did a lot of really a lot of work, and they were just behind the scene. So please, no, come here. Then Alessandra Alviti, who, well, they did everything, but she looked in particular after the app. Elena, you come closer to the stage. We call you all, so. Elena Flores Ruiz, come on the stage, please. I will not go ahead. Tiziano, detto Frederic, who look after the travel. Also, I would like to acknowledge Benedetta, who is not here, and she also helped with all the travel. Costanza Ludovica on the communication side. Enrique is following from home, but Enrique is our designer and look at the overall, also the overall coordination of the session and of course of the design. Special thank to Super Filippo for the overall coordination. I have to say that we challenged him a bit, assigning a lot of work, a lot of tasks, that you did it very well. Then we have other colleagues who I will not mention. They are at home, in the office, and they managed to have the ball rolling, so to prepare for the next event that we have in November and December. So I really thank them for not being here and still working from home. And last but not least, I want to thank Cécile for the... Cécile is looking at the overall logistics, and she's also looking after this great team. I have something personal for you. And do you know how many pillars we have? How many pillars we have in our program? The right answer is from the back. Aldo Decker, 4 pillars. We have our 4th pillar. I will introduce it. It's called Nadia Rumic. Who I have to say, well, I have also a personal present for her. And, well, Nadia really looked at the overall organization of the session, and she's also looking after me every day. So thanks a lot. Last acknowledgement to the Petruzzelli and all the guys that are on the back. Fantastic job. Really nice team. very professional and also very helpful to solve problems, problem solving beyond their mandate. Thanks really a lot. You have met also our colleagues on the technical side. They were on the stage. I will not call them here, but really I would like now to acknowledge the work of these guys, because that is the work that make possible all of this. Thanks a lot, and see you in 2 years' time. Now you can go.